Well, hello everyone. Welcome to the 12th Annual World Stem Cell Summit and RegMed Capital Conference from West Palm Beach, Florida in 2016. This is our second time in 12 years, and the first time, I should say, that we've returned to a city to present our summit, and we are thrilled to be in West Palm Beach again. The World Stem Cell Summit is a global meeting. Over the next 4 days, we'll have over 1,200 attendees from 30 countries, 35 countries at last count, leaders from all over the world in the field of stem cell, regenerative medicine, industry, a major contingent from the country of Japan that is one of the leading countries, regulators, members of the FDA and the NIH. This is the most consequential meeting in the field. Because it is the single meeting that not only presents the ecosystem, but it also provides a voice to patients and patient advocacy and medical philanthropy, providing them a key seat at the table. Before we start, I want to introduce Mayor Jerry Muyo of the city of West Palm Beach. Now, as many of you know, I'm a resident of Palm Beach County. The heart and soul of culture, of entertainment, of refinement, of excitement, right across the intercoastal from Mar-a-Lago, where Mr. Trump lives, is the city of West Palm Beach. Mayor Muyo is one of the leading mayors in the country. And I hope that while you are here, you will enjoy the amenities here, city places across the way, Clematis Avenue for your late night entertainment. We have shuttles for you, and for all of those that are here for the public day and throughout the meeting, the city has opened its doors and opened its arms wide to us. So let me present Mayor Jerry Muyo.
Thank you, Bernie. Well, good morning. I'm honored to be here with you today, this morning, to welcome you all to Public Day at the World Stem Cell Summit at Palm Beach County Convention Center in beautiful downtown West Palm Beach. This year marks the first time the World Stem Cell Summit will make a return to a previous location, and we're very proud of that. We're thrilled and honored to host this prestigious event. And we did so in 2012, and we are happy to have you back here again in 2016. This year's summit has so many new elements, especially this public day, where we are bringing extraordinary experiences and knowledge to the residents of West Palm Beach and Palm Beach County. As many of you know, I'm a former educator, and I am always happy when we're able to bring new learning experiences to our residents. So thank you, Bernie, and your crew for that. I'm in awe by the scientific discoveries made by researchers and doctors from the global regenerative medicine community, some of whom are joining us this week. Who among us hasn't been touched in some way by a friend or relative who is suffering from a disease or a traumatic incident and who might be helped by the use of stem cells? On a personal note, I have a friend who about a year ago fell from a ladder. He's currently a paraplegic, and I know that he is watching the research very carefully in the hopes that someday he'll be able to lead a typical life. Strides that have been made over the last few years are amazing, and I know that there's more to come. As mayor, I have placed an emphasis on economic development. We are not only open for business, but we're open to business. So many biotech companies and organizations have chosen to be here in Palm Beach County. So we think that you all will be very much at home. I hope you enjoy our beautiful weather and take some time to walk across the street to City Place, where there are many, many restaurants that you can partake in. But I particularly want to invite you, if you haven't already been there, to our waterfront at the end of Clematis Street, where we have our Christmas tree, which happens to be made of 600 tons of sand. It is lit. It's 35 feet tall. It's lit, and every 15 minutes, I think, they have a musical extraordinaire where music and lights are coordinated, and I think you'll really enjoy visiting it. So please venture down Clematis Street. Thank you for bringing the Public Education Day to our community. I'm excited to learn about the new medical breakthroughs, meet the NASA astronauts, and enjoy the art and science artwork created by our local school children. Learn, explore, and discover right here in West Palm Beach. Thank you, and enjoy your visit, and enjoy your day.
Thank you so much, Mayor.
Thank you.
Thank you, Mayor Muyo. And so we begin. To enjoy this meeting at all, you need to download the app, because some of you might not realize we have gone paperless. That means there's not an online program. Now, this is the first year we're using a particular app, and you might be surprised or not surprised when I had my own surprise when I opened it, and I saw some of the public day is a little mixed up on the app. But that's okay, because the World Stem Cell Summit, we're all smart people, and we know improv when we need to. So as that gets straightened out, what I'm going to tell you is that everything with the public day is either in the grand ballroom or around the corner in Ballroom C and Ballroom A. You will be able to see what sessions are going on when. The signs are posted outside the door. Now, in this room, everything is live streamed around the world. Here you're going to find lofty talks on all sorts of subjects related to human longevity, art and science. NASA is presenting in the Center for Advancement of Science and Space the incredible research that's going on the International Space Station and how microgravity impacts cells. This is really where a lot is happening. But just around the corner, if you're a patient or you have a family member that suffer from some debilitating disease, you want to find out what's in the here and now. In Ballroom C, we have sessions running parallel to the sessions in this room on different disease indications, on how to join the clinical trial, what you need to do before you seek a stem cell treatment at some private clinic, what you need to know and what questions to ask. This meeting has also been supported by an incredible organization, Hadassah, which you may have heard of, that has over 330,000 members. I believe that's right, around the country. And they have supported this meeting tremendously. We have volunteers and over 300 Hadassah members attending this. There's some special sessions today on reporting some of the great work at Hadassah Medical Center, and also a private briefing that's also, if you're a Hadassah member, that you can enjoy. There will be food, coffee, Danish, and plenty of food for sale at the very excellent little snack bar that's been set up outside across from the Hadassah table around the corner. I hope some of you that are just here for the public day might consider staying for the entire event. And if you go to the registration desk, you can see what the day passes look like for our major conference that welcomes laypersons and the general public as well, that is starting tomorrow, goes into 8 different tracks. Now, the meeting has evolved in our 12th year. This year we do have 8 tracks: discovery, clinical trials and translation, regenerative medicine in the clinic, where we're offering continuing medical education, CME credit, ethics, law, and society, the RegMed Capital Conference, where companies will be making pitch presentations to investors. We have investors from venture capital firms, family offices, networks of investors and angel networks. And this is a very special event and also a how-to event, so how companies can commercialize and even have investment gurus. So if some of you are interested in the field of biotechnology and what to invest in, we have some of the leading stock pickers that are here who understand the industry in and out. We have a track called Hot Topics, Cell Manufacturing, and for the very first time, an equine World Stem Cell Summit, understanding this is one of our experiments, but understanding that if you are a resident of Palm Beach or the state of Florida, the equine industry is incredibly important. It's a multi-billion dollar industry for Florida and the United States and around the world. And the things that they're doing and veterinarians are doing related to regenerative medicine is a whole nother issue. Since our meeting cross-pollinates, you should attend things that you think that are not in your field. You should attend things that you might believe are over your head, but really are not. We try to make sure that all presentations are presented at least to understand the context. All of this is the future of medicine. It's going to impact your lives, the lives of your children, your grandchildren, the way medicine is going to be practiced. This is the future, and that's what you're hearing today. This is an optimistic message. In a sea of negativity, I want you to pay attention to everything that goes here. I want to introduce, just as we move into the session that starts in 2 minutes, around the corner in Ballroom C, it's what patients need to do to understand before they get a stem cell treatment, with Darcy DeFede from the University of Miami and Karen Kruker from Mayo Clinic. These are people that sit as triage as patients call and wonder what they should do and what's in the here and now for them. In this room, we're going to be talking about art and stem cells. And as you walked in, you may have seen the beautiful art, the striking images, photographs from labs of ELUS, who are our next speakers, but also the children's art in the afternoon, the afternoon school sessions of students in Palm Beach County that were inspired by ELUS art, working with the Center for Creative Education, the South Florida Science Museum, and funded by the great nonprofit Primetime, to create something artistic. This is, my friends, what we call STEAM. We've heard of STEM, science, technology, engineering, and mathematics, but together with art, it's STEAM, design principles and economic and encouraging creative solutions. And as I was researching myself about STEAM, I found a quote from a very distinguished professor of education and art, the late Dr. Elliot Eisner. He said, learning in the arts is cognitively a very sophisticated operation. It requires an exercise of imagination. It requires cultivation of human sensibility, the ability to pay attention to nuance, the ability to capitalize on surprise in the course of working on the project or topic, the ability to know when to shift goals when working on something. It is the farthest thing from an algorithm. Much of the problem is the lack of critical thinking in American schools. This has been due to an emphasis on subject matter and on processes that do not cultivate human judgment and other forms of higher level thinking. This is the world of ELUS, of Mark and Janet Hubka. Mark of the company Histogen, a prominent cell therapy company with clinical trials based in California. Janet, who is one of the great artistic minds and proponents of STEAM, and I want to welcome them up to the podium now to give the first presentation of the World Stem Cell Summit.
Thank you.
Have a wonderful time.
Thank you very much.
Enjoy.
Thank you. Good morning, everyone. Thank you, Bernie. I am Janet Hubka. I am the founder of ELUS Art, a life science art and education company based in San Diego, California. And this is my husband, Dr. Mark Hubka, who runs clinical trials for Histogen in San Diego. I am not a scientist, but with 3 family members working in the field, I thought I knew a little bit about the work being done in research. I was proven wrong one day when I mistook this image for peacock feathers. Mark was, Histogen had just moved offices. He had nothing on his walls. As a matter of fact, Gail Naughton, the CEO of Histogen, said, we have nothing on the walls in the conference room, in the hallway where all these investors and board members will be coming in less than a week. So Mark said, well, that's a great idea to print that, take that out and print it on canvas, but those are skin cells. The picture was taken right here in the lab by Dr. Mike Zimber at Histogen, and I said, well, that's an even better idea, to use something that relates to what you're doing instead of buying just any old artwork for this office. So 2 great things happened that day. Histogen ended up with some really fabulous cellular artwork for their walls, and an idea for a company was born. I started this as a cellular art company. I work with leading research scientists around the world. I collect and vet their images. I look at the composition, the colors, whether or not, how big that picture will print without becoming pixelated. And then I look at the story behind the picture. I work right now with over 90 scientists from around the world. And if an image sells, or we sell a picture, that lab and that scientist receive a percentage of the sale. And in addition, the person buying the picture is always happy to know that some of their purchase price is going to help further the research. In 2014, Biocom in San Diego was asked by the San Diego International Airport to curate a life science art project at the airport as part of their rotating art exhibit. Biocom knew that I had started printing on metal versus the canvas. So the images that you see up here today, these are metal prints, the ones out in the hallway too with the children's art. And the metal becomes much more luminescent. A couple of scientists have said that that's actually what they see when they look through the eye of the microscope versus on the canvas, it still looks great, but it becomes a little bit less, the colors become more diffuse. It's not quite so vibrant. So not only did we print the whole exhibit, it was about 30 large pictures for the airport, but we also hosted information on our website about the project, about each lab, the scientist in that lab that took the picture, and what the picture represents. And it was at that time that several teachers asked us to bring this project to their schools so that the kids could know what's going on in regenerative medicine. So 2 years ago, Mark and I created an educational program, the one that we're going to share with you today. We're actually going to do the program for you so you know what we're teaching people about. Those are their scientists. You know, we're teaching them about what you do. The exhibit was up for 7 months at the airport. The educational program is geared towards kids from kindergarten through high school. We have presented to adults as well at, for instance, Chamber of Commerce meetings and at meetings such as Kiwanis. What we found is the artwork helps to draw people in. When we give a presentation, we usually have about 20 pictures with us. So there'll be large ones, small ones on a table in the front. We're not usually on a fancy stage or have this many people, but we have spoken to about 500, 600 kids at a time in an auditorium. And when they first come in, they look at the artwork and there's some oohs and ahs because they love the colors. Even with adults, they come in and they're happy to see all the artwork. So that captures their attention. And whether it's the colors or whether it's the abstract nature, and people can look at it and come up with their own ideas of what it is, for instance, peacock feathers instead of skin cells. I'm not sure exactly for each person what captures their attention, but it does. And then we hit them with the science behind the art. And that is one of the big reasons I got Mark involved in the education program. He can pronounce all the big words that I stumble on. I remember the day that I pronounced endoplasmic reticulum, probably something simple for the rest of you, but it wasn't in my daily conversation with people in the line of work I used to be in. But I pronounced it correctly. I didn't call it the green squiggly things next to the nucleus, so Mark was happy about that. But there's still words I stumble on. But believe it or not, even the kindergartners, when they hear the talk and hear about what's going on in regenerative medicine, they remember something. Whether it's a big word, whether it's the word nucleus, whether it's a concept that something's going on in research that they didn't know about before, and they share it with their families. Last year, we had spoken to 300 kids at a elementary school. It was kindergarten, first, and second grades. And the next day, the principal called me and said, a parent just left my office. She had come in and said, what's going on here? What happened yesterday at the school? Was there some kind of a science program? And the principal said yes, and explained what we had discussed with the kids. And she said, well, keep it up, because my first grader came home from school, started tearing through the house looking for an old microscope that her older siblings had long since abandoned. And we found it, got it set up on the kitchen table, dusted it off, cleaned it up, and then the little first grader went around the house, proceeded to look for germs that she could look at under the eye of the microscope, and told her mom that when she grows up, she wants to be a scientist. So the principal said, I want you to bring this back and teach it to all the kids, not just up through second grade, but let's do it for the fifth graders, third and fourth and fifth graders too, which we did. When we present, we hope to inspire someone like that little girl to look at an education in one of the life science industries, or to find their passion in life, whatever that is, whether they're interested in art or being a teacher or mathematics or technology. We just love to inspire a kid. Our own daughter, who is a very creative soul, was inspired as a sophomore in high school to look at a career in mechanical engineering, and is now working on her PhD in bioengineering, so is doing what all of you are doing. She should be finished by next spring if she's lucky and if we're all lucky. But that is an extraordinary thing to see a light go on in someone's mind. I don't take the responsibility of sharing what you do lightly. I work hard to learn about what you do if you're a scientist, to learn about why you do it, and then I translate it to something that someone like me, a non-scientist, can understand. We have presented this talk to over 11,000 kids in San Diego County, and earlier this year we were asked to come to Florida and partner with the Science Center, the science educators at the South Florida Science Center, the art educators at the Center for Creative Education, and as Bernie mentioned, the wonderful organization called Primetime West Palm Beach, or Palm Beach County, PBC?
Yes.
They funded the whole project, which was fantastic. So the pilot program involved students in 4 grades, 2nd, 3rd, 4th, and 5th, 4 schools, approximately 160 students. We came in last summer. We worked with the science educators to teach them about what we talk about in stem cell research and regenerative medicine. They had another additional part of their curriculum that involved other aspects of science, like looking at the brain, some dissection and other things. So their educators incorporated our talk into theirs. And then the art educators from the Center for Creative Education came up with really creative ways for these kids to create art projects that they could be excited about, that would translate what they learned into something that they can be proud of later. So we'd like to thank all of those organizations, several are represented here today, for all the great work that they put in, and all the wonderful student artists that did so much great work in that short after-school program. I mean, really, it was just 5 or 6 weeks, and they came up with some great projects.
Nearly 200 pieces of art.
Yeah, 200 pieces of art.
Are outside.
And I think that those kids are going to be here this afternoon. So if you have a chance, you might even meet some of these great budding artists and scientists. They're supposed to be here, I think, this afternoon about 3. So now we are going to give you the presentation the way we give it to school students. So I'm going to switch from looking at you all as adults to looking at you as middle school students. I'm going to teach you about cells, something you know nothing about, because you probably are going to be learning that in junior high school. And so suspend your disbelief for just 15, 20 minutes here, and we'll get started. Well, before we do, I want to show you that we did get some pictures of the Art of Science program, which are fantastic. So they are represented out in the lobby, but just in case, this is a reminder for you to go out and see all these great works. These silk pictures were actually stretched, so they look a little different out there now, but they're fantastic. Quilting, imagine that. They did these little quilts, and they painted some images on that fabric. Anyway, they're very inspiring.
So today's topics we're going to start with are these 4 areas that we walked through. What is eLoose art? What it means, eLoose. What are cells? We're going to talk about why scientists look at cells. We're also going to talk about stem cells. And we're just going to really do a high-level point about this, talking about the 3 primary stem cells. And then finally talk about multimedia used in research as well as in art, because it's important to bridge that gap as far as STEAM. And, oh, go ahead.
I founded eLoose in 2012. As we said, it's an art and education company. The education came along a few years later, but now it seems to be one of the primary things we do. When we started, I called it biotechnology art and decided I wanted something a little bit different, something more like Google or Uber, something that no one knows then what I do. But the girl that was helping us with branding, a friend of our older son, said her boyfriend had seen the images and thought they were Ilus. He's from Estonia. And we thought, well, you know, that will be a good topic for kids, and we're going out and talking to kids about this. They know very often how to say beautiful in other languages. In Spanish, it's bonita. In French, it will be belle. And in Estonia, many of us did not know this, but it's Ilus. So there's a local architect, Russell
Yes.
Forrester.
Russell
that we
Forrester
met.
is a local in San Diego, in La Jolla, and he's built some of the nicest, most beautiful structures, but he's also an artist and sculptor. And Russell Forrester had a quote. He used to take his pictures out to schools also and give lectures and his quote is, "Do not just look, but see." And that's what we try and do in the Ilus Art presentation is don't just look at that image of ORS cells, outer root sheath cells for the hair follicle. Don't look at it, but we want to look and see. And so that's what we're going to try and do is translate that so you see what these images are. So why do scientists look at cells? One is to understand cell function, obviously understanding development, but when you look at the images, all those different pictures are cells of the heart, of the small intestine. So there's cardiac muscle, there's smooth muscle, there's skeletal muscle, all muscle, but yet very different morphology or shapes of those cells. And the purpose is that scientists are trying to understand how a disease process takes place, how to help stop that disease process, and also help restore normal function and or possibly even grow organs if necessary.
Cells are the building block of life. Also, many times we get questions. Yes, they're the building blocks of plant life and animal life as well. Every cell is going to be different, but every cell is going to have, except red blood cells, will have a nucleus like the center little round object you see there, and the squiggly green lines are the endoplasmic reticulum. And inside that little tiny cell that you can't see, except if you're a scientist looking under the eye of the microscope, there's lots of little organelles, so it's amazing the nano nature of human life. And all of us are made up of 37.2 trillion cells.
That's
and
a
if
test
you
question.
think of cells as like a Lego set. You have all these different little Lego pieces. You're going to build a castle with them. All those little pieces look a little bit different. They're different sizes, they're different shapes, and put together they make that castle. Well, cells are like that. They work together to form an organ, to form tissue, to form skin, to form your brain. Everything in you is made up of cells. And eventually, when they're all put together, they make a person. And then stem cells are a little different.
So today we're just going to focus on the two primary stem cells that there are. There's embryonic and there's adult stem cells. Now, to use a slightly different term, we're going to say pluripotent stem cells versus multipotent stem cells. So the pluripotent stem cell can become anything. It can become a heart, it can become a neuron, it can become a liver cell. So pluripotent can go down any path, and however, once it has decided where it's going, or it determines to go to become a heart, it now is an adult stem cell at that point. And it can only become, now it's already, fate has been determined, so now it's multipotent instead of pluripotent. So just get this concept, and even kids pick this up. But these multipotent stem cells now, they can still produce and regenerate forever, for the rest of their life, but they can only be multipotent, meaning they can produce every cell in the heart. So they can become the cardiomyocyte, they can do different tissue, or even a neuron, it can become a oligodendrocyte, an astrocyte, a glial cell. So these multipotent cells are very important. Those are the adult stem cells. We all have adult stem cells. This is one of the misconceptions out there is some people don't even realize that we have stem cells in our body right now. In fact, if you didn't have stem cells, you would not even survive more than 24 to 48 hours, period. So stem cells are also very sensitive to the environment and different signals. So if the environment's different, for example, if you're an athlete and you go work out, and you work out up in Colorado and you want to go running up there, you'll notice the air is thin, and they do that for a reason, because the environment is different. It's low oxygen in those high altitudes. So your stem cells have to adjust to that environment. And actually in your bone marrow, kiddos, in the bone marrow, that's where you make your stem cells turn on, and they make your body produce extra red blood cells so you can transport oxygen around your body more efficiently. Now that athlete that's been working and training up in the high altitudes comes back down to sea level, and they can go and play and actually work out and get much more oxygen delivery to their muscles so they can last longer, not get as tired as fast. So this is how the stem cells work, and they actually do adjust to environmental signals and to whatever's going on as far as toxins and different things. So in this picture up here, it shows that the terminally, down at the bottom, are actually in the villi in your small intestine, as an example. Now, all your cells in your body replace themselves on a regular basis. Those cells, the intestinal villi, is every 5 days. So you know those stem cells have to really work. They're cranking them out because they have to replace every 5 days those cells. You're sloughing. Red blood cells, 90 days. Liver cells, every 120 days. You're constantly replacing. And all the way up, in fact, only recently, thanks to the wonderful work of stem cell scientists and researchers, they figured out that there are stem cells in the brain and spinal cord. They used to say, the dogma was when we were in school, that everything replaces itself except the brain and spinal cord. That's it. What you're born with, what you develop to, they don't regenerate. But actually, Dr. Fred Gage, Rusty Gage, out of Salk Institute, figured out that no, there are stem cells, and it's very, very critical research and important research that was validated. And now it's almost common knowledge. Everybody here probably says, of course, brain stem cells, yeah, of course they have them. But that was only 8-10 years ago that was fully validated. So the potential uses of stem cells in research are over here on the left side. You know, Alzheimer's, Parkinson's, they're doing research for baldness of all things, blindness, obviously diabetes, spinal cord injury. Over on the far right, in the middle there are the cultured stem cells. And right now, those stem cells are pluripotent. So they can go all those ways. They can go to become liver cells. They can become cardiac cells or nerve cells or muscle cells. Now, see over there, even the intestinal cells. Now, once it goes down a path to become a heart cell, it is no longer pluripotent, it's now multipotent. So it only stays over there on that heart side or on the nerve side. It stays in and becomes all the repairs of whatever you need brain-wise if you have a stroke or if you have some type of injury to the brain, a traumatic brain injury. And it doesn't go back. However, there is one additional stem cell I'm going to talk about which scientists have now picked up, and that's called the induced pluripotent stem cell. And that is the one that we're going to have, there's a lot more discussion on, because it actually takes your own cells and reverts them through some special treatments or changing its environment and makes it go back to become pluripotent when it's actually already been down a path. Here's this picture here is of the ear. You have your outer ear here, the ear canal, the tympanic membrane, which is your eardrum. And then you have that cochlea, which is the snail-looking thing in the ear. And up at the top, there's normal hair cells. Those are the hair cells that's inside that cochlea. And if you can normally hear, those hairs are all even and coming across the top there like you see. Down below it is damaged hearing cells, or those hair cells are what'd you call it, Janet? You had a term, wonky.
The medical term is
The
wonky.
medical term is wonky.
Yes.
And so they get a little wonky, and all of a sudden they have to go, hello, what did you say? And that's the middle one. The bottom one is dead hearing cells. Those hair cells have been damaged so bad. And this happens very often when somebody's listening to very loud music and bang, bang, bounding music, or in blast injuries in the military if they're out and all of a sudden a bomb goes off and they'll have the ringing in the ears for a while. And sometimes it comes back, sometimes it doesn't. So these are research. This picture just to the right of it is actually the work being done at Stanford University on stem cells being grown to grow these hair cells. And that's 1200 magnification just to the right of the ear picture. To the further right, the far right, are actually 23,000 magnification, and that's the SEM, the scanning electron microscope of those hairs. And what the scientists do is they grow those up till they're even, and then they'll either implant them into the ear and/or they're trying to stimulate your own body's stem cells to generate those hairs for the goal of healing deafness.
When a scientist has decided to study a cell line, they take a petri dish, they get the cell line, they put it in, and over the next few weeks, they do what's called waiting until it gets to the right confluence, which means it's the right density, the right number of cells. During that time, they're feeding it a liquid form of what we eat, which is healthy forms of protein, carbohydrates, and fats. Once it has reached the confluence, they fix it, and they fix it with many different things, usually a chemical, so let's say a formaldehyde. Fixing kind of freezes it in place. And then they stain it with antibodies. And the interesting thing is that when I first saw these images, and most people, most of us that are not scientists, think that it looks almost like a scientist must have painted that or directed a color to be in a certain position, because otherwise how could it come out as pristine as they do? But actually, with that little petri dish, they're just dropping the dye in, just dropping it in, and it attaches itself. Each different color of dye has a different reagent that attaches itself to different structures within that cell. And that way, the scientists can study the reaction of that cell to different scientific experiments that they want to do, or medications, or drugs, or that kind of thing. So once they've stained it, they're going to look at it under the eye of the microscope. That microscope up on the screen is the kind that I used in high school and in early college, but it's not the kind being used by research scientists. They're using great big ones. The one on the left is at Rice University in their bioengineering lab, and that takes up a whole room. Many of you that work at universities have seen that many times over. It has several camera attachments, a laser. It has several computers that are attached to it. And when a scientist is taking pictures, they might take 30,000 pictures to substantiate their research. And for scientists, it's just as important to keep the information and the research images of the experiments that don't work as it is for the experiments that work. Because inevitably, they're going to come back to it and say, did this work? Let's go back and look at it. On the right is at Histogen. That's actually Dr. Zimber that took some of these images today. He is working on the confocal microscope at Histogen. You see a computer behind him. You can't see the camera attachment up at the top. But institutes around the country, and you can see some of the names at the bottom, quite a few of those are in San Diego, but when we work with some of the scientists at these different institutes. But research is going on at the major hubs of biotechnology, which a lot in California, up in the San Francisco Bay Area, Southern California, and Boston, but it's happening everywhere where there's university or where there's biotech companies and medical research. We found that very often when we speak to high school students that they're interested in learning more about internships, and quite a few institutes and universities do take summer internships. We always encourage students to look in your area to see if you can find something. We've had several contact us and actually placed a few people, whether they were young college students that are looking at what they want to do in grad school or high school students. We've placed them in different labs in San Diego. This is a working laboratory. The person at the far left is working under the hood. That is a sterile environment. That's where she would go to, or he or she would go to feed their cells, to run some of their experiments. And the reason why it's sterile is because it's a reverse airflow. Is that what it's called, Mark? Yeah. The air is going out of the unit versus air just being able to go in. So there's no outside contaminants going into that area. The other two are setting up, getting ready to look at things under the microscope. More people looking under the microscope. It's great to see that there's lots of women that work in biotechnology. Well, we're not going to show her video, but that happens to be our daughter that's getting her PhD at Rice. I'm going to pull out, you're back to being scientists for a second. Normally we show this to high school students for sure, because it really helps for them to relate to someone closer to their age than the old people on stage today. But the little videos are very short, and it's the kids get a kick out of seeing a tour of the lab.
This research is done at the Lowy Medical Research Institute in La Jolla, and this is an interesting picture because they figured out, the scientist, Dr. Martin Friedlander, and his team and also the Scripps Research Institute, by the way. But he figured out that the signals that in the back of the eye, in order to figure out which way the blood vessel is going to form, they actually go wherever the blood vessels go out and grow wherever the green and gray glial cells are. So in other words, if they're together, if there's only green, then it won't grow that way. If there's gray and green, the red blood vessels grow out. So the importance of this research is actually figuring out certain types of blindness from macular degeneration and those type of things, and they've actually picked up a lot of information from this research to deal with that. The Lowy Medical Research Institute actually focuses on that. This research is on diabetes. Now, everybody in the room, even children, we say, how many of you know anybody that has diabetes? And of course, their hand goes up. And this is research growing the beta islet cell from the pancreas, actually from stem cells, but directing it to become pancreatic beta islet cells. And those are the cells that actually secrete insulin. And insulin you need in order to restore or normalize blood sugar from going too high. And so people that diabetes, particularly type 1, have to take injections every day. And this company or this research is where they put these cells in a packet, inserted into the abdomen, out in the peritoneum, which is outside of your organs, but inside the belly. And the goal being that these will actually react to your blood sugar, and you won't have to take injections every day. So if those packets are in there, these cells actually recognize if the blood sugar goes up, they secrete insulin. If the blood sugar goes down, they secrete glucagon. So these cells are research that was funded by the California Institute of Regenerative Medicine. They supported it, and a company called ViaCyte. They're actually in phase 1 clinical trials. There's 3 phases of clinical trials. They're in 1 year in of a phase 1 in man. So they've taken this and they've inserted these packets, and so far it's been very positive, meaning the body hasn't rejected these packets. They hope that someday they'll only have to insert this packet once every couple of years, and that person won't have to take daily injections of insulin. Pretty exciting.
Many of you will recognize this actor. This is Christopher Reeve, very well known for having acted in the Superman trilogy back in the 1980s. He was also well known as an equestrian, and he ended up with a very severe spinal cord injury when he was thrown from a horse in a jumping accident. The injury was so severe that he was not able to breathe on his own. He had to, the rest of his life, live with a breathing apparatus to help him breathe. He and his wife created something out of that tragedy, and that is a foundation in their names that helps support people with spinal cord injuries, and it helps support the research community in finding a cure. His foundation is funding research right now out of the Kierstead lab at UC Irvine. Sharon Rossi is the doctor that was taking these images. These are the motor neuron progenitors. They were working on an FDA-approved clinical trial of therapy based in human stem cells, which was very exciting.
This was also funded by the CIRM in California.
Oh, CIRM was the co-sponsor on this, along with the
Sue and Bill Gross Stem Cell Center, and obviously the Christopher and Dana.
Another actor that has come out and talked about a disease that he has, Michael J. Fox. Even the littlest kids know who Michael J. Fox is, because those of us that love the Back to the Future movies have required that our children and grandchildren watch them. So everybody knows who he is. He's come out and let everybody know that he has Parkinson's disease. He also created a foundation that is in his name. He's funded some research out of the UC Berkeley lab of Dr. David Schaefer, and Tandis Vazin also worked on this project. Dr. Schaefer is the head of the Stem Cell Center at Berkeley. These are the dopaminergic neurons. Dr. Schaefer, when we went to visit him at his lab, told us that when a person goes in for the medical diagnosis of Parkinson's, that at that point, they've already lost 50% of the dopamine in their brain, which means no matter what treatment their medical advisors are giving them or recommending they use, that they're already at a disadvantage. So they're working on finding ways to create dopamine. When I first saw this image, and it's actually behind Mark here on the stage, you can tell on the metal print how vibrant it is. I thought it was coral in the deepest part of the ocean, and our high school nephew thought it was a nebula in outer space. So
we actually call it Orange Nebula.
And we called it Orange Nebula because of that. But That's great research. There's also great research going on in Parkinson's in the Loring lab at the Scripps Research Institute in San Diego. You are very lucky to be able to hear Jean Loring this week speak. I think she's speaking later today on about another project. This is work being done by Andres Brat-Leal in her lab. The great thing about her lab is they work directly with Parkinson's patients. And they are doing that induced pluripotent stem cell therapy that we talked about, taking a patient's own skin cells, reverting it to be pluripotent, driving it down a line to become dopaminergic neurons. And the beauty of that is it becomes more of a personalized medical approach to treatment for that patient. You can see whether or not using different therapies are going to work for that patient or whether it be harmful. And hopefully, my understanding is that eventually they would like to be able to grow dopaminergic neurons and somehow use those, get them back into that patient so that they...
Earlier this year, Dr. Loring was awarded got an award from California Institute of Regenerative Medicine. I believe it was 2.4 or 2.5 million, and that's to actually get it into the clinic.
Right.
So they're talking hopefully by next year, not next year, by 2018 to actually go into the clinic with a patient's own iPS cells from their skin and revert them to dopaminergic and have them start secreting dopamine for that patient again. Very exciting.
The great thing about working with the Loring Lab in San Diego is no matter where you go, if you go to a Parkinson's walk on a Saturday at 7 a.m., they are there because they know those patients and they really care about the people they work with.
This is the work that we talked about earlier about Dr. Rusty or Fred Gage and Carol Marchetto at the Salk Institute. And this research was the one that basically opened up the idea and understanding that the neurons in the brain and spinal cord do actually regenerate. So it's very, very positive research, exciting. So these are the nerve precursor cells. And Janet, I like the way you explained that.
Well, I did not understand how neurons spoke to each other. I did study this in high school biology, but did not remember it. So all of us that are learning it, all of us high school students, on the left of the picture is one neuron, on the right is another neuron. And through the dendrites and axons, yes, I remembered how to pronounce them, information is sent back and forth for that person. And so whether it's information about things that you're aware of, like I'm going to start writing on my notepad or I'm going to work on my computer, or whether it's information you don't think about. I need to breathe, I need to swallow when I chew, all of those kinds of things. Those neurons send information back and forth all the time.
The kids like that.
And these are healthy neurons, so that's interesting to see.
This is work at the Sanford Burnham Previs Medical Discovery Institute.
Say that 3 times.
They just recently changed their name after. But this is at Evan Snyder's lab. Dr. Snyder, this is the neural rosette, and this is the adult stem cell that's in all of our brains. And how we explain this is, in your brain, these are just hanging out. They don't look like this. They're just hanging out, doing nothing. And then all of a sudden, you're an NFL football player, and you take a hit, and you have a concussion. And that concussion actually can trigger that neuron, and boom, it turns on. So the neural rosette fires up. And now from that, everything, whether it's a glial cell, an astrocyte, a neuron, a regular neuron, a dopaminergic, or an oligodendrocyte, this is what generates it. And it's exciting research. Janet?
We had a 5th grader say, it looks like a party in my brain. Because it really does look like a popper that you would use at New Year's Eve or you'd use at a birthday party. So now even Dr. Snyder calls it his party in the brain picture. When I said earlier that the Fred Gage's picture is of healthy cells, or neurons in the brain talking to each other, this is neurons at the top and bottom of the picture that are sending information via a lot more dendrites and axons than the last picture. But the little red dots are Alzheimer's plaque that is disrupting the flow of information. So imagine if you have a line of ants on the sidewalk during the summer afternoon and you're looking at that line of ants and you put something there that blocks their path completely. So this side kind of starts circling and goes back to where they came from. The other side is confused and I'm supposed to be taking food to the nest and where did it go? but the communication line has been damaged. So the same thing happens with an Alzheimer's patient. And some of you have grandparents possibly, or friends of your grandparents of your friends that have developed Alzheimer's. My father had Alzheimer's, so I definitely know how it works and how it feels to have someone not remember you, even though they knew you very well for your whole life or their whole life. So the flow of information has been affected. This is also out of David Schaefer's lab at UC Berkeley, the stem cell center there.
Yeah, those are cortical
And they're
neurons.
looking at ways to treat Alzheimer's patients and maybe reverse Alzheimer's in the future. I mean, there's not a clinical trial on that yet. This image is also Alzheimer's, but if you took a tiny image from the last picture, a little tiny section of it, it would be what this image represents. So this is a very close-up of just a tiny part of a brain cell. The healthy tissue is blue and the purplish aqua and the aqua color. The damaged tissue with amyloid plaque is the tannish, goldish color. I thought when I first saw this, it was either a beautiful piece of marble or something that they took, a picture they took for the International Space Station looking down at the Caribbean, you know, land formations and water. But it isn't, and it is an ugly disease, but I did go and speak to Dr. Schubert at the Schubert lab at Salk Institute to tell him thank you for working, being a part of the airport project that we worked on. And I gave him this image in a small size, like a 10 by 10, as a thank you for working on that project, a picture he could hang up in his lab. And I said, this is quite a beautiful picture, I really do like it. And he said, well, I don't like it, it's very ugly to me. And I said, well, I do understand why you think it's ugly. It's Alzheimer's. But my dad died of Alzheimer's. And I actually printed this for myself, hung it in the hall at our house, and I see it every day. And I don't see the disease anymore. I see the research that's going on. And I see the hope that maybe I won't develop Alzheimer's, my siblings won't, and our kids won't, and everybody affected by it will reap the benefits of the research you're doing.
Well, what
He
Dave
said he's going to look at it that way. He went ahead and hung it in his office. And he told us that he's working on clinical trial, and Mark's going to tell you about
phase 2
that.
clinical trial, and meaning they've already passed phase 1, that was successful, now they're in phase 2, and it's for a small molecule, but it's based on a natural herbal supplement, which we've all eaten and you know, seasoned our foods with, called turmeric, which is an Indian herb and a common spice, curcumin or turmeric. And the point is that he's finding that a lot of Alzheimer's obviously is related to some, not just this amyloid plaque, but the combination of the inflammatory process taking place. And they found that a certain fraction of that herb actually does help with reducing inflammation in the brain and elsewhere. So needless to say, Janet went right out to Whole Foods.
I did start taking turmeric immediately. The other interesting thing about Dr. Schubert is, if any of you have been to Southern California and seen the Salk Institute, it's 2 giant monolithic buildings, very modern, beautiful water feature in between. It's in La Jolla, right over, on the ocean, so they've got a beautiful view.
Overlooking the
very
glider
nice
port.
modern labs. Dr. Schubert chose one of the old bungalows, one of the original little structures on the property, the very front of the property, so he has one of the best views. But when we went into his lab, I was a little surprised because he is one of the senior scientists there. But he said, I chose this lab because I sit at the desk that Jonas Salk sat at, and I get inspired by the view of the ocean that Jonas Salk was inspired by. And I thought I would share that with all the students we talk with.
This is research at the Salk Institute also, but this is at the Juan Carlos Belmonte lab, and this is growing a kidney from stem cells. Very exciting, because what they've figured out, again, those signals, they've directed it down a path to actually generate a kidney. And so if we look at the blue circle, here's a drawing in the middle of the kidney with the ureter coming out. And look at the blue is the kidney part of it. The bright green is the ureter. And this is 3 days in a Petri dish. So this is Yunzia's.
Yeah, it's exciting research for all of us that know someone that has waited on a waiting list for an organ,
Kidney.
or a kidney, or someone that has not survived, way down that waiting list, or has had a rejection of that organ, of a donor organ. With personalized medicine, you really do eliminate those problems. Now, obviously, this isn't in clinical trials yet, so a ways off for all of us, but in the future, our children will certainly be benefiting from the research that's going on.
And Yunzia now is in Singapore, so
Yeah.
now Janet has her scientists down in Singapore that she's getting images from. But the interesting thing is the number one reason for kidney failure is end-stage diabetes. And diabetes being so rampant that if the kidneys go, and so people can be on waiting lists forever. If you have a kidney failure, you're on dialysis 3 times a week at multiple thousands of dollars for the rest of your life until you find a donor. If you find a donor, even your own brother or family member, sometimes it doesn't take. So then you're back on dialysis. So this is a real process. And with the stem cell research and with the science going down and figuring out how to direct that cell to become a kidney or to become whatever organ we're looking at, the brain and so forth, it's very exciting because then you'll get not only personalized medicine, but real therapies to possibly not have to wait for a donor.
I think this picture looks like a little portion of Van Gogh's Starry Night. It does look a little bit like outer space. It's actually human hair follicles. This is research by Dr. Zimber out of Histogen, where Mark works. They're in phase 3 clinical trials for growing hair. When we first added this to this deck for taking out to schools, we used it a couple of times, and I said, Mark, I'm just not feeling it. It's male pattern baldness, and it isn't as critical as someone that's researching, the issue of Parkinson's disease or spinal cord regeneration or growing a kidney from stem cells. And he said, well, it's interesting that you said that because I just met with somebody they're working with, an oncologist that works with Histogen, and he said, when someone has been diagnosed with a very severe form of cancer, let's say a stage 4 type cancer, the research has shown that the 2 top questions that people ask are, will I survive the treatment, the cancer and the treatment, and will I lose my hair? Hair is a part of our self-image. Having hair or not having hair is not just an issue of men that are going bald. It is also an issue for women that have alopecia, children that have, men and women and children that have alopecia, people that have been in an accident. If there's scar tissue, hair will not grow. If there's been some kind of a burn to the scalp, the hair will not grow. We were speaking last September to a group of school children in San Diego. There was 500 little kids in an auditorium. They were using up all the oxygen, and it was very hot in there, and there's no air conditioner. Mark was wearing a sports coat similar to what he has on, and the 2 of us were really hot and sweaty and bothered. And all I could look at was this little girl in the front row, a 2nd grader, with a little hat on, like a ski hat, like she was cold. The rest of the room, not one hat. At the end of the talk, it just happened that a teacher we had known from she had taught our youngest daughter, who is now 25, in kindergarten. That was that teacher's very first year of teaching, so we both remembered each other. And she said, did you see little Lily go up to your picture that was on the table? And it happened to be this picture. And I said, I did see her, the little girl in the hat. Yes, that is Lily. Well, Lily has alopecia, not one hair on her head, no eyebrows, no eyelashes. So she is going to be talking about this for a while. The teacher emailed me the next day and said that Lily reported to her family that there's a company that's going to be growing hair for her pretty soon. So let's hope. I said, Mark, we're keeping this in our slide deck forever because of Lily, because of all of us that have some family member or someone that has some form of problem with growing hair.
Autism is, well, it's been reported on the rise and at least recognized. This is work out of Dr. Jean Loring's lab at the Scripps Research Institute and Dr. Michael Boland. And again, this is talking about those induced pluripotent stem cells. So these are taking those biopsies from a patient's skin with autism and reverting it and directing it down a path to whatever neuron or neural cell that they're trying to create or want to help support for this patient. But the nice thing about it is, so these are 2 different patients, and this is Michael Boland's work, but the point is, in the clinic, you can actually, in a Petri dish, then use a different medication or a little different thing and see if those cells react to it. And if it's reacting positively or negatively, they can actually alter or affect what the treatment's going to be for those patients. So this is really personalized medicine at its best.
We get to the technology side of how this art is created. Like I told you earlier, I used to print on canvas. And it wasn't until I saw these images printed on metal, this is all thin sheet of aluminum, surprisingly light for how they look. There is a framing material around it, that's the thing that makes it a little bit heavier. But the great thing about it is it's not printed on the metal, it can't peel off, it's infused into the metal. They're guaranteed for up to 100 years, which means that we can take these little pictures without being framed and pass it around to all of you, school children, so you can feel it, you can't hurt it. It's really great. And because scientists say that's what they see when they look through the eye of the microscope and that's how it looks, we decided that's the only way we're going to print from now on. And fortunately, a friend of mine who's an engineer had come up with a patented way to produce these art pieces. He takes this machine, the bottom part of it slides out, they put the image on that table, the piece of metal goes on top of it, it slides back, compresses, 2 tons of pressure and 450 degrees of heat, and voila, a piece of art is made.
And it gets almost a backlit appearance without having to use lights behind these images.
So this is the project that we talked about at the San Diego airport. You can see on the far right, if any of you are from Southern California, that's Joe Panetta, who is the CEO of Biocom, being interviewed by the press. At the bottom is Yunzia. I'm talking to Yunzia about her picture with the kidney picture. Some of the scientists were very happy to see that their name as artist. So now they have 2 things to add to their billing. They're scientist and artist. We also did a project at a hospital in Los Angeles. The great thing about this project is that the day that Mark and I spent there helping to get it installed, 18 big pictures in 2 hallways, that we had just as much requests for information from the staff as we did the patients that are going through these hallways. So from the surgeons and medical staff to the nurses to the hospital administration to the maintenance people to the clerical people, everybody wanted to come and know what each piece represented because everybody knows somebody with some kind of a disease process.
Well, I'll point out that Janet creates these plaques that actually gives credit to the scientist, the specific scientist, and the lab. But more importantly, at this hospital, Dr. James Lally, who's the chief medical officer, he asked, because of his demographic at that hospital, he said, can you do the plaques in English and Spanish? Look, a little extra work to get them translated, the scientific information, but yet there it is, English on the left, Spanish on the right, and it was very powerful.
This is Dr. Adam Gazzaley at UC San Francisco. I'm not sure how many of you have heard of the glass brain. It's a YouTube video. We're going to show a very brief part of that video, but if you want to see the whole video, it's more like a 7 or 8 minute thing. You can go home, look at it at your leisure.
They have a few different videos.
So he's wearing a skull cap, he does a EEG and a functional MRI. They actually look at how neurons fire in the brain. The reason why his lab is doing this is because he's an MD, PhD, he's working with patients as well as doing the research. He works with a lot of people with cognitive issues, whether it's dementia, Alzheimer's, or maybe an accident victim or a stroke victim. And they look at what part of the brain is functioning correctly and what part isn't, what part they can help to strengthen. And he has also created video games. So you see he's holding a game controller in his hands. He hires gamers to help him come up with these video games. So when we went to his lab up at UCSF, he has a room with a giant TV as big as one of these screens and a bunch of people sitting there playing video games all day, but also creating things.
I think they're actually coding with a joystick.
So a lot of kids are very interested in this because even if they consider themselves not as good in the sciences, everybody's good at video gaming. And medicine and research is so integrative, there's a spot for every one of us.
So this shows the kids that they actually can look at these things and get excited about this, that even if they don't think of themselves as scientists, they could actually definitely do a video game and go in there.
So this is how the glass brain looks, the project looks. He hollows out the different lobes of the brain, they become glass. You can see the neurons firing when someone is reading or listening to music or any kind of activity. They follow it, they look at how it turns, they look at how the neurons are firing in the different lobes.
They'll put a VR glasses on, and as they're looking at the virtual reality glasses, they'll actually see areas in the back in the occipital region where the eyes register, and they'll see that firing. If they're listening to music, for example, or hearing something, they'll actually see the the temporal lobes firing off, which you'll see here. And then if they're actually, for example, one of the drummers from the Grateful Dead, Mickey Hart, actually helps support some of this research, and he'll actually have, on one of the videos on YouTube, he'll actually have the whole skull cap and eyes on there, and he's banging away on these drums, and you see the neurons firing down the spinal cord, so really, the motor neurons. So very exciting for somebody to see this. The kids, what do they say, now they know there's a party in the brain, and that it actually fires, how it works. So,
I'm going to go move on.
next.
I'm trying. There we go. So if you want to see that whole video, you can see it on YouTube, The Glass Brain. This is 3D printing, mechanical engineering at its best. Mechanical engineers have very long, manufacturing companies, toy companies, automobile companies, that industry uses mechanical engineers and 3D printers to create their prototypes. Medicine uses it as well to create prototypes for knee replacement, for hip replacement, that kind of thing. Dentists now are using them for orthodontic retainers. I was at the dentist recently and they created a crown for me right there in the office using a 3D printer. PR companies, and toy companies, like I said, also use them for instance, this company in San Diego needed a reproduction of the Smithsonian woolly mammoth at a moment's notice, got a 3D printing company to create that version for them. You've seen that in Night at the Museum. Amazingly enough, desktop 3D printers are under $1,000. They will be in all of our homes someday. It seemed like it was just yesterday that they were $50,000. You only found them at the biggest institutions, but they're readily available. This is a very short video on how a 3D printer works.
Double click.
Oh, this is Olo.
Meet Olo. Imagine being able to
That's
print
okay.
3D objects directly from your smartphone for under $100.
The wrong one.
Olo is the first smartphone-powered 3D printer. Simple and affordable, Olo uses the light from the screen of your smartphone to print 3D objects. Half of its hardware is already in your pocket. Olo works with phones of all sizes. Its app is available for iOS, Android, and Windows Phone. Creating 3D objects is as simple as picking a design from Olo's easy-to-use library. Or you can use any 3D mobile app, including 3D scanning, to make your own. You can also choose designs from the web that are created by others or shared via social media. Olo can print files from the 3D design software of your choice. Select your object, pour in the resin, and Olo does the rest. Olo uses a special photopolymeric resin. Olo prints your design by using the light from the screen of your smartphone to harden the liquid resin layer by layer. Olo can even print multiples of the same object, or several different objects all at the same time. After Olo prints your 3D object, all you need to do is rinse it under warm water. Olo has different resins for different jobs. Olo can print hard objects and flexible ones. Olo even has a castable resin that can be fused, so you can create an object in silver and gold or other casting metals. Olo's resins can be used to print translucent models to light up your designs.
Actually, that's a Kickstarter campaign that one of our daughters shared with us. The other video that we won't show you shows a desktop 3D printer, and you can see how a company might use it for something else. Olo, $100. We can have a little 3D printer in our own home with the power from your smartphone, which is pretty cool.
So in our talks, we just then ask the kids, does this inspire you? Does this excite you? And then we even have at the end, so what we learned today, we review with them. Of course, what is eLoose art? What does it mean? Beautiful. And then we walk through the cells and the 3 different stem cells. And this is something that we just wanted to bring out to you, is that eLoose art is actually working to create a traveling life science art exhibit, just like we did at the airport and hotel, at the hospital, and taking this around the country and stopping in major cities for 6 to 8 weeks at a time, and then even giving these type of presentations and let it at 3 or 4 schools in each city and teach the kids about the art to get them excited about it. And these are some of the things that eLoose has expanded the additional product offerings. These laser cut wooden puzzles with a microscope and a double-stranded helix and so forth. There's the brain. There's that dopaminergic neuron right here with the brain and out of the head. And these are wooden puzzles.
Science you can play with.
Science you can play with.
And a clothing manufacturer asked if they could use some of the images on a scarf. So we're working on that project. Isn't finalized yet, but
it's science you can wear.
Yeah, science you can wear. Anyway, great projects. The great thing is that we are trying to help, back to looking at you as scientists and the general public. We're hoping that what we can do is help inspire people about what you're doing and get people to understand what's going on in the field of regenerative medicine. We admire all of your fortitude and moving forward and trying to get things moving along. It seems like we've come a long way in the last 5 to 10 years in research. We thank you for all your hard work. Our families and everyone that we know that has some kind of a issue with their health, thanks you for all the great work you're doing. And that is the end of our presentation. Thank you.
I think we're all set.
We'll leave that there.
I think there's a break.
I think there is a 15-minute break.
And then what NASA, I believe, is next.
good to go? How about that? Good. Well, hello everyone. My name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation. My organization is the producer of the World Stem Cell Summit. We are delighted that you are part of the public day and understand the sessions from the Grand Ballroom are being seen all over the world, live streamed. We hope you're going to enjoy yourself while you're in Palm Beach, and we're certainly happy to host you. Now, this particular panel is going to take you to off the planet. One of our co-organizers this year is the Center for Advancement of Science and Space. Many people do not realize that what is going on the International Space Station, the science of microgravity and cells, is a game changer in an enormous way. Some of the research has spawned one of the great new commercial enterprises, Zero Gravity Solutions, and you have around your neck on your lanyard, Zero Gravity Solutions. You'll be able to hear of a case study later on in the main summit. But for now, what we are going to be presenting in this session is the mind-bending science that's going on in space, how it's going to impact the field of cell biology and stem cells. We're even going to Skype, and we're going to meet one of the exciting astronauts that's part of NASA and the space program. Chairing this session is Liz Warren. Dr. Warren is the Associate Program Manager of the Center for Advancement of Science and Space. Let's welcome her and the CASES team. Thank you.
Thanks, Bernie. Well, we have a very exciting day to do today. Thank you all for joining us this morning at the World Stem Cell Summit. As Bernie said, my name is Liz Warren. I work for the Center for the Advancement of Science and Space, CASES. You'll be hearing from us later on today. We have a simulated mission to the International Space Station. We've got a panel on Thursday as well. So Center for the Advancement of Science and Space, that may not mean a whole lot to this audience. So let me tell you briefly what our organization does. First of all, we have an amazing laboratory in space. It's orbiting about 250 miles up right now. It took several years to build and a lot of effort, a lot of launches right out of Cape Canaveral up the road here. And this laboratory in space is called the International Space Station. It goes around the entire planet every 90 minutes, and it is a truly world-class, off-the-world laboratory where some incredible research is taking place. As CASES, we are responsible for the U.S. National Laboratory on the International Space Station. We're responsible for selecting and manifesting science ranging from life sciences, materials, and physical sciences, consumer product development, Earth observation, STEM education, and technology development. We're responsible for about 50% of the research allocation and crew time on the International Space Station, as well as 50% of the available up mass and down mass. That's bringing research and samples to the space station and bringing them back to Earth. The biggest difference between CASES and NASA, who are our partners in this endeavor, is that our research team selects research and manifests research to the space station. It must have an Earth-facing benefit. It must benefit all of us here on Earth. So a lot of NASA's missions are intended to keep humans productive in space longer, and they're really exploration-related goals. Our goals are to bringing benefits from that microgravity research back down here on Earth. So for this audience, how can microgravity impact life sciences or stem cell research to improve human health here on Earth? How can we better understand disease or develop better, more potent therapeutics to improve our way of life? And how can microgravity help to unlock some of those secrets that maybe aren't even possible in laboratories here on Earth? which is why this panel is a great way to introduce you to what is possible through microgravity inquiry on board the International Space Station. And honestly, we have really a great treat for you this morning. We have two brilliant scientists here to tell their story and talk about the research that they sent to the International Space Station. And we have the actual astronaut who conducted their research on board the International Space Station to join our conversation. So that's enough introduction. Let me get to our guests here. I'd like to introduce Arun Sharma, furthest to your right, formerly of Stanford University's Cardiovascular Institute, and is now a research fellow at Harvard University, and Dr. Peter Lee, to my immediate left, from The Ohio State University. They were both co-investigators on a very recent space station experiment focused on the effects of microgravity on stem cell-derived cardiomyocytes, or heart cells. And now it is my great pleasure to introduce, joining us remotely from Houston, Texas, NASA astronaut and biologist, microbiologist, and virologist extraordinaire, Dr. Kate Rubins. So Kate can't see us, but we can see her. Fantastic. So this is what we're looking to do this morning. I have a series of questions for the panel. We'll tell you all about their investigation in stem cells research in space, and then we might have some time for questions and answers at the end. Does that work for everybody? Excellent. Well, let's go. Kate, let's start with you. Welcome back to Earth. You landed on October 29th, so just about a month ago, after 115 days in space, living and working aboard the International Space Station. Can you tell us a little bit about your mission and what your daily schedule looked like?
Absolutely. It's great to be back on the planet with all of you guys. We were living off-world for quite some time. And it's, as Liz said, it's really an incredible laboratory. So a lot of the day is actually devoted to scientific experiments on board. We performed over 275 different science experiments. One of the more interesting ones, I think, was the cell culture experiment. So in this experiment, we were really able to grow cells in culture for the first time for a really long duration aboard the International Space Station. Of course, your day up there involves a lot of other things, looking at the Earth, taking photos, physics experiments, fluidics experiments, and of course we have to fix the toilet and maintain the space station as well.
Awesome. And for this audience that may not be aware, living in space while it looks like a lot of fun to float around in microgravity, it's actually not that great for the human body. Some of the effects include muscle loss, bone mass loss, cardiovascular deconditioning, and so that brings me to my first question for Peter. You're a cardiothoracic surgeon with an interest in astronaut heart health. Can you tell us about how astronauts' cardiovascular deconditioning is similar to cardiovascular issues here on Earth?
Sure, absolutely, and thanks for that great introduction. So as many of you may know, a lot of what happens in space affects the human body, and most systems in the human body are affected one way or another, and it's usually detrimental. Things that we are most commonly aware of are, as already mentioned, muscle loss, bone loss, but changes to the vestibular system, and the cardiovascular system certainly is not immune. And it appears when you see the changes that happen to the human body, it mimics what you typically see with aging. And so it almost seems like an accelerated aging process. And that's interesting for several reasons, but one is that we study what happens in space in a short period of time. We may be able to get some information and understand what happens with the aging process, which takes obviously a lot longer. And so there are applications to that research for here on Earth. For the cardiovascular system, we know that there's not only a shift in the body fluid, so there's a shift of fluid from the lower legs more up to the head, which has a lot of impact to the whole cardiovascular system and how much blood volume there is, which overall goes down. There is, we know that the heart function appears to be affected. There appears to be some degree of muscle atrophy or decrease in muscle mass. and also the control systems for the heart. The neurological control systems appear to be impacted negatively. And there's also, at least incidentally, there's an increase in arrhythmias or irregular heart rhythms. And so, fortunately, till now, the astronauts that go into space are generally relatively young, they're very healthy, and they have time to exercise and do countermeasures to help prevent some of the changes. But in the future, we're looking at very long duration space flights, so more than just the 6 months, looking at possibly a couple of years. And then we're also looking at, with space tourism in the forefront, individuals who are a little bit older, less healthy, going into space, and we don't know what kind of effects those will have. And so understanding how these changes impact the heart are going to be very important. And some of these changes we also will be able to correlate with cardiovascular disease on Earth. And so that's what we're looking at.
Right, so a lot of exercise is done in the daily lives of astronauts to, like you said, to help prevent or mitigate some of that cardiovascular deconditioning. So is that what led you to the heart cells investigation, to proposing this research to occur?
Yeah, so I've had an interest in space-related research for many years, even before I decided to go to med school and pursue the PhD and do research as well. And as I gradually got into my clinical field, which is primarily cardiac surgeries, I do heart surgeries and I do heart transplants. Obviously, my focus started going towards the cardiovascular system and the heart. And the heart hasn't been studied as much as muscle, skeletal muscle, and bone in the past because it wasn't as big of an issue for the shuttle era when they were in space for 2 weeks, 3 weeks. But now we're realizing as we look at longer duration, we're looking at the effect of radiation as we think about going to Mars. The heart is now much more vulnerable and we need to better understand it. So we're at a point now where it's really important to get beyond where we've been able to study the heart. And now we also have technology that allows us to study the heart cells that 5-10 years ago was not really possible.
That's awesome. So Arun, tell us more about how you arrived at this heart cells investigation and your involvement. And if you would, tell us about some of the implications or applications of using the induced pluripotent stem cell-derived cardiomyocytes to study cardiovascular biology.
Sure. So as Peter mentioned, until now, we haven't had a really good model system, a model cellular system with which we can study cardiac function. And so that's where the induced pluripotent stem cells, or iPSC-derived cardiomyocytes, came into play. These iPSCs, as a lot of us know, we can derive them from a small sample of a person's own skin or white blood cells over a process that takes about 1 month. And after that 1 month long procedure, you get pluripotent stem cells which can transform into pretty much whatever cell type you're interested in, whether it's a neuron, a skin cell, and since we're cardiovascular researchers, we're interested in cardiomyocytes or heart cells. And so there are thought to be 3 major applications for these iPS-derived cardiomyocytes. The first application is using these cells as a platform to screen pharmacological compounds, screen compounds that are under development by drug companies. To give an example, some of these compounds can have off-target cardiotoxic side effects. And this iPS-derived cardiomyocyte system allows us to have a platform with which we can screen some of these toxic side effects. Another application of iPS-derived cardiomyocytes is potentially for the purposes of cell therapy. So for example, during a heart attack, you actually lose a substantial portion of the cardiomyocytes, the heart cells in your heart. And the heart is actually a really unique organ because those heart cells that are lost during a heart attack usually don't come back. It's very difficult for the heart to regenerate itself. And so we're thinking that we might be able to utilize a different cell source with which we can replace those lost cells in the heart. And that's where the iPS cardiomyocytes come into play. And the third application, which is kind of what we were doing here, is using these iPS-derived heart cells, these cardiomyocytes, as a model system, a system with which we can model disease or model physiological function. To give an example, if there's a patient who has a genetic abnormality causing an irregularity in a heartbeat, that same patient's iPS-derived cardiomyocytes will exhibit the same irregularities. So you can model those arrhythmias, those heart rhythm problems, using cells derived from the patient, these iPS-derived cardiomyocytes. And in a way, that's what we were trying to do here. We were trying to use iPS-derived cardiomyocytes to model the effect of microgravity on heart function. We've been able to look at heart function on the organ level, but it hasn't really been well understood in terms of the effects of microgravity on the cellular level. And that's why these cells are very important and why this is a very useful system. So what we were doing was we derived iPS-derived cardiomyocytes from 3 separate individuals and sent these cells to the International Space Station, where they were cultured for about 1 month by Kate Rubens, who's joining us here today. And on orbit, Kate was examining these cells for changes that may have occurred in response to microgravity, whether it's changes in cell shape, size, beating ability, and also gene expression, changes in genetics. That's also something that we looked at. And so she was able to replace the nutrients on these cells on a weekly basis for 1 month, and then return a sample of cells live back to our laboratory at Stanford University, and we're still in the process of doing some additional analysis on that.
Well, that's fantastic. Something that you mentioned is we have a variety of ways of looking at cardiovascular health on the space station. And in fact, earlier this morning, Dr. Rubens was getting a post-flight medical exam, actually collecting data to look at part of her cardiovascular system using ultrasound. And there's actually an ultrasound on board the International Space Station as well. And it appears there's actually been a fire alarm over at NASA. So Kate will be back with us when the fire alarm is over. But this actually brings us to an interesting point. For both Peter and Arun, it's a little unusual as a scientist for you to not be hands-on with an experiment that is occurring here. It's not in your lab, it's in a lab that's only 250 miles away, but it's pretty far out of reach because it's in space. So tell us about some of the challenges and tell us about translating your experiment from your lab bench to space. It's probably a little not as intuitive as one might imagine.
Yes, absolutely. Doing research in space is certainly a lot more complex and more expensive, and it takes a lot more planning for sure. I've had the privilege of having done other spaceflight experiments back in the shuttle days and also in zero-G parabolic flights and a self-contained fruit fly experiment about 2 years ago on the space station. And one of the things that I've learned early on is that the way you think about planning and setting up an experiment is completely different. You know, the essentials of coming up with a hypothesis and the design of the experiment and the analysis and all that doesn't change, but how you set up the experiment and how it is actually conducted is very different. So there are several factors. One is the fact that it's a one-time deal. You know, so if you're doing a simple cell culture experiment, so for example, what we had done for the ISS, if we had done that in the lab, we could have done it and found either didn't work or found some things that we could have done better. We just repeat it. And it's not a big deal to reculture the cells, plate them, and do it again. You can do again over and over again. Here, we have one shot. If it doesn't work, or it gets contaminated, or something was wrong with the hardware, the design, basically all that work, time, money, resources is lost. So we're putting all our eggs in one basket in a sense, so we have to really plan it well. The other issue is the hardware. Anytime you fly something in space, you can't just take the Petri dishes and culture dishes that you just get out of a package that you order. This is, for the most part, custom designed and very expensive and complex. So even the simplest things, just having cells in a culture dish, that all you have to do is feed media is relatively complex. The other aspect is changes in flight or even just before the experiment. We really can't change the experiment because it's set in stone in essence, because the protocols, everything has been set up. So even leading up to the flight, once it's decided, you really can't change it. And definitely once you're in space, you can't change when you want to change the media or things like that, with some minor exceptions. But for the most part, everything has to be done with good planning. If something happened in a lab and we need to change the media in the middle of our experiment, we could easily do that normally. And the other issue is the limited resources, whether it be equipment that we would normally have in a lab, or availability of astronaut time. And crew time is a big issue. And we're very fortunate with this being a CASES project that we had some priority in terms of crew availability, but that's not always the case. And even then, we have to really work closely in terms of how much time we can get for the astronaut. In this case, Kate Rubins we're really fortunate to have her, and with her experience being the one taking care of our experiment. But you're not always that fortunate. Sometimes you have someone with no prior biomedical experience. They have to be trained to do some of these things. So there are certainly a lot of limitations, but with good planning, good implementation partners, and support, it can be done. But certainly it's not like doing something in your own lab.
Yeah, absolutely. And I think you touched on this a little bit, Peter. We're biologists, and we needed a team of other expertise to actually help us implement our project to the International Space Station. So we know how to do the cell culture and the cell biology, but of course, it's a pretty complicated process to take your biological experiment and actually transport it to the station. And so science these days is a very collaborative effort. That's a fact. And so we actually had the great privilege of collaborating with a group of aerospace engineers from the University of Colorado, BioServe Space Technologies. And they actually helped us design a lot of these custom pieces of equipment, these fully enclosed 6-well plates in which we could grow our heart cells and actually transport these cells to the space station, where they could be changed, the media could be changed by Kate. So it was really a team effort, and we were really fortunate to have a team of people like BioServe to help us out.
So if you will, just think with me for a moment. Instead of growing cells like in a petri dish or in a flask, that wouldn't work in microgravity because the fluid would climb all over the walls of the flask. It wouldn't stay in contact necessarily with the cells. So this hardware that both Arun and Peter are mentioning is very unique in that it allows the cells to adhere to one side, be covered so that the fluid doesn't leave the container, and that covering had to also be gas permeable so that air could exchange with the media as well. And then it had to have a port on it so that the crew member, in this case Kate, could change the media and give the cells new food to eat. So that's just kind of a hint at how complicated it is and how much you have to think about when you're making an experiment for space. So, and let me back up to something that you said, Arun. You said just sending your cells to space, but you were actually at the launch, right? What does it feel like to have an experiment in your hand it over to some folks that load it on a rocket, and then watch it leave the planet? What is that like?
I think from a scientific perspective, it's so unbelievably rewarding to see that something that you've worked on for 3 years it's leading up to this moment, this one launch. And we were all very fortunate in that our launch was, very successful and everything went really well. So as a scientist, you want to be able to have access to these really unique environments, such as microgravity, so that you can do the coolest and most informative science. And we were really fortunate in that way. And from a personal perspective, I grew up wanting to be an astronaut as well. I think every little kid wants to be an astronaut at some point. And so to be there at the launch and to witness that in person was just absolutely incredible. We actually were part of a SpaceX launch where SpaceX was testing out a reusable rocket. And that was also really cool to experience as well, because about 20 minutes after the launch, the capsule separated, the capsule containing our experiment. It went off to the space station, and that contained our experiment. But the rest of the rocket was actually able to come back and land successfully upright on the launch pad. And if you know anything about aerospace engineering or rocket science or anything like that, you should realize that that's an incredible accomplishment. And just to be a part of something like that was really special.
Exciting, definitely very cool. So let me ask you, how connected did you feel to your experiment? I know once you did a lot of prior preparation and planning and a lot of thinking into the possibilities of what could go wrong and how to deal with issues that are occurring on orbit. But did you feel pretty connected to your experiment? Did you feel like you were getting information and updates and were you able to watch your experiment happen live?
Yeah, absolutely. For my perspective, where I've had other experiments that go up, this was night and day in terms of how connected I felt. My first experiment was back on John Glenn's flight on STS-95, and it was a small experiment. I was still a grad student at the time, and basically handed it over, and then they sent it to me by FedEx a few weeks later, and that was about it. So I didn't really feel connected at all, other than I got to see the launch and all of that. The last one was with fruit flies, and same deal. Essentially, the fruit flies were all in a self-contained system, and handed it over. It went up. There was nothing that happened in flight in real time, no immediate feedback, and then it came back. This was very different in that not only, again, having Kate Rubens be part of this and feeding the cells on a weekly basis, she also imaged the cells, so we got to see the actual cells essentially near real time in terms of how they were doing. There was immediate feedback in terms of how they looked and any suggestions that we might have. So having all of that has been a very different experience and really did feel connected, as much as you can be, given that your cells are 250 miles away.
And we see you back with us, Kate. Fire alarm must be over. So you launched on July 6th, and then just about 2 weeks later, we had this SpaceX launch in which the heart cells were delivered via the Dragon capsule. Now, of course, there was thousands of pounds of equipment and supplies to the space station. You, Kate, were still kind of getting used to living in microgravity. Did it make you feel a little more at home when you got to pull out a microscope and use a glove box and do cell culture? Did that make you feel like, okay, now I'm doing science in space?
Absolutely. The Dragon launched, and one of the first few things we end up pulling off there is any science experiments that need to be kept at a particular temperature. So my crewmates laughed a little bit at me because I was saying, where are my cells? Where are my cells? we need to get those going today. And it's actually, it's quite fascinating that we are able to do long-term cell culture in orbit now. So this is really a capability of the space station that we can launch cells, that they are orbiting the planet at Mach 25, and we can study the effects of free fall on cell culture. So cells are going to behave differently in free fall. It was fascinating being a human being in free fall inside the space station. You're falling all the time, and so that was actually a big objective of mine, was to try to understand how we can do science processing like you would in a normal laboratory, except for you're pulling it off of a spacecraft that's just launched off the planet, and we're doing it in this orbiting laboratory instead.
That's awesome. And for each of you on the panel, and Kate, you included, we've all talked a little bit, or we've hinted about how magical microgravity is. Can you tell us, what is different for cell culture in microgravity? What might cells do, or how might they behave that is different than doing cell culture just on a plate or a flask on Earth?
Sure. I guess on the simplest level, and I think it's something that we touched on, you don't have to worry about, on the planet, you don't have to worry about the nutrients and all those different components going all over the place, right? So that's one thing that you just don't have to worry about, but that's a very realistic possibility in microgravity. And of course, this is a very, very unique environment. It's something that's not very easily accessible to us here on Earth. And we don't really know too much about how individual cells perform in the context of microgravity. And I think that's why this particular experiment was so important.
Yeah, and we talked about, just the practicalities of simple thing like changing media that you can train a high school student to do in a short matter of time. You just take out the fluid and put it back in. That becomes a much more complex process. So those are practical limitations, but as Arun had mentioned, a lot of these cells, for example, these heart cells, as far as I know, is the first time heart cells have ever been flown in space. So we don't know what to expect. We didn't know if they were going to last this long in microgravity, even though our ground tests show that they would last a couple of months. We didn't know if that would be the same once they're in space. So, to a certain degree, we have to take a chance and make our best educated guess. And so that's one of the limitations with doing any kind of microbiology research in flight.
Yeah, and I was particularly interested in how the cells were going to aggregate. When you plate cells on Earth, of course, they will often settle to the bottom of the dish, and they will stick to each other, but they'll also stick to the plastic in the dish and form in certain ways. And one of the things that was really interesting to see was the structures that the cells formed as they aggregated together. And that is not disrupted by fluid flow like you would get if you have a rotating vessel to have cells constantly free floating. So it allows you to look at cellular interactions without that force of gravity, which is the physicists would disagree, but coming back to the planet, I'll tell you, gravity is a very strong force. So it constantly has an effect on the cells. And to me, it was very interesting to be able to do an experiment essentially in a place where the laws of physics seem to have changed, where we did not have that constant vector of gravity pushing the cells down onto the plate.
Did you have a follow-up?
Yeah, so, and we've been talking a lot about the limitations and difficulties of doing spaceflight experiments because in the microgravity environment. But one thing that came to my attention, I was thinking about as Kate was talking about the gravity and how the cells are pulled down. In some ways, that could actually be helpful, for example, with tissue engineering. And that's one of the things that, CASIS and with the US lab, that they're trying to take advantage of this environment for applications for research. So tissue engineering is a field where the lack of this vector of gravity may actually be helpful. And I think there are ways we can take advantage of this environment to take advantage of these possible applications.
Absolutely. The tissue engineering, we have a tissue on chip challenge as well, and using microgravity as a tool to unlock investigations and unlock the secrets of many biological systems that we just can't do on Earth. So that brings me to my next question, Kate. You worked on well over 200 experiments. Heart cells wasn't the only thing that you got to work on. Talk to us about the space station as a research laboratory for other biological sciences, and what other facilities exist that allow you to do life sciences research in microgravity.
Yeah, so one of the parts of this experiment that wasn't just about looking at cardiomyocytes was really proving that we can do culture long term. And that has application for a variety of different cell types. Just for example, a fibroblast would sit down and have some interaction with the plate. So there's a variety of structural things about the cell. What happens to structural proteins inside the cell? Everything in microgravity balls up. Everything wants to form spheres in fluids. And so we also look at what happens to fluid dynamics, processes like convection change. And then, of course, one of the things I was very interested in was a lot of the molecular work on space station. So we started the very first experiment to do DNA sequencing in space, and that was also a technology proof of concept, but it's very interesting to be able to get real-time analysis back from your experiment. So you could take a sample, for example, of the microbiome of the space station, which is a separate microbiome from Earth, and rapidly assess what's going on in the microbiome. Or you could take a sample from one of the rodents that we have in rodent facilities up there and understand what's happening with bone loss in real time. So there's sequencing capabilities. There's now a couple different ways to do real-time PCR capabilities. So it's essentially a full-up cellular molecular lab. We have microscopy and fluorescence microscopy through our Japanese counterparts. And so one of the things that was very interesting is to look at this and try to think how we can do some end-to-end experiments where we are taking human samples or cell culture samples, we're growing them up, we're analyzing them, and then we're using molecular tools to get real-time analysis off the planet.
Fantastic. Got a little echo there. So we've touched a little bit on the fact that you got your cells back from the SpaceX capsule. Do you have any preliminary results that you'd like to share with us?
So we're still in the process of doing a lot of our downstream data analysis. A few things that we can share is that in general, on the broadest level, these cardiomyocytes, we believe, are a really good model system to be able to study microgravity. And that's because before the space flight, they were contracting, and during the space flight, they continued to contract, and there's a sample of cells that we still have back at our laboratory that is continuing to contract. And so at least on that level, this does seem to be like a good model system. In terms of that contraction, there were some changes that occurred in response to microgravity, but we actually also observed that those changes in contraction that occurred reverted back to normal for the most part when these cells were turned back to planet side. And in terms of our gene expression analysis, that's still something that we're actively doing right now back in the lab.
And that highlights one of the capabilities that have been afforded to us is the fact that we can bring some of these cells back live. So fixing some in space, it kind of fixes it in time in terms of how the genes were expressing in space, but then be able to bring them back, continue to analyze them and see how they change or potentially revert back. That's a capability that is not always or has not always been possible in the past. And so with the Dragon capsule and our ability to retrieve our samples is also a great improvement in terms of our capabilities.
So next steps then, would you want to send additional cells back to the space station, or what is next in your analytics then? You're going to get to the gene expression. Seeing that the cells actually responded differently to microgravity in the way they were contracting and then reverted back to normal when they returned to Earth is really pretty fascinating. I'm fairly certain that probably something similar happened to Kate's heart. It acted a little bit differently in microgravity. Now that she's back on Earth, she's technically still in her reconditioning phase, getting used to being back on Earth. And as Kate, as you mentioned, being in the gravity well is you can definitely tell you're back in gravity. But what would be the next step for your research?
So the reason we like to do cell culture-based research is that it affords us a lot of control over the environment, as opposed to doing in vivo or animal research, where there's a lot of variables that affect the cells that you're interested in. So if we're looking at the heart, there are hormones and there are cytokines and there are a lot of factors and neurological input that affect the heart that we don't know if it's something that's direct at the tissue or some of these other factors. So we can isolate things in a cell culture. But cell culture is not the same as being in a whole heart either. And so I think once we get the data back, and especially with the gene expression studies, that'll help us focus in on certain genes and certain genes that may be a target for therapeutics, for example. So those would be interesting experiments that we could follow up with in terms both in cell culture, but ideally to take it to the next level, which would be a three-dimensional tissue culture model. And that's something that I have been interested in, and there are grant opportunities that we're looking at, where we would do something very similar, but instead of looking at it in a two-dimensional culture, it would be a three-dimensional tissue that's beating, that'd be a little bit more like heart tissue. And then following up on that would be to maybe do something in an animal, for example, now that we've narrowed down the areas that we want to look at. But obviously there are a lot of steps in between to get to that point.
Yeah, and I think these sort of studies are, of course, not only limited to the heart. there are other investigators who are also interested in looking at liver cells and all the other organs of the body to see how their individual cells respond to microgravity. I think there's a lot of interest in seeing at the cellular level what's the effect of this really unique environment on human physiology.
Absolutely, and this is just the perfect time to be doing this type of research on the International Space Station. It's so capable. We've mentioned that there's freezers, you can fix tissues, there's microscopes, glove boxes, incubators for doing cell culture. So it's a wonderful time to be doing research on the space station. So Kate, when you do assumedly go back to the space station, what other types of experiments would you want to do in space? Or what did you not get done while you were in the space station?
Well, we got a lot done, but as I usually say when people ask what kinds of experiments you want to do, I just say all of them. I think some of the most interesting experiments coming up are ones that are investigating gravity as a tool in basic biology. So we talk a lot about how we're going to look at cells in microgravity for astronaut health, but it's also just a fundamental mechanism that we haven't been able to probe before. And so when you're studying the basic biology of a cell, for example, how cells interact, we now have a new variable to play with. And so we can remove gravity and as was mentioned, look at three-dimensional models of cell interaction. Does that change how the cells are communicating with each other, how they're associating with each other in tissue? Does it change our understanding of cellular signaling and of cell structure and function? And I'm quite interested in some of the molecular work because with some of the high throughput sequencing for broad analysis as well as real-time PCR for some focused analysis, we can look at a variety of tissues everywhere from microbial effects in space station to effects of radiation on DNA. We have a very interesting low Earth radiation environment that we cannot simulate right now on the ground. We could put cells into a beam that will do a single ion, but we can't simulate the complex mix of heavy ions. And so just being able to expose cells to that is really going to lead to advances, I think, in our understanding of cellular and molecular biology. So I go back up to the space station tomorrow and have quite a lot to work on. But I think one of the really interesting things is for scientists to try to figure out could either radiation or the lack of gravity be a factor in their experiment, and design some experiments that are going to probe this really unique place that we have to study this that now has the combination of a lot of tools that you would find in a regular laboratory that's not going along at 17,500 miles an hour.
Fantastic. That's exciting. Actually, I have a question for this entire audience, including you, Peter, and Arun. By raise of hands, who here wants to go doing science on the space station? This sounds like the best possible place to go. Yeah, pretty much everyone raised their hand, Kate. Before we wrap up, I want to ask, because Peter and Arun, you probably haven't had a chance to even speak with Kate yet about your research. Are there any questions that you've been dying to ask her that you haven't been able to yet?
Well, first I want to thank you, Kate, for taking such good care of our experiment and ourselves. We're very fortunate in that there were no real major issues or complications, and we got our cells back, and hopefully we'll have all really good data. But I imagine not all experiments had gone perfectly, as I know from past experience and from other investigators I've talked to. As a scientist who have done hundreds of experiments on Earth, what is the biggest limitation or frustration in terms of what you're able, not able to do, carrying out these other experiments? And are there any particular things that you wish you could have had, or that potentially could try and fix or improve for future investigators?
Yeah, one of the most difficult things on board is getting enough time to do all of these investigations. The space station is an international laboratory, and we have countries from all over the world that want to fly their science. And so I think one of the things that we can actually work on moving forward in the future is multiplexing science on board the space station. So running a process like a sequencer reaction or a PCR reaction where we're actually running, or something as simple as pipetting, where we, rather than doing one sample at a time for one investigator, we're actually doing 96 samples or 384 samples. We've now proven a lot of the technology that allows us to do these kinds of lab experiments in low Earth orbit, which is quite surprising that they even are going to work, because there are things like micro bubble formation that we don't have to deal with on the ground. But we have proven that almost all of these capabilities work. And so now the question is, can we actually turn this into a really high throughput laboratory where it's a bit more of a core facility type concept, and we're able to run hundreds of PI's experiments at the same time. And that's going to take some expansion of our sampling techniques, how we pull samples out from our animal models on board space station, how we take human samples, getting more accessible samples, as well as ways to understand how we might multiplex science and do 10 or 100 or 1,000 more experiments in that same limited crew time that you have on board.
So once again, Kate, I just wanted to say thank you so much for helping us with our experiment and being such an integral part of our experiment. And I think it's really cool to see an astronaut who's also a biologist, because, maybe one day I can follow in your footsteps as well. I had a question about the technical challenges of growing cardiomyocytes. You're, of course, a cell biologist in your other career, and you've grown a number of different cell types. Where would you say cardiomyocytes, these heart cells, rank in particular in terms of their difficulty of being cultured and, that sort of thing?
Well, they actually were quite easy to care for, and there were two challenges that I was worried about. One was, are we going to get bubbles introduced to the cell culture media? On the ground, of course, if you have a bubble or two, it just floats to the top and pops and disappears, and you don't really see bubbles in suspension. But in microgravity, when everything is in free fall, you'll get bubbles that will stay in suspension in your solution because there's no gravitational vector to bring the fluid down and the bubbles up. So this is, a really interesting area to experiment with and to understand how that's affecting biology, but could be a technical issue. And so I was very glad to see that we got introduction of some small bubbles as we were changing the media, but that this wasn't something that prohibited us from growing cells. And the cells actually clump together very nicely. They are communicating with each other. And when you're doing cell culture, you're always dreading a big cell die-off or a contamination of some kind. And it was actually very similar to just setting up a hood, cleaning it off, and keeping the cells healthy and alive and uncontaminated for I would have to say this is a bold experiment, you guys. Like I'm very impressed how long we were able to keep the cells alive, because in my lab we used to grow cells for a much shorter duration and then infect them with a virus, and the experiment was usually done in about 48 hours. So the long-term cell culture was an interesting challenge, but it worked incredibly well on board.
And I want to thank all of our panelists for joining us today. I hope that you have a better appreciation for the challenges, but the unique aspects of microgravity that the International Space Station affords us. Kate, again, did several hundred experiments while she was on board. Heart cells was as she mentioned, a pretty bold and really, really cool experiment. We've had a great time during this panel, and I will remind you that later on today, we have another cases panel. It's called the Simulated Mission to ISS. It's in Ballroom A from 3:30 to 4:30. And that might also be a pretty cool experience to learn about doing science in space. And thank you very much. Appreciate everyone's time.
Hello, my name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation and Chair and Founder of the World Stem Cell Summit. Thank you for coming to another session of our public day. In the history of World Stem Cell Summits, we had a great meeting back in 2010, and one of our partners was the University of Michigan. It was in Detroit at the Renaissance Center there, and it was there that I was acquainted with Dr. Gary Smith, our next speaker. Dr. Smith is a leading scientist in the field of pluripotent stem cells, embryonic stem cells. He's created stem cell lines that are with the National Institutes of Health registry. I can't think of a better person to explain some of the basics of stem cells as the engine of regenerative medicine, let's all welcome Dr. Smith.
Great. Thank you very much, Bernie, for the kind introduction. And as we get started today, I'd like to thank all of you for taking time out of your busy schedules to participate. And this is really a great day and a great couple days for discussing stem cells, regenerative medicine, and where the science is going. And so today what I'd like to do is talk to you. In the next 30 minutes, we cannot cover all the basics of stem cell biology, but we definitely will cover some of the differences. And before I start, I'd like to acknowledge the team that I work with. It's really their hard work and their dedication, their talents that really give me the opportunity to talk to you about the things that we're doing. So, discussion, what I'd like to talk about is what is a stem cell? Very basic. What are the different types of stem cells? How can stem cells be used? And specifically, we'll talk about their potential use in regenerative medicine, and then also in disease modeling. And when we talk about disease modeling, we'll break it down into discovering how bad genes cause a disease, and then also how these cells can be used for potential therapeutic screening in the future, and I'll give you some examples of that. So it was mentioned earlier today that our bodies naturally have stem cells. And so while stem cell science is a fairly new area of study, the actual importance of stem cells has been around us from the beginning of time. We are losing millions of cells every second, and without a supply of new cells, we would lose our gut, which might not be a bad thing for some of us, but lose our gut or the lining of our gut in a few days. We would lose our skin in 3 weeks, and we would lose our red blood cells in about 4 months. So within our bodies, there are stem cells that have specific functions. Some of the important things is trying to figure out how they function and how they work, how they might be kept quiescent, or how they might actually function when they're damaged. So very basic properties. What makes a stem cell a stem cell? And this list can go on, and as our science progresses, we find that there's more descriptions. But very basically, a stem cell is a cell that has the ability to self-renew or replicate and survive for a very, very long period of time. And sometimes this might be indefinitely, like some issues that I'll talk about with human embryonic stem cells. In addition, besides self-replicating, while they replicate, they need to maintain a normal chromosome number. And third, they have to have the ability to develop into a cell with specific functions and characteristics that are different from the cell of origin. So they self-replicate, but they also can become a specialized cell. So one thing I'd like to point out as a learning moment is not all stem cells are the same. There is some stem cell inequality. And I'll use an analogy here. If we look here, all of these are animals, but they're surely not the same. My dog, Grace Kelly, is not the same as a hippopotamus and is not the same as a giraffe, yet they are all animals. The same is true with stem cells. Embryonic stem cells, induced pluripotent stem cells, child, adult stem cells, they are all stem cells, but they are not all equal. So let's start talking about these different types of stem cells. First, embryonic stem cell. And I will say this is the stem cell that I spend most of my time working on. There are controversies around embryonic stem cells, but that's not what today's discussion is about. Embryonic stem cells come from an embryo. In the work that we do, they come from human embryos. The egg is fertilized, it begins to divide, and by about day 5, we have what is called a blastocyst. It has a layer of cells that are green here that will eventually become the placenta, and those that are pink, which would eventually become the fetus. At day 5, these cells, there's about 30 of them, are undifferentiated. There is no difference between those pink cells, one to the other. We can isolate those and make human embryonic stem cells. We also see here an electron micrograph, and my pointer doesn't work, but we actually can see what is called an inner cell mass, or an ICM. That's where the embryonic stem cells come from. The outside cells are those cells we call trophectoderm. So inherent in making a human embryonic stem cell is the need for a human embryo. These human embryos come from one source, from in vitro fertilization. You cannot take an embryo out of a person, and there are very strict regulations, state and federal regulations, that say that we cannot make a human embryonic stem cell from an embryo that is not made for reproductive purposes. So what that means is that we cannot make an embryo to make a stem cell. Every embryo that we use to make an embryonic stem cell comes from an embryo that was made for reproductive purposes. So how does that happen? That's in vitro fertilization. So briefly, as a review here, we know that women get hormones to cause many eggs to develop on the ovary. Maybe 10, 15 eggs develop on the ovary. Those eggs are harvested. Once they are harvested, they are mixed with sperm. This has generally been done for the last 30 years in treating infertility. Day 1, we look for fertilization. Day 2, day 3, day 4, day 5, the embryos are grown. And then it is that blastocyst that has the inner cell mass that we can actually use to make the human embryonic stem cells. Well, a lot of my background is in human embryology, and I'll show you this in a moment. I have, and I still do, in the last 20 to 30 years, treated human infertility from a laboratory perspective. So if we think about in vitro fertilization and how embryos can be obtained to make human embryonic stem cells, there are 2 sources. One is a source that most people know about, and the other is one that most people don't know about. So the first source is a couple who has infertility. They go to a doctor, they get treated for their infertility, they do in vitro fertilization, and they actually generate multiple embryos. In this example, they've made 9 embryos. They use some of those embryos to have a child. They freeze some of those embryos to make children in the future. And in this example, let's say they're very efficient, and they have 7 children, and they have 2 embryos that are remaining that they do not want to continue to build their family. What do they do with those embryos? They have many options. They can donate those embryos to another couple. They can discard those embryos, that's their choice. They can keep those embryos frozen indefinitely, or they can donate those embryos to research, and one of the areas of research is to production of human embryonic stem cells. So this is something when we consent people to donate embryos, all of the options are known to them. We talk to them, we tell them their options, and it is their choice. In fact, it is mandatory that there is a line of demarcation between our laboratory and the healthcare provider. So we cannot be the ones that actually talk to them about the options. The healthcare provider provides them the options. And if they want more information about donating embryos to stem cell research, they can contact us. The other group that most people don't think about are couples who have already had a child or know they have a family history of a devastating disease. And so there are many of these diseases that we call monogenic diseases, known to be caused by one single gene mutation or a few gene mutations. So you can think about cystic fibrosis, some of the muscular dystrophies, Tay-Sachs, SMA. Many of these different diseases are monogenic diseases. And so many of these couples, as I said, either know they have a family history or have already had a child that's been affected and may have passed away from the disease. They no longer want to have another child to go through that process. They want to have a healthy child. So what can be done is they will do in vitro fertilization not because they have infertility, but because they want to have their embryos tested in the laboratory as to whether or not they carry the gene mutation for the disease or not. If they don't have the genetics for the disease, they will transfer the embryos and have a child that doesn't carry that disease in their family any longer. In the past, all of those embryos that were found to be affected or that had the mutation in the gene were discarded. What we've done at the University of Michigan is made a program to where these families can donate those affected embryos to us. We make embryonic stem cells that have these mutations and then distribute them to researchers across the world to study the disease. And in fact, over the last 5 to 10 years, this is actually something that many families now ask us for because they recognize that, well, they have built a way in their family to remove Tay-Sachs, to remove cystic fibrosis, to remove Fragile X syndrome. They still have relatives with these problems. And so they are actually not only having a child without the disease, but now also contributing to research across the world towards understanding the disease. So a little bit more about the embryo analysis. So this is something we do quite commonly now, where we can take one cell out of a day 3 embryo called a blastomere, or we can take 5 to 6 cells out of a blastocyst, and then we analyze it. We can freeze the embryo, get the analysis back, and know which embryos are normal and which ones are abnormal. And as it says here, there have been over 190 genetic diseases for which embryo analysis at a genetic level can be done and circumvented passing that disease on to the children of the family. And I think the other thing I want to emphasize that most people don't recognize is those affected embryos in the past, their destiny was discard. Now they have an ability to donate to actually help us understand these different diseases. The other thing I want to point out now is that as a scientist, I think we have a love for discovery. We're very passionate about the things that we do. We want to make progress, but progress takes time. And it would be great if we could all hit home runs, but it actually takes some of those RBIs and some of that building on what everybody can contribute to make advances. And so many times I have people asking me, why haven't we cured cystic fibrosis? Why haven't we cured Tay-Sachs? You've been working on this for 10 years. And I would say that good science takes time. So it's been mentioned recently about induced pluripotent stem cells. Very, very important. I was at the meeting when Dr. Yamanaka announced their ability to transform a skin cell into a pluripotent stem cell. And I think this is a perfect example of how very basic research allowed an advancement So understanding what were the transcription factors in an embryonic stem cell that made it pluripotent is what gave rise to the ability of actually reprogramming fibroblasts to become pluripotent. So there are some great advantages of iPS cells, and even though I work primarily with human ES cells, my true belief is that we need to push both areas of science forward. It's too early in science to make a decision as to which will give the best results in the future. The last is what we usually call adult stem cells. Actually going through this presentation and thinking about a more general population, I thought, really adult is not the correct terminology. It's not like children don't have these stem cells, they do. So I've termed them here children/adult stem cells. These are what we call multipotent. They can't become every cell of the human body. A human embryonic stem cell is pluripotent and can become every cell of the human body. Some examples here are heart cells, bone marrow cells, brain cells. Some things to think about with regards to these types of stem cells is that they are usually very rare. They are difficult to identify. They can be very difficult to isolate. They may have limited divisions, and they may have DNA stability issues. Again, with that said, there's been some great progress in using adult, child, stem cells. An example of that is bone marrow transplantations. So now I'm going to talk a little bit about why study human embryonic stem cells. I've already mentioned it is controversial, and I know that. But there are biological reasons. Human embryonic stem cells are the gold standard. And if you go to the basics of biology, these cells give rise to every cell of the human body. And therefore, inherent in what they are is to become different cells of the body. Induced pluripotent stem cells from skin, it has been shown that they have developmental memory. There may be experiments or things that they may be used for where that does not matter. And again, this is why I feel we need to study both. Resource ethical issue. We've talked about the controversy of human ES cells, where they come from. You can't make a human ES cell without using a human embryo. But I would say that from a resource perspective, every embryo we use to make an embryonic stem cell would otherwise have been discarded. Therefore, we feel that it would almost be unethical to not put forth this option for people to donate their embryos to try to help the living and help those that have diseases. Last thing I'd say is I have personal reasons. I was raised in a family of firemen. My grandfather was a fireman, my dad was a fireman, my uncle was a fireman, my brothers are firemen. They're all service-oriented people. And as I became a scientist, that's what I wanted to focus on, doing science that was service-oriented. And in fact, that's why I first started in the area of assisted reproductive technologies, helping families that were infertile build their families. And it's something I still do today. I did it at the University of Chicago for many years. I do it at the University of Michigan still. But there was a time 4, 5, 6 years ago when I asked myself, is there a way I could use my skills, my science, my technologies to help beyond infertility? And my answer was to produce disease-specific human embryonic stem cell lines. And I'll get back to the use of those in a few moments. So the promise of human embryonic stem cell and any type of stem cell research can really be taken down to 2 levels. The first level is regenerative medicine, taking pluripotent cells, stem cells, converting them into bone marrow cells, nerve cells, beta islet cells, heart cells, and treating a disease, regenerative medicine. The other area that we focus on primarily in my laboratory is actually modeling diseases, using these pluripotent stem cells to understand how a cell goes from a pluripotent stem cell to a neuron, from a pluripotent stem cell to a heart cell. What are the mechanisms of normal development, or what are the mechanisms of abnormal development? With that, we feel that we're going to be able to understand developmental abnormalities, developmental birth defects, as well as diseases that are late onset, primarily monogenic diseases, not multivariate diseases. In addition, we can use these pluripotent stem cells for screening new or repurposed therapeutics, and I'll give some examples of that later. The other thing I want to point out, and I think it's an important issue, not just for scientists, but also for the general population, is to balance the hype and the hope. There is no doubt that stem cells hold great hope for the future of medicine. It will take good time, studies, and dedication to get there. With that said, we have to be careful about overhyping it. There is nothing that will cause something not to move forward than bad data. Good data will make things go forward and will make them have a strong bedrock in the area of medicine. So when I think about regenerative medicine and stem cells being an engine for regenerative medicine, it comes down to very simple issues of balancing, promise, and caution. And I'll give you an example of something that I did an interview about a year ago. And this was for PBR, public radio. And I was asked about Gordie Howe, Mr. Hockey, being from the University of Michigan, Detroit, very, very famous person up in the colder country. And Gordie Howe had gone to Mexico, and he had had a stroke, and he went to Mexico, and he had a stem cell therapy. And it was being reported that he had a great recovery. And as many of you probably know, Gordie Howe has now passed away. But when I was interviewed, and the interview is here in blue if you want to listen to it at some point, the thing that I tried to emphasize was clinical trials are a must. Clinical trials take time, but they are a must. They tell us about safety, they tell us about efficacy, does it work? It's not anecdotal. It allows peer review and it allows FDA approval. All of these things are very, very important if stem cells are going to be the engine for regenerative medicine. A few things I want to bring up as limitations of stem cells for regenerative medicine and some caution. Cord blood, child, adult stem cells do have a limited ability to proliferate and to become all types of body cells. That's not true with human embryonic stem cells or iPS cells. In addition, if you put an adult stem cell in the wrong area, a place where it normally doesn't occur, it does not necessarily mean it's going to develop normally. It could develop abnormally, and there are evidence of that. So that has to be, even though they are self cells, they still can have abnormal development. Using self stem cells for genetic diseases or regenerative medicine of genetic diseases is really almost illogical. To say that I've got type 1 diabetes or I've got cystic fibrosis and I'm going to take one of my own fibroblasts, reprogram it, and make it a nerve cell to treat my disease, if it's caused by a genetic defect, we have just made a cell with that same genetic defect. So those are important things to remember. The other thing, when we think about pluripotent stem cells as being important for regenerative medicine, we're not talking about them as a pluripotent stem cell. We're talking about them as a pluripotent stem cell that's been made into a specialized terminal cell. So making a heart cell to treat a heart attack. And this is very important because if one was to think about just transferring pluripotent stem cells, we know they have the ability to form tumors and to form teratogens. So very, very important to know these subtleties. In the last bit of my talk, I'd like to talk about this modeling or disease modeling. And as I've mentioned, this really makes sense for single gene diseases, where we know a gene problem causes an abnormal cell or organ system. But what we don't know is how that disease develops and how it progresses. That's really our black box here. However, if we can make human embryonic stem cells or pluripotent stem cells, iPS cells, for these diseases, now we have a way of looking at how they develop into abnormal cells, tissues, and organs. And as a scientist, one thing I think holds true: knowledge is good, lack of knowledge is bad. So the more knowledge we have, the better off we're going to be in trying to fight some of these diseases. The other thing I want to bring up here is that it's a quote by Helen Keller: "Alone we can do so little, together we can do so much." And one of the very important concepts in our program, M Stem Cell Laboratories at the University of Michigan, is not that we do everything. We do what we're good at. We use our embryology skills to make very good and efficient human embryonic stem cell lines. As soon as we make those lines, it's not the best use to stay in our hands. It's the best use to go into the disease-specific researcher. So when we make a line, we will reach out to any researcher we can find at the University of Michigan, as well as other researchers worldwide. So our goals are to derive novel disease-specific human embryonic stem cells and get them out into the worldwide research community to accelerate our understanding of human diseases and to develop new treatments and cures for these novel diseases using these novel disease-specific human embryonic stem cell lines. We support good, rigorous science, and we believe this is going to support development of treatments. Lastly, we feel it's very important that we spend time engaging and educating not just the general population, but actually the scientific population. So here are some of the different stem cell lines that we've derived over the last few years. These are all disease-specific. These are all embryos donated to the University of Michigan under informed consent, IRB approval. Every stem cell line we make, we send all of our information to the National Institutes of Health. And I'm very happy to say every stem cell line that we have produced has been accepted on the registry of the National Institutes of Health. So you can see there's some for aniridia, blindness. There are some for Huntington's disease. There are some for SCA3, spinal cerebellar ataxia. We just made one for ALS, frontal temporal dementia. Again, any disease caused by a single gene mutation, we can actually obtain embryos that are being tested for, that are affected, that would have been discarded, and make these disease-specific human embryonic stem cell lines. The other thing I'll mention here is that you can see some of these in gray. One of the neat things that we're starting to do is have multiple lines of stem cells from different embryos. And some of them are from the same family, and some of them are from different families. So if we think about science and wanting model systems, we want things that are genetically similar, but some that are genetically not genetically abnormal, but genetically dissimilar. So we can start testing, does this happen within one family, or does this happen in multiple families? In addition, we make control stem cell lines. And again, every line we've made is on the registry, and any researcher in the United States, as well as around the world, can obtain these lines from the University of Michigan. Let me give you some examples of some of the things that we've done with these stem cell lines. The first line I'll talk about was one that was for hypertrophic cardiomyopathy. This is a lethal disease. You know this, many young athletes will die of a heart attack without ever knowing they had a problem. This is a family that had a mutation in what is called myosin-binding protein C3, causing hypertrophic cardiomyopathy. We were able to make a human embryonic stem cell line from this embryo, make heart cells from the stem cell lines, and demonstrate that they actually mirrored the disease in a petri dish. Adrenoleukodystrophy. This is a disease that's, again, lethal. Most boys die of this by the age of 19. We've been able to make a human embryonic stem cell line from an embryo that had the mutation causing this disease, getting it to researchers across the country, and they're starting to understand how this disease develops, how it interacts with other cells, and bringing about the disease, such as looking at its interaction with the immune system. Fragile X syndrome, causing intellectual disabilities in young men who have this Fragile X mutation. We're actually able to make neurons from these embryonic stem cells and study the disease in a petri dish. If you could please click on this slide, please. This actually shows the heart cells that came from a normal human embryonic stem cell line, as well as the heart cells that came from our disease-specific hypertrophic cardiomyopathy stem cell line. So these are the heart cells. It takes about 30 days to make them from the stem cell. But you can see that the heart cells from the stem cells that had the mutation beat abnormally and with much more abnormalities compared to the controls. And this allows us to say, now we have a model to study the disease in a petri dish. Now we can start doing things like screening of therapeutics. The last thing I'd like to do is acknowledge funding for this project. Human embryonic stem cells, because of its controversial nature, cannot be funded by the National Institutes of Health or with federal funds. Therefore, all of our work on deriving new human embryonic stem cells is funded by private, foundation, or local funds. From the university, there's been a commitment to this project, one of the few universities in the country committed to this project. One individual I'd like to mention, Mr. Alfred Taubman, who has passed away, actually a Michigan man, but also from Palm Beach in Florida. And he was one of the very important founders of this project. Presidents of the university, the executive vice presidents of our health system, the dean of the medical school, Dr. Eva Feldman, and Dr. Tim Johnson. The last thing I do is leave this slide up for you. This is an information slide. If you or you know of anybody who might be thinking about pre-implantation genetic diagnosis of embryos for a familial disease, instead of discarding their affected embryos, we have the ability to accept those embryos and very efficiently make embryonic stem cell lines that then can go on the NIH registry and be given out to researchers across the country and the world. Thank you.
Can you advance the slide for the next presentation, please? Everyone take a good look at that. Okay. Is Dr. Hare here? Okay. Come on up, Dr. Hare. So first, Dr. Gary Smith gave a fantastic presentation, and I just wanted to point out that Alfred Taubman received one of our organization's Stem Cell Action Awards for his philanthropy in 2010. Just one of the great people that have contributed and helped the field. So thank you, Gary. So it's my distinct pleasure to introduce as our next speaker, Dr. Joshua Hare. I serve on a couple of committees at the Interdisciplinary Stem Cell Institute at the University of Miami Medical School, Miller School of Medicine. And of course, I graduated undergraduate in law school at the University of Miami, and I'm certainly proud, very proud of the work in the stem cell field that's going on there. I remember a few years ago, Dr. Hare, when he first came to the university and had a presentation, I was allowed to speak, and I pointed out that this nascent institute was going to be very, very important to the University of Miami. It has been a magnet of some tremendous gifts from philanthropists, including those that we're honoring at our Stem Cell Action Awards dinner on December 7th, tomorrow evening, Bernie Marcus and Don Sofer. Dr. Hare is doing some landmark work with MSCs, mesenchymal stem cells, to mend a broken heart. And he's going to share with you some of the important research that's going on right now at the University of Miami in South Florida. We're also very proud that the University of Miami ISKI is also a co-organizer of our event this year. Josh?
Good morning. It's really a great pleasure to be here with you. And Bernie, I want to thank you for putting on, first of all, a superb meeting again, and second of all, for allowing us to participate. It's also, I think, a very nice opportunity to follow with my presentation over the previous one by Dr. Smith, who very elegantly talked about the basic science of pluripotent stem cells, but he also made the case that clinical trials are the essential way to develop any therapeutic field. And indeed, that's what I'm going to talk to you about in the next few minutes. So our group at the University of Miami is very committed to basic science research. We have basic science laboratories, but we also have a clinical trial core. And our commitment has been to do clinical trials with the best available stem cells that are available for use in humans at this time, as regulated by the FDA, but also with the understanding that as the science evolves, there'll be new generations of stem cells that will come down the pike. These will be available for clinical testing, and we will have forged the way with the methodologies we've developed now. So this is actually a fortuitous time because literally in the last 2 weeks, we presented 2 new clinical trials, one for heart failure and one for aging frailty. And so I want to spend the majority of my time this morning showing you results from rigorously conducted clinical trials that are done under the auspices of the Food and Drug Administration and often funded by the National Institutes of Health. I think it's very important to lay the groundwork of why this is so important. These are statistics from the American Heart Association about the very exciting reduction in death from cardiovascular disease, coronary heart disease, and stroke over the last 10 years. So you could see there's been about a 14% reduction in total cardiovascular disease. That's the good news. The bad news, if this pointer works, is that that number, 224 deaths per 100,000, from a heart attack, and that's what's depicted in this slide. The upper panel shows what the heart looks like right at the time of a heart attack. You could see the darker area on the top wall of the heart is the area of heart muscle that's died from a blocked coronary artery. And how that heals over time is in the bottom panel. It forms a scar, a very large scar in the heart that causes a process that we call in medicine remodeling. And what that does is it leads to an enlargement of the ventricle, and that is a substrate for congestive heart failure and sudden cardiac death. So we still have 224 deaths per 100,000 in the United States per year, and we're still spending about $300 billion a year to treat the consequences of heart attack, and that's after all of the advances in medicine. So there really is a need for regenerative therapy, and that's why I think there's so much enthusiasm about stem cells as a way to heal injuries that we presently don't know how to heal. Our group at the University of Miami, as many other groups around the country, have been very actively involved in attempting to do this over the last 10 years and have been doing it both in the basic science laboratory but also in clinical trials using largely a cell type that comes from the bone marrow called the mesenchymal stem cell. So this is how we manufacture mesenchymal stem cells. These are not pluripotent stem cells as described in the previous lecture, but these are an adult stem cell that have a limited repertoire of what they can turn into. But there's been a lot of enthusiasm in our group for using these cells in the hope that they would turn into heart muscle. Now, I'll tell you ahead of time that our research over the last 10 years have shown that they have a very limited ability to form new heart muscle cells themselves in the heart, but they do have a dramatic capacity to heal the heart, and I'll show you all of that data. So I think a big important part of clinical research is that you learn new things that you didn't anticipate when you went into the basic science. And that's still a good thing, because if we can cure some of these conditions, if I could you take it to the back slide, please. One slide back, please. It's still a good thing even if the cure wasn't the way it was intended. This is how we manufacture the mesenchymal stem cells in our GMP laboratory. We take a bone marrow aspirate from a young healthy donor. We take the mononuclear cells in the bone marrow and we can plate that out in a culture dish and take it through several passages. The nice thing about this is that we can quality control the production, and we can make sure that what we're giving back to patients is in essence a cellular drug, because we can very precisely quality control it. And you see in the bottom panel there, one of the things we do is measure the markers on the cell surface, in this case CD105. Once we have a healthy batch of cells that's very pure in CD105, it's very viable, we can give that back to patients as a drug. And this is what we've been able to accomplish. This is the data from an animal model, which we did for the FDA before they allowed us to do in humans. You could see on the left what the heart looks like with a healed myocardial infarction injury. It's got that thinned area on the top wall of the heart, the anterior wall. And in an example of an animal that received stem cell injections directly into that scarred tissue 3 months after the infarction occurred, so once the scar had already formed, you could see the dramatic difference. The scar is much, much smaller. It's much less in circumferential extent, and the overall shape of the heart is much, much better. So we've set about in the last 10 years to see if we can do this in humans. So what do the mesenchymal stem cells do in the injured heart? Well, they remove the scar tissue. That's a very good thing. They grow new blood vessels in that scarred area. That's an important thing. They reduce inflammation, and that's going to be very important in my talk. I'll show you how we've deployed that anti-inflammatory effect for many, many medical uses. And very importantly, they do stimulate new heart muscle cells to grow. They don't turn into heart muscle themselves, but what they do is they send signals to the body to grow new heart muscle. And we've proven that in the laboratory, and we've shown that that occurs in humans. So in essence, we are using these cells and have had very nice results at doing all of these 4 things in the human heart. Just 2 weeks ago at the American Heart Association, we announced the results of this study, a randomized comparison of allogeneic cells, those are cells from a donor, with autologous cells, a patient's own cells, in patients with non-ischemic dilated cardiomyopathy. Non-ischemic dilated cardiomyopathy is the other important cause of heart failure in the world. So what I talked about in the last few slides was people who have a heart attack and get a remodeled ventricle from a scar. These folks get heart failure because they either have a genetic disease or an overactivity of their inflammatory system, which causes the heart to fail. And we wanted to see if the same cells we used in the ischemic heart disease trials could work here. So the background is that non-ischemic cardiomyopathy is an incurable disease caused by genetic or inflammatory causes. Because of the properties that I described to you about mesenchymal stem cells, we wanted to test whether they were affected in this condition, and we randomized patients to the two cell types in advance of a trial that we'll now do with the best of these two cell types compared with a placebo. So this was a phase 1/2 randomized comparison of the two cells. We did this at the University of Miami. We enrolled 37 patients beginning in 2011, and we injected these cells directly into the heart muscle with a NOGA catheter system. That's what the system looks like. And then you can use this catheter with this needle to inject the cells directly into the heart muscle at the desired area. As already alluded to at this session, it's very important that the first thing you do when you're developing a therapy like this is to test safety. We did that, and then we looked at a whole host of efficacy endpoints. How is the heart functioning? How are the patients feeling? What's their inflammatory situation? So the first thing was the safety. It turned out to be very safe, and there were no serious adverse events within 30 days. Every patient tolerated the procedure, was able to undergo it, and as I said, at 30 days, we're doing just fine. Now, things became very interesting as we followed patients out for 1 year, because we started to see that around between 6 and 12 months, that the patients getting the allogeneic therapy were having fewer side effects than the patients with autologous. So if you look at this side effect called MACE, which are multiple cardiac endpoints, only 20% of the patients with the allogeneic therapy had a MACE event, whereas almost 60% of the autologous patients did. If we looked at that further, we looked at any reason for being admitted to the hospital. It was reduced in the patients taking the allogeneic cells. More than 50% reduced. It was 28% versus 70%. So there are hints here that the allogeneic cells are working better because the patients are doing clinically better and requiring less hospitalization. Now let's turn to the heart function, and this is what I think everybody's interested in the field, particularly in cardiology. These hearts are very weak. Can we make them better with the cells? And we measure what's called the ejection fraction, which is the percentage of blood ejected in each beat by the heart. And lo and behold, the patients with the allogeneic cells had a very dramatic increase of 8 units, and it was not nearly as good, but still a nice improvement in the autologous. Now, what really struck us and really was very exciting to us is that 50% of the patients getting the allogeneic cells rose their ejection fractions to above 40%. 40% is actually the cutoff for the diagnosis of the disease. So about 50% of the patients treated by strict criteria no longer had the disease 1 year after the treatment. And here's a very dramatic example. You can see how enlarged this heart is and how poorly it's functioning. The ejection fraction is only 19%. This same patient, 1 year later, getting all other best conventional therapy, has a dramatic recovery. The ejection fraction is now 43%, and the heart has dramatically reduced in size. In essence, I don't want to overstate this, but this is as close to what we can consider a cure for this condition. This patient no longer has the criteria to meet the disease and is much, much better 12 months later. If we ask the question, how's the patient feeling? How are they doing? There are also several signs that the patients feel a lot better. Their symptoms of congestive heart failure are reduced, as evidenced by asking them questions on this questionnaire, as well as grading them in the New York Heart Association classification. These are very standard tests that are done in the field, and on both instances, the allogeneic patients are doing better than the autologous patients. How far can the patients walk? When you have heart failure, you can walk barely any distance, and we use 6-minute walk testing as a standard clinical tool and in research studies. Well, what we see here is that the patients can walk a lot farther with the allogeneic cells, and they can do so at a statistically significant greater degree than in patients with the autologous cells. Same holds true for their breathing function. So there are a number of measures that I'm showing you that the patients' hearts are getting better, their quality of life's improving, and their functional capacity is improving. Now, we've become very interested in how this happens, and we've measured a number of things. I told you that the stem cells stimulate the body to produce their own stem cells to a greater degree. And one such stem cell is called the endothelial precursor cell. This is the cell that lines our entire blood vessel tree. It's a critically important cell. With all cardiovascular disease and many other diseases, you lose these cells in your body. And what we showed in this study was that, in fact, the mesenchymal stem cells stimulated the patient's own body to produce a large number of endothelial precursor cells and bring the level of the endothelial precursor cells up to normal. Now, I've told you that the mesenchymal stem cells are anti-inflammatory. They have a number of interactions with the immune system. But one thing that we're most interested in heart failure is something called TNF-alpha. This is a cytokine in the bloodstream. The TNF-alpha drives the progression of heart failure. It's very elevated in patients with heart failure. And here we show that the allogeneic cells reduce the TNF-alpha levels by 70% and do so in a sustained fashion, and that this decrease is greater than the patients receiving the autologous. So by a number of measures, we see that the allogeneic stem cells improve the patient's condition and improve the biology of the patient. The endothelial precursor cell levels are back to normal, and that TNF-alpha levels have been reduced to 70%. So a very consistent story. So we were able to conclude in the study that the 30-day safety and tolerability was excellent in both groups. The allomesenchymal stem cells produced improvements in ejection fraction, 6-minute walk distance, Minnesota scores, reduced MACE events, and all-cause hospitalization. It appears that immunomodulation is critical. This is important in this disease, which is driven by inflammation, so the anti-inflammatory effects are very important. And what this kind of trial has allowed us to do, and I think this trial is a model for what should be done in the field, which is to first do smaller studies, show that the approach is safe, optimize the cell, which we've done. We've compared 2 versions of the mesenchymal stem cell, shown that one is better than the other, and this now paves the way for what we call pivotal trials, which is where we now compare in a larger population this cell to a placebo, and hopefully have these results bear out that the patients getting the cells do better than the patients not getting the cells. That is the pathway to getting approval from the Food and Drug Administration for the use of these cells in broader populations. Where are we going next with this field? Well, we're part of a network that the National Heart, Lung, and Blood Institute funds called the CCTRN network. And the CCTRN is very actively interested in seeing if we can enhance the efficacy of what we're doing. So we are now actively enrolling patients at 7 centers around the United States. I'll show you the 7 centers in a trial called the CONCERT-CHF trial. In this trial, we're testing the idea that mixing 2 different types of stem cells might enhance the efficacy of what I've already shown you. These are the 7 centers. There are two right here in Florida. There's one in Texas, Minneapolis, Louisville, Stanford, and Indiana. And it's directed by the National Heart, Lung, and Blood Institute, and the data coordinating center for this is at University of Texas. We're very proud at the University of Miami that our laboratory is manufacturing all of the cells for this, and I'm very proud that the cell lab folks are right here in the audience. Now, what's the idea between mixing 2 cell types? Why would mesenchymal stem cells mixed with another cell type, a cardiac stem cell, be better than the mesenchymal stem cell alone? Well, I already told you that the mesenchymal stem cells stimulate the heart to make new cardiac stem cells. So we tested the idea that if we mix them together, that we could perhaps enhance the efficacy. And this is an experimental model from a mouse heart that shows how this works. In this experiment, which was just published a few months ago, the cardiac stem cells are labeled by this green color, green fluorescence protein. And what we've done is taken a chunk of the mouse heart that has the green fluorescence protein labeling of the cardiac stem cells and placed it on a lawn of mesenchymal stem cells. And what you see is a massive proliferation of these cardiac stem cells. Not only do they proliferate in number, but they migrate out of this tissue into the surrounding milieu. So it's this kind of biology that we think is driving a greater efficacy. We've tested this in animals to show that it works in animals, and now we're going to go on and do the clinical trial. We are in the midst of the clinical trial. So if folks are interested in participating in a clinical trial, this would be the state-of-the-art clinical trial to participate in the United States today, and it's fully funded by the public source of the National Heart, Lung, and Blood Institute. So that's what I want to say about heart disease. I want to change the topic a little bit to a broader impact on health. Can these cells do more than heal an injured heart or heal an injured brain? Can they improve the overall health of the aging human being? And what happens as we age is we all develop a condition called frailty. And frailty is a biologically driven process that underlines aging, and we all get frail, but at different rates. So if you look at different populations of older individuals, some are more frail than others. And it turns out that the biology of frailty is very similar to the biology of heart failure, and therefore our group at the University of Miami has become very interested in using the mesenchymal stem cells to treat frailty. So aging frailty is a common geriatric syndrome. It affects 10 to 15% of individuals over the age of 65. Again, it's a chronic pro-inflammatory state, just like heart failure is. There's a loss of physiologic reserve. And the more frail you are, the more likely you are to have an adverse health outcome from another medical condition that affects you. This is why this is so important. This is the impact of the aging population in the United States of America. We estimate that in 2020, just a few years from now, we will exceed 50 million people who are greater than 65 years of age. And by 2050, that'll be almost 90 million people. We live in an aging society. So one of the first things we asked as we became interested in aging was, is there a problem with the stem cells? Do they lose their effect in older individuals? And we were able to look at that from our different clinical trials. We have studied about 300 patients in cardiac clinical trials now. So we did a simple experiment where we looked at the effects of our stem cells in people with heart failure if they were less than 60 or greater than older than 60. And it turned out that was the midpoint of the age of our trials. We had the same number of folks above and below the age of 60. And we were very pleased to see that the effect of the stem cells in the heart failure population was not lost in the older patient population. If anything, it was slightly greater. So this was one encouraging sign for us that this idea of using these cells to treat frailty had some merit. This is the prevalence of frailty in Japan. Japan is the country that has the greatest number of older individuals and therefore the greatest amount of frailty. I show these data because they're dramatic, but the truth of the matter is the United States is not far behind Japan in terms of the impact of this. So if we start to look above the age of 75, you can see there's a dramatic increase in frailty. And folks who are in this group, the 80 to 84, have a 20% rate of frailty. If you're greater than 85, you've got a 35% chance of being frail. So this is a major health problem. And again, because of the things I already told you about allogeneic cells, we thought it was very reasonable to do a clinical trial of administering these cells by intravenous infusion in patients with aging frailty. However, we did this very rigorously, as I've shown you before, doing randomized clinical trials. And in this case, the data I'm about to show you, we used a placebo control. So about 2 weeks ago also, we had a very busy week where we presented 2 results. I showed you the Poseidon DCM. We also announced the results of the Kratos trial, which is allogeneic mesenchymal stem cells for the treatment of older individuals with frailty. And the background of this trial is frailty is a syndrome of multi-system physiologic dysregulation. It is characterized by having at least 3 of these factors: weight loss, exhaustion, weakness, slowness, and decreased physical activity. There's an increased vulnerability to adverse health outcomes. There's absolutely no treatment for this. We have no medicine that the entire medical community is aware of that can have a positive effect in this condition, and therefore it was very reasonable, based on what I've told you, to use mesenchymal stem cells in this condition. So the hypothesis is that, first of all, we always start with safety. That's been mentioned a number of times. The hypothesis is that the mesenchymal stem cells are safe, but could also have efficacy in this population. The study endpoints were 1 month safety, as in the other study, as defined here, and then as in the previous study, we had a long list of efficacy variables. In this study, we randomized patients into 3 groups. We wanted to, first of all, identify the optimal dose. And so one group of patients got 100 million, another got 200 million, and the third group got a placebo. This was strictly double-blind, meaning that the doctors didn't know what the patients were getting and the patients didn't know what they were getting. So all of the patients were followed for 1 year, not knowing what they got until the end. And here is the list of endpoints. Again, they characterize a patient physical capacity, quality of life, lung function, and then the inflammatory biomarkers. So just to quickly take you through these results, the 6-minute walk distance is a very important measure of how people are doing. If you're old and tired and weak and you need support, how far you can walk is a very important measure of your quality of life. If you can walk an extra 100 meters, that makes the difference between being able to go out and conduct your activities of daily living or not. So we measured the 6-minute walk distance, and lo and behold, we found that folks in this study could walk more than 50 meters more. It was an average of about 70 meters 3 months after getting the cells, and that was sustained for 6 months. The 2 bars represent, on the left, the blue is 3 months and the green is 6 months. What was very interesting was that the effect was lost in the 200 million group. So 100 was preferentially better than 200, and the placebo group did not have an effect. But there was a statistically significant 70-meter increase in walk distance in the patients with the 100 million cells. Now, we looked at another way to index this, which there's a test you can do called the Short Physical Performance Battery, and we were very pleased to see that, like the 6-minute walk distance, this was statistically improved in the 100 million group, but not seen in the 200 or the placebo. When we looked at lung function, it was also improved in the 100, but not the 200. So I hope you're seeing the pattern here, that the endpoints are the same for the frail individuals as they are for heart failure. There's the same degree of improvement using similar endpoints. There's another, this is what's called a patient-reported outcome, where the patients, you give a questionnaire and the patients tell you how much physical activity they're capable of. You see that it's actually declining in the 200 group and the placebo group, but there's a trend towards increase. Certainly there's a reversal of the decline in the 100 million group. And finally, our old friend, the TNF-alpha, well, I should say our old enemy, because TNF-alpha is bad. It's bad in heart failure and it's bad in aging. It's not nearly as high in aging as in heart failure, but it's still elevated compared to normal people, perhaps 2 to 3 times elevated, whereas it's about 10 times elevated in heart failure. And again, you see a very nice statistically significant decline in the 100 million group, lost again in 200 and placebo. We're not entirely sure why the effect is lost at the higher dose, but it is something we've seen before in heart failure trials. In the very first heart failure trial we did a dose response, and again, it's lost at 200. This is a very important lesson because it shows that too much can be bad, and this is also another critical lesson in developing therapeutics. You have to know the correct dose. People who've been doing drug development for the last century know this. The first thing you do is find the right dose, and using cells as a drug is no different. We should not assume that the more you give, the better. You have to find the right dose in an early study, and then you go on to do your pivotal trials with the right dose, the right delivery, and the right time and the right patient. So just to summarize, this is the decline in frailty It's progressive. You start with isolated functional decline, progressive functional decline, dependence, disability, incapacitation. We think that the early phase is where you could have an impact and reverse it with cell therapy. And here is the summary of what we showed in the CRADA study. There was a 44% improvement in the inflammation, a 41% improvement in the endothelial function. I didn't show you that data. Endurance is improved by about 20%, and you see the other parameters. So we are hoping, and our paradigm is that we can actually impact upon this reversible period of time in the frailty process. Bernie mentioned that we have a wonderful center at the University of Miami. It's called the Interdisciplinary Stem Cell Institute, and what we've been able to do, because we've had a clinical trial unit, is expand our sphere of activities into 20 different disease areas. As shown here in this busy chart, it lists all of the different disease areas and all of the different numbers of trials. And I'm happy to send this slide to you if you want more information and think you might fit into one of these trials or might want to refer a patient to us. But this shows what we've been able to do with limited cell availability, but very carefully doing biology, understanding the impact of the cells, and then very carefully doing a clinical trial in appropriate patients. Now, I'll just end on one note, something we're very proud and excited about. We focused on diseases of aging, heart failure, and aging frailty, but we've also recognized that cell therapy is useful to children and neonates. There's a terrible condition, a congenital heart condition called the hypoplastic left heart syndrome, where the child is actually born with a single ventricle. There's only a functioning right ventricle and a non-functioning left ventricle. A group of surgeons, very talented surgeons over the last 30 years, have figured out how to correct this over 3-staged operations, as shown here. So these children and families have to endure 3 major cardiac surgeries starting at 2 weeks of life, and only 50% of the children survive the 3 surgeries. And many of them go on to develop heart failure after the surgeries. And so we've been working with a partner at the University of Maryland, Dr. Sanjay Kushal, to see if we could add our stem cells into this mix as the 4th step in trying to make these patients better. And we've begun to enroll patients in a trial called the ELPIS trial at the University of Maryland. So you can see how cell therapy is useful for all human beings, from children to older age individuals. So just some summary comments on the CRADA trial, the aging trial. Intravenous infusion of mesenchymal stem cells is safe in older individuals with frailty. The 100 million group was better than the 200 and increased 6-minute walk distance, the short physical performance, battery score, the pulmonary function. Both groups had a favorable effect on inflammation, but taking all variables together, we believe that the 100 million dose is better, and we are planning to conduct a larger trial that looks more carefully around that dosing. So we conclude that allogeneic human mesenchymal stem cells are safe and well tolerated in older individuals with frailty, and larger pivotal clinical trials of mesenchymal stem cells for aging frailty are warranted. I want to end by thanking a very superb team of colleagues and trainees at the Miller School of Medicine. I want to particularly thank Aisha Khan, pictured here, who's the director of the Cell Laboratory, and with her team, also pictured here, has manufactured all of the products right at the university that I've talked about. And Darcy DiFetti is also here in the audience, who has served as the director of research for us, director of the clinical trial unit for the last 10 years. We have been very fortunate to be funded by several foundations, the Marcus Foundation, the Star Foundation, and the Sofer Family Foundation, and we'll be honoring some of these foundations tomorrow night. at the dinner. And also, I have to mention that this would not have been possible without the public support through the National Heart, Lung, and Blood Institute, through several mechanisms, one being the Specialized Center for Cell-Based Therapy, and the other active program right now, the Cardiovascular Cell Therapy Research Network, that's conducting 3 or 4 different clinical trials. If it wasn't for the public support, we would not be able to get to the point where we could take advantage of the foundation support to accelerate the trials. So I believe we've made a lot of progress in this field. It's very exciting. I think there's some therapeutic potential here for the use of mesenchymal stem cells. I think that the new generation of cells, embryonic stem cells and iPS cells, will enter into clinical trials, but are probably about 10 years behind where we are now, because we haven't even started to define the safety profile of iPS or ESC cells in human populations. That work is just about to begin. So it's, I think, a very exciting time in regenerative medicine. I think if we talk about the hope versus hype paradigm, there is some real hope, and there is significant data that is out there, and data that is accumulating. And I think the way forward for this field is to continue to do the rigorous clinical trials that have the solid foundation in basic science. So I'll end there. I'll thank you all very much for your attention.
For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
we're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. We would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine.
Hello? Are we on? Well, welcome. It's the afternoon session of the public day. My name is Bernard Siegel. I'm Executive Director of the Regenerative Medicine Foundation and the founder and chair of the World Stem Cell Summit. And we are in the afternoon session of our public day. So as you know, the sessions from our grand ballroom are being live streamed around the world at worldstemcellsummit.com. This topic is really of interest to me personally because I find the whole concept of frozen zoo, the fact that we might be cloning or saving from extinction the northern rhino, and all of the esoteric, interesting, popular culture aspects from the technologies that we have today are sort of brought home with the individuals that I will be introducing for this session. One of the speakers is my dear friend and colleague, Dr. Jean Loring from the Scripps Research Institute in La Jolla. And Jean is a member of the Science Advisory Board of the Regenerative Medicine Foundation. Her work is exemplary in all aspects of the field. She's also a commentator on policy issues and an advocate's advocate, a Parkinson's disease advocate, someone of extraordinary talent, and she's also lent those talents to the Frozen Zoo. Also, we have Oliver Ryder of the Frozen Zoo, who is here with us today, which is a fascinating project. So this is an opportunity for those that are in the room and live stream to learn more about this exciting project. So I'll leave it to both of you to present the session. Thank you.
Thanks, Barry. Good job, guys. So what you see on the screen right now is NOLA. NOLA was the last of the northern white rhinos that lived at the San Diego Wild Animal Park. This was a film taken in a few years ago. She died in 2015, and you'll see what the consequences of that is in a moment. She's a northern white rhino. There are currently, they have been hunted almost to extinction. In fact, they are practically extinct in the wild. And they've been hunted for their horns, which are made of keratin, which is the same thing that your fingernails and hair are made out of. So the idea that this keratin comes from a rhino imbues it with magical properties that makes it very popular, especially in the Far East, as a medical and also for decorations. But the consequence is that animals like this are becoming extinct. So I'm going to introduce the idea. Since this is a stem cell meeting and I'm a stem cell biologist, I want to let you know what my connection is and stem cells' connection is to this northern white rhino and the issue of extinct species. Just to give you a little bit of a review, the first pluripotent stem cells were it doesn't really work. The first pluripotent stem cells from humans were derived in 1998 from discarded embryos. They use the same methods that have been developed in 1981 to make mouse embryonic stem cells. So we've had the concept of pluripotent stem cells around for a very long time. What made a huge difference and made stem cells a lot more useful than they had been was the advent of reprogramming technology. In 2007, Shinya Yamanaka took some skin cells from a human, reprogrammed them into induced pluripotent stem cells, which are, in fact, exactly the same as embryonic stem cells. These cells can vary from each other, but those variations are trivial. They still have the ability to make all cells from the root of a tree and all the branches and all the cell types that you can make from those pluripotent cells by following a particular pathway. And finding that particular pathway is really the challenge that we have now in order to make cells that will be useful for clinical use and also, as you'll see, for conservation use. And you'll notice down here, in the lower left-hand corner are gametes, which is the key to using these cells for restoring endangered species. Okay, so what I want to talk to you about is a collaboration that started in 2008 with my friend Oliver Ryder, who's the director of conservation research, the Frozen Zoo, at the San Diego Wild Animal Park, as it was known then, now called the Safari Park. And what Oliver had been working on for much of his life was freezing specimens of animals before they died, after they died, collecting them from all over the world, and cryopreserving them in a way so that they would be still viable. And as you see from the map on the right, we're only about 30 miles apart, so it's not a really very long drive for me to go up to the zoo or for Oliver to come down to my lab. So we figured that if you could make pluripotent stem cells from humans using these reprogramming factors, why couldn't you make pluripotent stem cells from other animals using the same methods? So that's what we did, and surprisingly, we found that we could use exactly the same methods, that is, using the human genes that are used for reprogramming. They worked on northern white rhinos. So that was a surprise, obviously, and it laid the groundwork for everything that's come since. We published this work in 2011. I'm trying to keep track of the dates here. 2011, we published this in Nature Methods paper, and as you'll see, we got the cover, which is essentially an ark filled with all those animals that we're going to be trying to rescue. This work was done by a postdoc in my lab called Inbar Friedrich Ben Nunn. We took cells from the Frozen Zoo, we thawed them, they're fibroblasts from skin, we reprogrammed them, and we came up with pluripotent stem cells. And the stem cells look very much like the human pluripotent stem cells do, and they stain with some of the same markers, some of the same markers that tell you that the cells are pluripotent. In this case, I'm showing you nanog, which is a nuclear protein, which is typical of pluripotent cells. And they had a normal karyotype, and normal for a northern white rhino is 82 chromosomes. So we had the karyotype done on 3 of the clones that we obtained from this culture, and they all were pretty much normal karyotype, although there are a few variations, which is another subject, I think. Okay, so we started by reprogramming cells from Fatu, who is on the lower left-hand corner. She's the youngest of the animals. We thought that might be an advantage. It turns out it doesn't matter. But as you'll see, when that is not good. Can you turn off my backup, please? Wasn't supposed to be doing that. So thank you. Oh, yes. All right, we're moving forward. There's always something. Okay, so there we are, thank you. So when we did in 2008, when we first started the project, there were 8 northern white rhinos in the world. There were 2 of them at the San Diego Safari Park, 2 in the Czech Republic, and then a group of 4 others that had been transferred to a special reserve, a conservancy in Kenya for their protection. And then by now, there are only 3 left. The 3 that are left are 2 females and a male that live in the conservation location in Kenya. So, and a lot of them have died recently. As I said, Nola, our local northern white rhino, died in late 2015. This is the conservancy. You can see there are 3 guards for this one rhino. That's necessary because poachers still want these rhinos in spite of the fact that they're harder to get to than the other rhinos in Africa. So the next thing that happened really was in 2005 when we had a conference organized by Oliver Ryder and a colleague of his in Germany, Thomas Hildebrand. in order to get a bunch of people together to ask whether we might be able to use these technologies, like stem cell technologies, to restore a species. And the meeting was held at one of the oldest zoos in the world in Vienna, Austria, in the winter. And it's really quite lovely to have, I guess the people at the zoo are used to this. They get to have meetings at the zoo all the time. But those of us who are lab-bound don't normally get to go to conferences at zoos. So it was really interesting, and the people there were extremely diverse. This is some of them. This is what we call the scientist exhibit, which is a temporary exhibit at the zoo. It was only there for the time that we were there. And that is one of the original cages. And interestingly, and also very efficiently, this group put together a publication which is published in Zoo Biology. I had not published in Zoo Biology before, but it was remarkable because the editor did not send it out for review, for peer review. He decided to accept it as is, and that's never happened to me before, so I thought maybe I should be publishing in Zoo Biology more often. But it was really, we talked about the whole process, which I'm going to leave to Oliver to tell you more about how we could start over on the left there with rhinos, and on the bottom you can see the pathway for going from rhino skin to induced pluripotent stem cells, and from there to primordial germ cells, to gametes, to embryos. So we're started on the path. We're at the first step. I wanted to introduce Oliver. We've had a long-term collaboration. You can see in this picture, Oliver is the one standing up. I'm the one in the white hat. And this is one of the last pictures we had taken with Nola at the San Diego Wild Animal Park. Before Oliver comes up, I want to show you something that you could have. You could have a picture of northern white rhino iPS cells on a t-shirt. And I especially like this front part. And that's an actual micrograph of the northern white rhino cells. Oliver.
Thank you, Jean. It's a pleasure to come here to talk to this audience. It's a new one for me. Most of my meetings are in zoos. And this is my first trip to West Palm Beach. But stem cells have changed my life because I believe they are changing the possibilities for what I want to work on, which is conservation. Let's see, I advance the green button. So this is a picture of a northern white rhino in the wild in Garamba National Park in what was Zaire, now Democratic Republic of the Congo. It died, we don't exactly know how, but most likely at the hand of man. There are none left there. It was a sentinel, it's an emotional image for those of us who work on saving it. Imagine, this is what we would like to see in the future, is to have these rhinos back in the wild. But they are basically functionally extinct now. But Jean Loring mentioned the frozen zoo. And it's a small room, but maybe a very, very important one. It's important to save cells for conservation applications, and the frozen zoo is a collection of living cell materials, early passage diploid fibroblasts. Our collection is the most extensive, most diverse, best characterized, and most utilized collection of its kind. We have cells from approximately 10,000 vertebrate individuals that represent 1,000 species. It's contributed to hundreds of scientific studies. Studies such as naming species, describing the tree of life, and for managing small populations. Those are some of the conservation applications, but one of its major uses has been in human medical research. The greatest users of the frozen zoo are researchers at NIH, Howard Hughes investigators, investigators at medical schools, who want to understand the evolution of the human genome, the evolution of our physiology and our susceptibilities to disease. So they ask us for samples from animals, many of which are endangered, and we are a contact point that can provide these kinds of samples. The fibroblasts, as I said, we have different orders of, most of the samples are mammals, but we have birds. It's one of the most rapidly growing sessions are birds, reptiles. We're the only place in the world, I think, right now that's banking viable amphibian cell cultures. Amphibians are under a great conservation threat. It's suggested by the World Conservation Organization, IUCN, that publishes the red list that 30% of the amphibian species of the planet are in imminent danger of extinction. We're going to want to study these animals, study their diseases. We're going to want to do that in vitro. We're going to need cell cultures. And so we're trying to establish methods to bank them. They're not as easy to grow as mammalian cells. And then we also have my colleague, Dr. Barbara Durant, has been saving sperm and reproductive tissues. And she also has a very extensive collection. We have frozen tissues and DNAs, so it's really a very remarkable resource, one that we've used, as I mentioned, for many conservation studies, but we always knew that it would be used for things that we didn't yet appreciate were going to take place because of scientific advances. And one of the two of these that are most remarkable to me was learning about Dolly, which meant that we could turn the cells in the frozen zoo into animals, and then learning about the report out of Shinya Yamanaka's lab that fibroblasts could be turned into induced pluripotent stem cells. So at that point, Jean and I started to work together. So the idea is that if we can turn these cells into animals, can they be used for genetic rescue? Well, genetic rescue is a real phenomenon that is known. It's to produce increase the robustness of small imperiled populations via migration. It's usually been accomplished by moving an animal into a population that's genetically diminished. And the Florida panther in this state is a case in point where that species was on the brink of extinction. A few females were brought from Texas. They were allowed to reproduce. They were removed and kept in captivity for the rest of their lives, but their young caused a resurgence in fitness, we call it, of the population. And so the population is growing now. The major threat now is they're being killed on the roads. So genetic rescue is a real phenomenon, but could it be done with cells? Well, in some extreme cases, genetic rescue may depend on access to viable cells from appropriate individuals. And the northern white rhinoceros, Nola here, who you've already met, is a case in point. And it's this project is the subject of an enormous commitment by my organization, the San Diego Zoo Global, a nonprofit zoo, and that has two campuses, the San Diego Zoo and the San Diego Zoo Safari Park, and also has the San Diego Zoo Institute for Conservation Research, where I've worked for 40 years. Well, commitments are also being made by others, like Jean Loring, and so we have a growing effort to try to use advanced genetic and reproductive technologies to prevent the extinction of a species. Well, you ask, well, what are we talking about? This is the northern white rhinoceros. Is there a southern white rhinoceros? Well, yes, there is. The southern white rhinoceros went through a genetic bottleneck a century ago. As few as 30 individuals remained. And the northern white rhinoceros is at the headwaters of the Nile. And so how different are they? Well, I was asked that question in the mid-1980s, and we did the first genetic studies on northern white rhinos, and we could easily see they were different from southern white rhinos. More recently, we've said the way to really find out how different they are is to sequence their genomes, so we've done that for 8 northern white rhino individuals from the cell cultures and southern white rhinos, and done a comparison, and we now know that they are really distinct, evolutionary, diverged populations differentiated at the subspecies level, which in some ways is a good sign, because they're much more, because in order for this project to succeed, it's going to require having surrogate mothers. And those surrogate mothers are not going to be able to be northern white rhinos. There's no female northern white rhino that's capable of carrying a pregnancy. So we're going to use southern white rhinos as surrogate mothers for northern white rhino embryos. And I'll tell you a little more about how we plan to do that. But first, here's a little more detail about the history of the samples that we have. We have 8 unrelated individuals. The 3 surviving individuals are in the orange, shaded in orange there, that have been removed to the old Pejeta Conservancy in Kenya. But you'll see there a father, his daughter, and his granddaughter. And I mentioned the females are not really capable of carrying a pregnancy, so this is a species that's doomed. It's alive, but it's functionally extinct. There may be a hope of collecting ova from these females, and there's a lot of effort to discuss how we might do that in that remote location. So the meeting in Vienna just a year ago that Jean Loring mentioned produced this roadmap that my colleague likes to say looks like traffic in Los Angeles at 2 p.m. in the afternoon. But it's an effort to construct all of the possible ways that we might be able to reach the goal being a self-sustaining population of northern white rhinos. Now, are those cells enough to do that? Well, when we sequenced the genomes, we looked at their genetic diversity at the DNA sequence level and compared it to southern white rhinos. And the northern white rhino, the genetic diversity is, well, first of all, it's more than humans. There's greater SNP density in these rhinos than there is in humans. And there's a comparable amount in the northern white rhino cell cultures as exists in the population of southern white rhinos that successfully went through the bottleneck that I mentioned before and grew to 18,000 individuals. So we believe if we can get to an interbreeding population of 30 individuals, there's no genetic reason why they shouldn't be able to expand their numbers successfully, as did the southern white rhino. So the overall goal is a self-sustaining population. To do that, we have to produce the first northern white rhinoceros calf. To do that, we're going to have an embryo transferred, as I mentioned, and to do that, we're going to need to have surrogates, and we're going to need to have northern white rhino embryos. So one of the ways that I'll focus on to make those embryos would be to use induced pluripotent stem cells to direct them to primordial germ cells that would then make sperm or eggs depending on their chromosomal constitution. Those materials would be the source of zygotes for embryo maturation, embryo development and maturation, and then transfer into the surrogate females. So this is a plane that brought surrogate females. We had to charter a plane to bring 6 rhinos to San Diego. And they arrived and got a police escort to the safari park and in the middle of the night. And they do the floodlights, loaders, crates, and biosecurity lights and everything remind you of any movie? Well, that's just my colleague's dramatic over-representation. We're not going to use the Jurassic Park word because that's not, don't tell them that. That's not what we really want to emphasize. What we want to emphasize is that this is we want to focus on the science because all of the efforts of traditional forms of protection, traditional forms of husbandry and trying to propagate these animals in zoos have failed. And now it's down to scientific innovation to make the difference, whether a species survives or not. So these are the females that we brought, 6 that now have been at the safari park for about a year. They are aged now between about 4 and 10 years old, and they are being trained. They're not. Most of our rhinos at the safari park, which is, if you're not familiar with it, is a very large facility. It's over 900 acres, and one of the enclosures is the size of the whole San Diego Zoo. It has mixed species exhibits. We have rhinos in there with multiple other species, and these the animals behave in a very wild way. But these females are being treated in a very much different way. They're being trained with positive reinforcement only, and we constructed a facility. It didn't work. We constructed a facility for them that was put up very quickly, where they live outside, and because the weather's good in Escondido, and we have a very talented staff of keepers and trainers that work with them. And it's right after they came, they were given reproductive exams. We brought in the world's experts in rhino reproduction and in advanced reproductive technologies, and did ultrasound exams to determine their reproductive status, and we could see that the females were cycling. They had ovarian follicles, and that started our baseline data collection. Now what we're doing, or what's been done, incredible work over the past year, can you click that for me so the video gets going, is to train the females for reproductive monitoring. Rhinos are really very intelligent, and white rhinos are social animals, and our keepers have been able to train them using standard methods of animal training to walk into this chute that can protect us, or not me, but protect the people who are the reproductive physiologists and veterinarians to do exams on unanesthetized animals. So the animals will of their own volition come into a crate. You can see this being given a target, and she approaches the target, gets a bite of hay. They're very food motivated, and they love to be petted. And so we now are have learned a great deal about these females and their reproductive cycles. And one of my colleagues, Dr. Parker Pennington, is getting ready to do an ultrasound exam on a rhino. And she does this now. Basically, all of the females are getting ultrasound exams basically almost on a daily basis. So those represent an enormous effort and investment to provide this surrogate gene pool. And we're going to, because the domestic horse is the closest domestic species to the rhinoceros, we're going to take information that's used in the domestic horse reproduction industry for like collecting ova and doing embryo transfer methods, because nobody's yet done an embryo transfer in a rhinoceros. And nobody's tried because we really haven't had many rhino embryos to work with. So we have partners in South Africa, and because of the dire situation for southern white rhinos now, the extraordinary poaching pressure, There are ranches that have rhinos in high security zones to protect them, but what that means is that they're closely managed. You can't really say they're living in the wild. They're living in a very high security compound. And some of those animals are made available for our research efforts and contributes to this project. So to bring you a little more up to date now, the publication that Jean Loring talked about from 2011 used lentiviruses, viruses that integrated to make the stem cells. Colleagues in Jean Loring's lab, Dr. Marisa Karodi and Tom Nguyen, are making stem cells now using non-integrative methods, using Sendai virus. And we have stem cells now from more individuals. Our goal is to make stem cells from all 12 individuals and then proceed with the in vitro development. I have to acknowledge the incredible team of people who've been banking cells and who are the world's expert on culturing mammalian cells. We just have this amazing collection and its diversity, and this work wouldn't be possible without them. We have a growing collaborative network that's very encouraging. The Scripps Research Institute and the San Diego Zoo has started the scientific collaborations for animal resources. It started with a zoo in the Czech Republic, and over time these relationships have just proliferated, and we now have colleagues in Japan, in Shinya Yamanaka's institute, and this is proceeding in a dramatic way. We learned earlier today about how teaching children about cells can can broaden their educational perspective. And I think that that's really important. Cells are life. Every organism, whether it's a single-cell organism or a multi-cell organism, begins its life or lives its life as a single cell. All of the experiments of biology that have produced the diversity of life forms that have lived on this earth, all of those experiments have taken place in cells. And to be able to bank those cells and use that knowledge for conserving biodiversity and for remediating some of the losses that take place in individuals, the diseases, or take place in the biosphere in terms of loss of species, is just an amazing activity that this is the first time in the history of life on Earth that can be done. Thank you.
I think we have time for just a couple of questions, but I did want to point out again, if you are interested in rhino iPS cells, this is an actual photo of a colony. It's one of the first colonies that we made, and you'll see that there's a little cutout on the other side shaped like a rhino. There are also puzzles available that actually have little rhino shape. The people from Illis who talked this morning are responsible for making these shirts. They're responsible for providing them to you, and they also have available instructions, at least, about how to get some of the puzzles that are made out of these photographs, these photo micrographs. So do we have one or two questions? No. Yes. Could you go to the microphone? Sorry. Is there one over there?
Hi, I'm Christina from Virginia Tech. Have you had any success in generating gametes out of these iPS cells?
I didn't hear what you said, but go ahead.
Well, gametes, the quick answer is we have not. But in the mouse, people have gone from a fibroblast to iPS cells to primordial germ cells to sperm and eggs to produce in vitro embryos that were transplanted into surrogate females that gave birth to pups that grew up to breed normally. So that's what we want to do. We want to do with the rhino what's been done with the mouse. Our major major hurdles or major challenges that we have to face, and because of the lack of knowledge of reproductive biology and of rhinos, and a big one too is they have a 16-month gestation period.
More or less.
More or less.
So I just want to point out that all of this started with mice, 1981, first mouse embryonic stem cells. So, and then the next thing that happened, which was remarkable, was the human iPS cells. But then I think it's remarkable that we discovered that we could treat the rhino cells just like human cells and reprogram them with exactly the same genes. And that was a real breakthrough for us. Yes?
I just wondered about the northern white rhino and if there is habitat left, if you succeeded in this venture.
That's a great question. People say, like, well, what about, could you put animals back into the habitat? The unfortunate thing about rhinos is that they've been selectively removed from their habitat. Their numbers are down 99%, 99.9% in the last 50 years. There's plenty of habitat for them. They've just been removed. So making secure habitat for them will require political stability and probably high levels of protection. But I think that we ought to embrace the notion that that's possible. And it's likely to be encouraged by, if we would have animals available, we could say, you know, now we'd like to see these go back. There's another question.
Oh.
Yes, a question here. Can you imagine like a cascade process where like you're using the southern whites as the surrogates for the northern whites, that there might be like an eastern white or western white that is not compatible to have the southern as the surrogate, but maybe the northern, so that you do a cascade process to sort of go further back in lineages to get really extinct species back?
That's a good question, and we don't know a lot about surrogacy. We know about hybrids that have been able to be made in the history of breeding animals. We know something about the evolutionary tree, the phylogeny of rhinos, and the northern white rhino and the southern white rhino are diverged probably 100,000 years ago. The closest relative is the black rhino, to them is a black rhino that's more like 8 or 9 million years. Whether they're going to be like placental incompatibilities, that's an area of important area for research to see how far it might be extended, or to see whether modifications can be made to make some species more likely to be able to be surrogates for other species.
I see. And another question, like how scalable do you think this might be able to be? Do you think there could be recovery at some time and efficient methods for recovering of thousands of species? Mm-hmm. It's really good to do it for the one, but what about for the thousands others?
This is our example. Can I do it?
Jean wants to take the question.
No, it's all right. We're very compatible. We see this as the first of many. As I said earlier, I think Oliver chose the species because this is one he's very fond of, and it's obviously been a very evocative sort of animal to try to bring back, but there are lots of animals that are becoming extinct, and if we can make iPS cells and primordial germ cells from them, if we can go through the whole process of surrogacy, then we should be able, this could be a normal technique, normal conservation method.
Thank you.
Can I just add on quickly to that? Yeah, I suggested the northern white rhinoceros, not because it was going to be the easiest, but it was very clear to me that this was the only way to prevent its extinction. The fast progress will be made for endangered species that are closely related to domestic species whose physiology and reproduction we understand well. And we have a number of candidate projects in mind, but our hands are full right now with this one.
I think we better wrap it up. Thank you very much. If you want a T-shirt over there on, right there. We have another session coming in, so we should, yeah.
Over 3 minutes.
Oh, we have 3 minutes, okay. 3 minutes to get a T-shirt.
So let's have a round of applause for our speakers, and hopefully we'll be seeing a lot more northern white rhinos. And we have Mark Hupke here, and some of these t-shirts are being autographed, correct? Yes. And they are going to be made available for sale for a worthy cause. And the next session will start in 3 minutes. So can we advance the slide, please, to the next session? Hello, everyone. My name is Bernard Siegel. I'm Executive Director of the Regenerative Medicine Foundation and Chair of the annual World Stem Cell Summit. We've all learned a lot of things this morning already. We've also even learned how we're going to save the northern white rhino from extinction, we hope. The sessions in this room are very special. Some of them are futuristic, some of them are a little esoteric, but I assure you, in a world that is future shock, a future shock world, what you're hearing from this podium is very, very important. For our next speaker, I'm really honored to introduce Eve Harold. Eve Harold served as the Director of Public Policy Research and Education of our organization, the Genetics Policy Institute, for approximately 2 years. At our very formative stage, Eve was instrumental in our first 2 World Stem Cell Summits that we produced, one at Baylor College of Medicine in Houston in 2005 and at Stanford University in 2006. Indeed, Eve, you were present at the creation. Eve also was present at the creation of the policy debates over embryonic stem cell research that happened back in the early 2000s and wrote a phenomenal book called Stem Cell Wars, Stories from the Front Lines, which is one of the few volumes that really discuss the history and the socio-political fault lines over embryonic stem cell research. It's a very, very important book. And her latest book, which I'm very proud to have the back cover blurb, or front cover blurb, I think it's the back cover blurb, is Beyond Human. Those books will be available for sale, I think, outside, and Eve is going to be signing some autographs, hopefully, for this. This book is on its way to be a bestseller. Kirkus Reviews gave it a rave notice and called it an extremely important book. What Eve has done in this volume is describe really the future of humankind, and the amazing convergence of technologies that I referred to earlier, and why this is a very special time for our species. Eve has honored us to come here today and talk about real anti-aging, which is a provocative title if there ever was one, the soaring potential of converging technologies. Eve.
Thank you so much, and thank you, Bernie, for inviting me to be here. It's a real honor to be part of this event. And it's amazing to me when I think of the early meetings that we had at the very inception of the organization, and to see what it's grown to today, and it's very gratifying, and I'm honored to be here. So let's see. I don't know why this isn't working. Doesn't seem to be working. Really? Well, I'm glad I brought them. Sorry about that. They didn't have my slides, but fortunately I brought a backup. Always bring a backup. That's my prime directive. Should take just a minute to get that set up. Mm-hmm. I wrote the book because I was living in Washington, D.C. at the time, and had been involved with some medical nonprofits. And one of them was the Stem Cell Research Foundation, one of the very first organizations that promoted stem cell research and actually provided funding for it. And it was just such a revelation to me, working in that field, to see the opposition and how it was opposed by right-to-life groups and kind of taken over, kind of co-opted as part of the right-to-life battle. And a lot of misinformation was going around because of that. I ended up getting into doing education about it because I wanted to kind of dispel some of that misinformation. And also, I was able, through the groups that I was affiliated with, to start to do some lobbying on Capitol Hill to try to educate legislators, to promote funding and a better environment legally for funding and legality as far as doing the research. So it really was quite a battle, and actually Bernie is the brainstorm behind the title Stem Cell Wars, because it really was a bit of a war going on. Very, very passionate on both sides, and it was an amazing experience. Okay, I think we're set up now. So I'm going to talk about the aging revolution, and I talk about a revolution not because, just people are getting older and living longer. I think we're all pretty much aware of the fact that the lifespan has expanded dramatically in the last century, and in fact, in the United States, in the turn of the century, in 1900, the average life expectancy was 47, and now it's 78.8, which is a bit of a revolution in and of itself. So, as I mentioned, people are living longer, and this is true throughout the industrialized world and even in a lot of developing nations as well. The lifespan is getting longer and longer. But that's not the only thing that's revolutionary about today. Not only are people living longer, they're having fewer age-related diseases. So we're living longer, healthier. And this has led to something that sociologists call the rectangularization of the lifespan. And what that means is that compared to previous generations, people tend to go along today and live in a relatively healthy state up until the very end of their life. So the period of mortality, when maybe there are several conditions or there may be one condition like cancer that is leading to the end of life, is tending to be compressed into the last few weeks of life. So people are spending less time disabled. We're actually getting better at aging and living much better towards the end of our life. Most of the life extension that's taken place now is called technophysio-evolution. This isn't from any kind of fancy science. This is all stuff that we've done just as society progresses and as economies develop. So it's related to things like nutritious diets, temperature-controlled dwellings and warm clothing and better environmental control, better hygiene and sanitation services is cutting down on infectious diseases. Vaccinations and antibiotics have made a huge difference in terms of longevity, and especially when it comes to children and young people not getting some of the diseases and infections that used to cut life very, very short in the 19th century and before. Also, leisure time has brought in an age when people actually do engage in healthy exercise. We kind of think in the past people might have been really physically fit because they were doing farming and things like that. But in reality, what they did was a lot of repetitive movement. It wasn't necessarily cardio workout. It wasn't necessarily healthy, and it tended to lead to a lot of chronic conditions like joint deterioration and bone loss. And then the whole project of modern medicine, just the fact that we now do bypass operations for people who have cardiovascular disease, we're able to extend the life in people who have cancer. Just the whole project of modern medicine has really extended the human lifespan. This is nothing exciting or revolutionary, but it's about to become very, very different. And as different as our old age looks to that of our grandparents, the old age of our children is going to be radically different even from ours, and this is for several reasons. First of all, life expectancy has doubled in the last 150 years without any major anti-aging medicine, but anti-aging is now an actual field. It's kind of coming out of the closet. In the past 20 years, scientists have started to hone in on and to identify targets that can be addressed in terms of anti-aging. So now we know much more about what happens as cells age and die. And that is something that is allowing us to learn how to address those various problems one by one and hopefully have exponential results. And then the last thing is converging technologies, which I'll talk a little bit about later, in which a great number of radical new therapies are now coming onto the market or in clinical trials that can really radically extend lifespan and health span. So one of the most well-known ways to retard aging is calorie restriction. This has been done in animals, and it's also been observed in humans as well. Extreme calorie restriction puts your cells into a kind of a hibernation state. They don't degenerate, they don't go into a state of senescence quite so quickly. They tend to kind of not use a lot of nutrients and resources. And then they don't age and die as quickly as if you were eating a normal diet. And so this has been found not only to work in a whole body sense, it also delays the onset of age-related diseases like Alzheimer's and heart disease. It's been shown to work in a whole range of species. Everything from yeast to mammals to human beings have been found to have this amazing ability to slow or retard aging strictly by chronic calorie restriction. And in fact, in mice, it can extend the lifespan for up to 45%. It's not the most realistic thing in the world. So a lot of people kind of look at that and say, well, that's great. I think that's wonderful if you can do it, but I'm not going to walk around being hungry all the time. I mean, what good is a long life if you can't eat adequately? So there's also a lot of research going on into the development of an anti-aging drug. The most promising drug, and probably the most famous to this point, is a drug called rapamycin. Rapamycin was derived from a bacterium that was found in, of all places, beneath one of the statues on Easter Island. There was a bacterium. It was discovered by a Pakistani-American researcher who was then able to isolate the different properties and to come up with rapamycin. It's being used already to suppress the immune system in certain cancer, I meant to say, I think that's incorrect, I meant to say transplants. It's used to suppress the immune response in certain transplants, and it's also used routinely in the coating of cardiac stents. So it's been found to delay the onset of Alzheimer's disease, cancer, heart disease, and it prevents the normal signs of aging in multiple organ systems. And the way it does this is by creating conditions that mimic calorie restriction. So rapamycin essentially does pretty much the same thing, only under conditions of a normal diet. There are other drugs that are out there available today, and it's possible that if rapamycin runs into some kind of roadblocks to getting approved by the FDA for a treatment in aging, some of us may be on a cocktail of drugs that prevent aging in different organ systems. These are all in existence today. One of them is metformin, which is a diabetes drug. That has been shown to extend the lives of mice. Dasatinib, an anti-cancer drug that destroys senescent cells. The reason that's important is because senescent cells basically become toxic in the body. They create toxic byproducts, and they're very hard for the body to break down and clear away, and they lead directly to a lot of the signs of aging. Then there's a drug called bimagrumab, which targets muscle loss and general frailty. So we all lose muscles as we get older, and after the decade of our 40s, we basically lose about 10% of our muscle mass every year. This drug, though, actually helps you to maintain muscle mass without necessarily exercising. If you include that with exercising, then you could very well have the same muscle strength as a person in their, say, 30s or 40s indefinitely. And then there are drugs that target the loss of cartilage in knees and joints. This would be a holy grail for people with arthritis and joint and bone degenerative diseases. So it's possible that we may all be on a cocktail of drugs soon, and if that extends our lifespan and our vitality and our health span, we may be alive when even more radical technologies come onto the market. So one of those areas that is very exciting in terms of actually radically extending the human lifespan is gene editing. And the way that scientists are going about this to learn what particular genes govern which particular processes is to mine nature and to observe what happens with other species, because not all species age the same way that human beings age. So some researchers at the Buck Institute, which is an anti-aging research institute, were able to basically quintuple the lifespans of worms by manipulating their DNA. So scientists can identify that particular strand of DNA, it's possible it could be inserted into a strand of human DNA, and this is called gene editing. Some species, such as Hydra, which is an aquatic creature, doesn't appear to age at all. It lives until it gets eaten by a predator and then it dies. It's possible that sometime in the future, if we can identify those particular snippets of DNA that govern the lifelong longevity of that species and splice that into the human DNA gene, then it's possible that human beings might actually be able to mimic those results. And a lot of people have talked about this and written about this, and I think there's a certain amount of skepticism out there as far as what can eventually come from gene editing based on mining information from other species. And the objection is that, well, it takes a very long time to sequence genomes. And it would take so many years to, elucidate which genetic processes are at play here, that this sort of technology is way into the future. Well, it's not way into the future because now we have super powerful computers that can sequence genomes in very short amount of time. I think HIV was sequenced in about 15 years. It took 15 years to sequence the genome. The genome for SARS, which was done much more recently, took place over a 15-day period. So we actually do have, with the help of computers now, the ability to sequence many, many genes. And, there may be many gene processes that can be teased out over the next few years, and it's something that I think could be in the foreseeable future. And then, of course, there's stem cell therapies, which everyone here is hearing more about. Stem cells obviously have tremendous potential to treat all kinds of diseases. They can turn into the tissues of any type of body part that you might be losing through age or disease. But in addition to that, the study of pluripotent stem cells in the lab can teach us kind of what's going on in very young cells and what is it that makes them healthy and makes them able to proliferate and to stay young and divide. We can learn a lot of that from observing stem cells in the lab. And then, of course, we can replace diseased tissues in the body. And then looking forward even farther into the future, and this is strictly theoretical at the moment, but there is a good bit of theory out there about nanotechnology. Nanotechnology, for those of you who aren't familiar, is using technologies that are on the size scale of 1/1000th to 1/110,000th the width of a human hair. So we're talking about work that's being done at the atomic level. So nanotechnology, in a way, is a convergence of many different fields. It's a convergence of chemistry, biology, physics, computing, and robotics. And the way that it's foreseen that it may potentially be able to cure aging is to create a countless number of tiny, tiny nanobots. This is being approached as an engineering problem, something that again has been criticized. Well, we don't want to look at the body as a product or as a machine because that's not really realistic. The body's more complex than that. Well, it's not necessarily more complex than that. Nanotechnology includes tiny programmable computers that would be injected into the bloodstream, and they would not need a battery because they would be powered by chemicals that occur naturally in the body. Nano-sized drugs and robots can penetrate cells, tissues, molecules, and repair them on the molecular level. They can hone in on broken DNA and fix it. Broken DNA is a major factor in aging. So the way that these nanobots would do that is they would be programmed with a blueprint for the healthy the genome of the person who's receiving that injection. So the nanobots go through the body and they tumble through the bloodstream, and they're able to identify DNA working by molecule. They can identify broken DNA, and then they can rebuild it based on the healthy DNA blueprint. And theoretically, what scientists hope they can do is reverse aging throughout the entire body, because nanobots will be able to go anywhere that if a blood cell can go there, nanobots can go there. So the thing that is exciting today that I write about a lot in my book, Beyond Human, is something called Converging Sciences. A lot of science fields have now started to become mature and to reach a stage of development where they can be combined with other sciences and come up with very, very novel, interesting, exciting new therapies. So some of these fields, this is just some of them: physics, biology, neuroscience, chemistry, computing, electronics, microelectronics, software development. And these are coming up with what we call converging technologies, and that's a way of combining information technology, biological information, cognitive science, and nanotechnology. Some other areas that are being used in converging technologies are cell therapies, as in stem cells, bioengineering, or the engineering of new body parts to replace broken ones, nanotechnology, wireless microelectronics. So now we have a whole spate of different kinds of medical products that can be housed in the body, that can be implanted in the body, and can be controlled and programmed and monitored without wires, without hoses. They work on wireless technology just like our computers do. And then robotics would also play a role. Doesn't seem to be working. So this is giving birth to some very interesting and new approaches to education. Scientists are becoming prepared to work with engineers and doctors and people in other specialties to bring together the various sciences and to integrate them in new ways. And one example is at the Johns Hopkins University, where there is a new academic track under the Institute for Computational Medicine, and that includes mathematics, computer science, engineering, medicine, and genetics. So in order to do this, and these collaborations are happening all over the country now, but to prepare students to be able to take part in this kind of developmental science, they need to have a very broad-ranged education. So medical doctors need to speak the language of computer scientists. Computer scientists need to be able to think like biologists. Bioengineers need to understand all the various sciences that go into the products that they create and put them together. So coming up are a few examples of some technologies that are out there now that converge many technologies, and that can be gotten today either on an experimental basis or they're actually on the marketplace. One of them is roboticized prostheses that operate with brain implants so that they can be operated purely by the thoughts of the patient. Also very well known, artificial joints. There are a lot of people getting artificial hips and knees these days. And of course, there's a long history in heart devices that take over some function of the heart or assist with the heart's function. This is a left ventricle assist device. Those have been around for quite a few years, but you also have cardiac defibrillators that will shock the heart in the case of an arrhythmia, and several other types of heart assist devices. But you also now have a total artificial heart. And this is being given to people currently in America and in Europe as a bridge to transplant. So I wrote about a woman in my book who had a total artificial heart. She was only 40 years old, and a virus attacked her heart valves and basically destroyed her heart. It was unsalvageable. At the time, there was no biological organ available when her heart gave out, and she was given a total artificial heart, which lasted about 40 days, and with tremendous results. She felt great. She walked, talked, exercised, went about her business, worked, did everything that she needed to do. But it had a few drawbacks. I mean, one of them was that she had a hose that she carried a processor in a backpack, and there was a hose that just reached around and let me see, with an open incision that is used on that. I don't know why I'm having a problem with this. There we go. So now that lasted about 40 days in that case, and she said it was just amazingly successful. She did get a biological heart after that, and now is doing pretty well on her biological heart. But the advantage of these hearts is that they cannot be rejected by the body. It's all polymers and metals, and is not rejectable like a biological organ would be. But it, at this point, is just being used as a bridge to transplant, and some new models will be on the market soon that are actually destination therapies, so they would not be a bridge to transplant. And one of the fields also is engineered body parts using stem cells and scaffolding, and a number of different body parts have now been grown in the lab. The image on the right is a lab-grown trachea. Obviously, the one on the left is an ear, but there have also been artificial organs that were grown using scaffolding and stem cells that have been transplanted into both animals and people, and that work just fine. They create urine, they do everything they're supposed to do. Another example of converging technology is a retinal implant. This is where you implant a micro array of, I guess you could say, computer chips, for lack of a better word, into the optic nerve, into the retina, and then it's combined with a camera, and it actually provides some amount of eyesight, although it's not perfect. Brain implants are coming along, especially in the domain of the U.S. military. The military is developing a brain implant that will help soldiers who have traumatic brain injury and that assist memory. And scientists are looking at very different parts of the brain right now to look at microstimulation. I don't know why it's doing this. One of the technologies that's been around for a long time is deep brain stimulation, and scientists have been using that for the relief of things like Parkinson's disease and epileptic seizures. There's a new push now to come up with brain implants that would assist in the retrieval of memories. It's being tested in some Alzheimer's patients. In a very strategically located part of the brain. It's hoped that the brain implants will help these patients to access the memories that they have, not necessarily create new memories, but to be able to use what they still have left. And then using gene editing with stem cells obviously would have some exciting possibilities as well. So one real-world example that's kind of close to human clinical trials now is the kidney project at the University of California in San Francisco. And this is for a total artificial kidney. That's a picture of it there. It's about the size of a coffee cup. The artificial kidney contains hepatocytes taken from the actual patient. So it is a mixture of artificial and biological material. It uses a nanotechnology coating and a hemofilter, which is a very, very fine-grained filter, which will will make sure that you keep proteins in the blood and can filter out things like salt and sugar. And it's powered by the patient's own blood pressure. It would be used as a destination therapy. There are other artificial organs out there now, kidneys, that are kind of effective, but you have to wear a belt and you have to be tethered to a machine to power it. So this would be a tremendous step forward. So we're looking at a future of technology-enhanced humans. And so I thought it might be interesting just to, on a closing note, to think about what it would be like to actually have many, many older people in society. So when I wrote about this, I decided to look at some of the psychological research in terms of what it's like to be an older person, and is that really something that is it desirable that we would live many, many years, and some of those years being quite old, although very healthy and fit. So what I found is that the research shows that older people are happier and have more stable moods than younger people. In the workplace, they are more productive and make fewer mistakes, which kind of flies in the face of age discrimination. But some studies were done in Germany in production jobs, and it was found that older people consistently, because they were so stable, was part of the reason, but they were more productive and they made fewer mistakes. Also, older people have vastly more information in their heads. And a lot of people who talk about and write about aging say, well, I don't know that I would really want to live very long, because isn't it inevitable that you start to lose your memory? Well, there was a study that was done in Germany by a team of linguists that tested people as far as word recall. And they used a huge database of words and expanded the search beyond what normally these type of studies go on. You have a small subset of words, and you test older people and younger people, and you find that older people take longer to access the same words as the younger people. Well, what they found was not that it wasn't that the older people were losing vocabulary, it was because they had such large vocabularies. So that when you compensated for the amount of time it took to go through the massive amount of information in their head, the speed of recall wasn't really longer in older people. It was just that they had so much more information to sift through. So that was very tantalizing, a very interesting piece of research. Also, psychologically, Older people are less geared to reward seeking and are less acquisitive. So maybe that could change society. I don't know if we had a lot of older people. Also, older people commit fewer crimes and they're unenthusiastic about war. So almost all crimes are committed by young people. You know, it is possible that society could change very dramatically if we had many, many more older, much older people who've lived much longer. We have to keep in mind, though, that if we have radical life extension, we won't have old, decrepit people shuffling around being disabled. These older people will look and feel like young people. Then there's the longevity dividend that society accrues. It's also kind of a widespread misconception that the more older people you have in an economy, the more of a burden that is on the economy, and the more difficult it is for everybody. But that's not actually true, because economists have run this through the numbers many times over, and there actually is a longevity dividend in which older people stay vital and work longer, and because they're very efficient and because they have all these other qualities, they actually generate wealth for the society that they live in. But another thing that we need to take into consideration is the obesity epidemic, and that could really complicate this whole picture in terms of aging. So we haven't seen the old age yet of a lot of the young and middle-aged people today who are severely overweight or obese. And the kind of health consequences those will have. So that could potentially throw a wrench into the works. Another issue is population control. If we do have radical life extension, and if a lot of people choose it, then obviously reproduction will need to go down, and that's an open issue that hasn't been resolved and will need to be resolved. And also the cultural changes from radical life extension could be much greater than anticipated. We just don't know, because we've never lived in a society. We've never lived in a society like the one we live in today, where we have a lot of people who are in their 70s and 80s and 90s. It's possible that people alive today in middle age could be able to take advantage of some of these technologies. So, for example, a lot of people die from heart disease. Heart disease is the #1 killer for men and women. If your heart gave out, you might be terminally ill. But a lot of people who die of heart disease are otherwise fairly healthy. So we don't know how long those people would live if you were to replace their heart. So we just don't know, but I think the cultural changes from life extension could be very, very great. And that's all I have today. I'd be glad to take some questions if anybody.
What is your take on human growth hormone injections and the result?
My take on what?
Human growth hormone injections.
Okay. Well, you know, that's something that, first of all, is not, you know, known to be safe. There are people who work on the assumption that by replacing the hormones of a younger body, that your body will then, you know, regenerate and be, you know, will be more like a young body. But in actuality, the studies about hormones in general having an anti-aging effect have not borne out. And at the same time, it is thought that there may be a connection between human growth hormone and certain cancers. So it's strictly experimental at this point. There are people who do it, but it isn't really backed up by science at this time.
So is there any evidence that stem cell supplementation or stem cell injection actually extends the lifespan or healthier way?
Well, we don't know, and I think that's why it's so important to study pluripotent stem cells in the lab, because I think what we need to do is separate out what properties of those cells actually do lead to rejuvenation and which ones have the potential to overdivide and turn to cancer. So, you know, there are a number of cosmeceuticals out there now, cosmetic pharmaceuticals, so to speak, that claim to use stem cells to rejuvenate the skin. I don't know how, you know, effective that is because that's not really regulated under the FDA. But I think at this point it's premature to say that we could actually inject stem cells, you know, pluripotent stem cells in particular, into a human being because we know that there's a possibility of cancer. Anybody else? Okay, well, thank you very much.
Over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine. I really do. Where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine. I really do. Where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine. I really do. Where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy.
Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine. I really do. Where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine.
Welcome everyone to one of the final sessions of the 12th World Stem Cell Summit Public Day. And so far the reviews are in and they've been A+. At least no one wants to tell me if they didn't enjoy it, so I'm very happy as the founder and co-chair of the World Stem Cell Summit. My name is Bernard Siegel. I'm also executive director of the Regenerative Medicine Foundation. So I had the pleasure of meeting Jeff Karp for the first time at a conference in Paris. How about that? Right. I remember. It's good. So, you know, what a great place to meet anyone. But Jeff really impressed me with the depth and breadth of his knowledge as it relates not only to regenerative medicine, but tissue engineering. And indeed, he is an inspiring figure. You may know him, of course. He's with the Harvard Stem Cell Institute. He's an affiliate professor at MIT. I've read about him. He is a bio-inspired medical problem solver. He is an expert on tissue adhesives, biomedical engineering, and mesenchymal stem cells, among his many talents. But he's speaking to us today about bio-inspiration and innovation. And I had the occasion to see a talk online, and as soon as I saw that, I knew that this is something that had to be live streamed, it had to be preserved to the audience of the World Stem Cell Summit around the world. So I'm very happy to present my friend Dr. Jeff Karp.
Thank you. Thank you. Thank you for the nice introduction. So today what I thought I would do is rather than just kind of show data, I really want to talk about the process. And I would say that, you know, in basic science, when we're doing discovery work, often, you know, 9 times out of 10, we conduct an experiment and it fails. And I think the challenge is, you know, how do we take the basic science and advance it towards the clinic? And I think, you know, anyone who's ever tried to translate anything, I think, would agree that the number of challenges, the number of problems that one encounters actually grows exponentially. And so we've really tried to make a strong commitment in my laboratory to not just publish papers, but to try to advance that basic science into therapies that can help patients in the shortest period of time. And I would argue that in many ways, we're actually biased against solving tough problems. And I don't know how many people here have ever seen, there's this TED Talk by Ken Robinson. It's actually the most watched TED Talk of all time. Have people seen that? There's a few people raising their hands. If you haven't seen it, go watch that talk. It's very entertaining. But there's a very powerful message there, which is what Ken does is he describes that when children are born, every child is born creative. But yet the education system over time actually basically takes that creativity out of kids. And I think that this really negatively impacts our ability to solve problems. And I would argue that what has happened is when we go to solve a problem, we approach it, we encounter a failure, we step back, and then we think we're being creative, and we approach the problem again, but we're approaching it from the exact same angle. And we expect different outcomes, and that never happens. So to really emphasize this point, what I wanted to do is share with you this example, which is a problem. This really happened. This is in Europe. And you see here that we have a car that's fallen into the water. And we have the problem solvers that are lined up trying to figure out, you know, what they're going to do. And they probably have, you know, 2 or 3 different options. And so what they did is they bring in this crane. And things seem to be going well until the following happens. So one problem has become 2 problems. And the second problem is even bigger than the first problem. Something I'm sure we can all relate to perhaps on a daily basis. So the question is, what are the problem solvers going to do next? What do you think? Go home. That's right. They're going to do what most of us, except this gentleman here, would do, which is go and get a bigger crane. And what do you think happens? The same thing. So to me, the challenge is, how do we break free from this repetitive thought process? And I would argue that our brains like to operate in these super low energy states where they anticipate what comes next, right? One example, if this has never happened to you, is this happened to me many times, is that you have your cell phone, it tells you to update your password. So you put in the new password, and then the next time you go to use your phone, you enter the old password, right? Because your brains like to operate at these really low energy states and anticipate what comes next. If that hasn't happened to you, there is something you can try, which is when you go home, change the location of the soap or shampoo in your shower, and I bet when you come back the next day, you actually reach for the old location. Again, our brains like to operate in these low energy states. And so the challenge is, how do we break free from this? Because I feel like that's really one of the keys to unlocking new ideas. And in my laboratory, we have been trying to harness a number of tools to break free from this repetitive thought process. And one of those tools is bioinspiration. And it's this idea that every living thing, every plant, every animal, anything that is alive around us today is here because it has solved an incredible number of challenges, right? Otherwise, it just becomes extinct. And so in essence, we're actually surrounded by solutions which I see as ideas for solving problems. Tens and hundreds of millions of years of research and development happening all around us. And we're only limited by our ability to go out into nature and make observations, and by the tools that we have available to uncover the mechanisms that nature has evolved to solve these problems. And what I find really remarkable is that tools constantly get better and better. We're in research, we have new tools coming online all the time and higher resolution. And so we're constantly going to be uncovering new solutions in nature, new mechanisms. And so it's almost like this endless encyclopedia of ideas where new chapters are constantly being written. And so what I want to do today is share with you an example in the lab where we were working on a translational project. We literally hit a wall, we didn't know what to do, and we said, well, maybe the solution is already out there in nature. Maybe we can find ideas of creatures that have evolved to solve similar problems, and we can bring those ideas back into the lab and apply it to our problems. There we go. So meet Dr. Pedro Del Nido. He's the chief of cardiac surgery, Boston Children's Hospital. And he contacted me one late summer evening, and he said, Jeff, I'm trying to treat kids who have septal defects. These are holes in between the chambers of the heart. He said, sometimes I go to suture those holes, and the tissue is so fragile it just tears. And he says, that's not acceptable. He said, there's devices that work in adults, and they work fairly well. They're not perfect, but they work fairly well. But they're permanent. They're non-degradable. And you can't simply downsize those devices and put them into a child's heart, which is going to be growing over time, because it's unacceptable to have to come back every couple years and do these revision procedures. He said, is there any way that we could develop a patch that you could put up against the hole right here? It would immediately seal the hole, and the patch would then enable cells to migrate on top of it and form new tissue. So now you have the patient's own tissue on top of this patch. The patch would dissolve away, degrade, and the patient would be left with their own tissue sealing the hole, which would then naturally grow with the patient. So we were really excited to work with him, but we knew that this was going to be one of the most challenging environments that we would be developing a tissue adhesive, probably inside the entire body. because you have on average 60 beats per second, so that's a lot of expansion contraction cycles that we have to consider. There's a lot of shear force that's happening from the blood flow. It's extremely wet. There's enzymes. There's lots of cells that are present. So this is going to be a really tough challenge, tough problem to solve. So what we did is, in majority of translational projects that we start, we like to put together design criteria for a solution. So this is a list of properties of what we would see an ideal solution would have. And to me, this is, first of all, highly iterative. In many ways, you never fully nail it because you're constantly learning new things. But it's hard to do because it's easy to come up with a list of 20, 30 things, but then how do you narrow it down to 5 or 6, and then narrow it down to 1 or 2 that's going to drive a completely different solution than anyone has ever tried before? And so we came up with this short list here. We said, we're going to be placing this inside a heart. We're not going to stop the heart to get this patch to attach. So it would have to work in the presence of blood. It couldn't be fouled. A lot of materials that have been developed that are tissue adhesives, they actually react when they contact water. Or if they contact proteins, they get fouled. So the adhesive won't work when you try to push it up against the tissue. So we have to figure out a way around that. The material is going to have to fully degrade over time. It has to be elastic so it can expand and contract with the heart. Otherwise, it will delaminate and come off. The material has to be biocompatible, so cells have to be able to grow and form new tissue. And then talking with Dr. Del Nido and a number of other clinicians, they said, there's some materials that cure within 1 minute or 10 minutes in the clinic, but we don't like those because we don't want to be at the mercy of the technology. We want to be in control. If we're ready to seal this at 7 minutes and 32 seconds, we want to be able to do that. So we put on-demand adhesion as one of the criteria. And this is a very practical consideration, something that I think we often forget in academia, are really the practical considerations of how is a surgeon going to be handling the materials that we're developing. And then also, because we're going to be curing this, and we thought using light activation would be useful, we've developed a lot of light activatable materials, we need some sort of a precursor form. So we have a patch and maybe a glue that is in a precursor form. We push it onto the heart, and we need the clinician to be able to move it around until they get it into the right position. And it can't wash out during that repositioning stage. So we said resist washout's another criteria. So we've been developing all kinds of materials in my laboratory, and some could fulfill some of these criteria, except for 2 of them. One is resistant to blood and resist washout. And so we really didn't know a way around this, and I think the field was really at a hold. We really haven't seen any major innovation in the clinic outside of sutures and staples, which have been around for decades and have tons of limitations. And so what we did is we stepped back and we said, how would nature solve this problem? Can we find examples of creatures out in nature that exist within wet, dynamic environments, similar to the environment inside the beating heart? And we found a number of creatures. We found sandcastle worms that exist in the sea, and you have the constant surf, and they remain attached to rocks. And then we have slugs and snails, which often you'll see in the rain are stuck on a leaf, and they're not falling off. They're able to stick under those wet dynamic environments. And so we took a look at these, and we said, well, what do these creatures have in common? And we realized there were actually a couple key things. One is that they had viscous secretion. So anytime you see a slug or a snail, there's always this kind of slimy stuff coming out from the back. Well, that actually is part of an adhesive mechanism. Anything that's viscous has adhesive properties. So like honey on a plate, for example, doesn't easily wash away. And then when we took a look at these viscous secretions, we also noticed that they contain hydrophobic agents, and things that are hydrophobic can repel water. And so that gave us an idea, and we started thinking, well, what if we developed a tissue glue that was entirely hydrophobic? We would apply this to the patch, put it inside the beating heart, push it up against the heart, and it would repel the blood away from the surface of the heart. This would allow us to perform this procedure without stopping the heart. And then we said, well, if not only is it hydrophobic, if it's viscous, it's going to stay there long enough for the clinician to position it, and it would resist washout until the final cure. And so we spent probably 2.5 years developing this. But one of the things that we realized along the way is that just looking at slugs and snails was not going to be enough, and sandcastle worms. We needed to make this material super adhesive. And for that, we actually turned to ivy. And I don't know about Florida, but in Boston, there's a lot of buildings that look like this. I don't know about you, but I've actually gone up to those buildings and tried to pull the ivy away, and it's remarkable the amount of force that's required. It's really, really significant. So next time you're in New England or somewhere where there's ivy, give it a try, and I think you'll be impressed. So lucky for us, the mechanism through which ivy attaches to buildings was recently elucidated. And it turns out they have these root hairs, and these are like heat-seeking missiles. They go up and down the building, and they look for crevices. And when they find a crevice, they insert the root hair into it, they secrete a glue, and then they shrivel up and they mechanically interlock. It's remarkable. So that gave us an idea. We said, well, what if instead of just using reactive chemistry, which is what a lot of others were doing, and we had tried that previously with some of our tissue adhesive projects, what if we could develop a glue that would infiltrate into the tissue, just like ivy goes into a building, and then when we cure it, it will actually lock into place, kind of like a tissue like Velcro. And so with that in mind, we advanced. And literally took us 2 or 3 years. Many challenges that we faced. We had a highly multidisciplinary team, probably about 15 people on the team, biomaterials experts. We had a number of surgeons. We even had someone to help us deliver light inside a beating heart, a fiber optics expert. And so what I want to do is kind of fast forward to the most challenging experiments that we performed, and this is one of them. So before showing we could put a patch inside a beating heart, we wanted to start with the outside of the heart, the myocardium. And what I'm about to show you now is from an animal experiment. So if anybody is, gets queasy at the sight of blood or animal experiments, you may want to look away. So here, what we're doing is we're making a hole inside the myocardium here, the outside of the heart. This is a large hole, and this is a rat. And we have this suture here, which is just going to be a temporary suture. We're going to remove it at the end, and we're going to attempt to seal this hole with a patch and our glue. No staples, no sutures. And you'll see that this is a very high pressure environment. We're not going to wipe away the blood. There's the patch, which is also biodegradable and it's transparent. There's glue on the underlying layer. We push this up against the heart. We shine the light for 5 seconds. And while the patch attached, this is one of the early animals we treated, we had something catastrophic happen, which is the patch kind of slipped away from us because we were nervous and there was a leak. Completely unprepared for what to do. We just scrounged up as much glue as we could find and we applied it directly to the leak.
Yep.
And you'll see that if you look carefully, it's less red where we've applied the glue. It's actually repelling the blood away because it's hydrophobic. And because it's viscous, it stays in place long enough for another pulse of light, 5 seconds. Remove the suture and we have a perfect seal.
Okay.
And we took these animals out 6 months and they did fine. And so now what we wanted to do is move to an even more challenging experiment, which was go from a small animal, a rodent, to a large animal, a pig, and see if we could get this to attach inside a beating heart directly to the septum where these kids tend to have the holes in between the chambers of the heart. And so for this, Dr. Del Nido had developed a device for delivering all kinds of different devices into the heart. And what we did is we attached the patch to the end of the device, made a small incision in the myocardium. This is a proof of concept experiment. We applied this to the septum. We shined the light for 20 seconds. We sutured the myocardium. And this is what it looked like from the echocardiogram immediately after the procedure. So it's 2 separate pigs, and you can see the patch is attached. The heart rate here is 82 beats per minute. We came back 4 hours later and added epinephrine to increase the heart rate to cover the full physiological range. And you see here that heart rate's 165 and the patches remain attached. If it wasn't attached, you wouldn't be able to see it be moving around so much. So then we came back after 24 hours and the patch was still there. You can see a suture here, which was part of the deployment mechanism. And you can see here that we have after the 24-hour time point, we have a thin capsule that's starting to form here. So for us, this was really a major step forward because it demonstrated that we could get this patch to attach inside a beating heart during a procedure in a large animal that mimics many aspects of the human condition. And so we were pretty excited about this. And you'll notice, though, that we had to make a hole to seal a hole. So that's not, this is really just a proof of concept experiment. And we constantly, we have our eye on the clinical application, not just publishing papers. And so we stepped back and we said, you know what, we really need to solve this problem. And so we teamed up with Connor Walsh at the Wyss Institute in Boston and continued to collaborate with Dr. Del Nido to develop a way to deliver the patch through a blood vessel into the heart so that we wouldn't have to make this second hole. And so I'm going to show you an example of how this device works.
Researchers from 4 top institutions in Boston have designed a specialized catheter that can repair holes in the heart without invasive open heart surgery. First, a clinician inserts the catheter, which is equipped with a UV light and biodegradable adhesive patch, through a vein and guides it to the hole in the patient's heart. Next, the adhesive patch and 2 balloons, one on each side of the hole, are released from the catheter. The balloons apply pressure from both sides of the heart wall, securing the patch in place. Then, the clinician turns on the catheter's UV light, which reflects off the balloon's interior and activates the glue on the side of the patch facing the heart wall. Finally, the clinician deflates the balloons and removes the catheter, leaving behind the biodegradable patch. In time, tissue will grow over the patch, and the patch will dissolve, leaving no foreign material in the body.
So this was really a way to solve the problem of creating a hole to seal a hole. And this is now a developed and functioning device. We recently published a paper on this demonstrating that it could work in a pig model. And so we're thinking of translation. And so as we're considering this application of sealing septal defects, we recognize that the bar is actually very high for treating pediatric patients. And so we continue to advance this in the laboratory. But because this material is so promising, we showed that it could seal the carotid artery of a pig, the aorta, could stick to intestine tissue. We formed a company called Gecko Biomedical in 2013. And the company has actually taken the technology from my lab and created a stable, a scaled product. And here's what it looks like. So Bloomberg came by the lab not too long ago and created this video. And you can see we're applying the glue to a vascular graft that it attaches very well to it. You see the precursor is a viscous kind of honey-like material. And this is Maria, who was a PhD student in my laboratory, who co-developed this material with Nora Lang, who was a cardiac fellow in Dr. Del Nido's laboratory. So the two of them together were really the leaders on this project. And now Maria is head of research at Gecko Biomedical, which is based in Paris. And the company now has been able to advance this technology that we developed in the lab and is currently testing this glue for vascular reconstruction in human patients in Europe. So I was talking a little bit about this, this glue when I was at a conference in Boston that was at the New England Aquarium. And I'll never forget that conference because it was right where anyone who's been there knows that there's a penguin exhibit. And it was literally like right in front of the penguins, which were kind of behind. And they were so noisy, I could barely even yell loud enough for people to hear what I was saying. But this guy was in the audience, Dr. Rosenblad, and he heard a few things that I was talking about. He's head of dentistry at Angel Memorial, which is really a top-tier vet clinic in the New England area, and he said, I think your glue could be very useful for treating not just humans, but actually animals, and I'd love to talk more with you about this. So about 4 or 5 months ago, he contacted me again, and he said, I have a dog, a patient that's a bulldog. It's 10 years old, and it has an oral nasal fistula, a hole from the oral cavity all the way to the nasal cavity. He said, it's so big I could put my finger right through. And he said, the dog's in miserable shape. It's like on its last legs. It's getting infected. He said, is there any way we could apply your tissue glue to fill the hole and help this dog? And so the concept was that we would need to create multiple layers of this glue. We were going to cure the glue right into the hole after debriding it. And then we were going to pull a tissue flap. We'd cure the glue and then pull this tissue flap over top and see if we could get that to work. And he had performed 3 surgeries on this dog, and all had failed within 2 or 3 days. He had just tried to place a flap over the hole, but because there's so much tension, especially in the mouth of a bulldog, it was just tearing open after each procedure. So you can imagine the owner and the dog and the vet have been, by this point, very, emotionally distressed about, just really trying hard to do whatever they could to save this dog. And so we had never tried this glue could work on in the oral cavity. And so Yuhan Lee in my lab, who's leading this project, next day we I talked to Dr. Rosenblatt, and the next day he actually brought this cow head in the lab that he got from a local butcher. And here you can see that he's actually applied the glue to the oral cavity, and you can see it's actually sticking really well. In fact, he's taken like a metal spatula and is trying to remove it. So we knew that this glue now could attach to oral mucosa, and so we went ahead with the procedure at Angel Memorial. And so here's what the fistula looked like. It was actually created from when Dr. Rosenblatt pulled the tooth and it didn't heal. And you can see it's infected here, lots of hair and all kinds of junk that was in that space. So they cleaned that out. We cured the glue in this spot, and then Dr. Rosenblatt created a tissue flap and pulled it over top of the hole and then sutured it. And about 2 weeks after the procedure, the first 3 procedures that were performed within 2 to 3 days had failed. But after 2 weeks, the dog came back, and you can see that the flap is still there. And literally, Dr. Rosenblatt and the owner were almost like crying. They were so happy because they had been through such difficult time. We were caught a little bit off guard because we just weren't in the same mindset as they were. And the dog, actually, this is what the dog looked like before. So you can see, not very happy. And then this is the dog afterwards. You can see a remarkable difference. And this dog, actually, the name of the dog is Poppy, named after Big Poppy. So anybody who follows baseball knows of Big Poppy, who's the star, just retired this year from the Boston Red Sox. And so, I was calling the dog Little Poppy. And I think, there's actually some resemblance to Big Poppy. And so here's the team here, and it's been now over 3 months, and Poppy's doing really well. So here's the team, Dr. Rosenblatt. There's Yuhan, who's leading the project, and some interns in the lab. Here's a technician from Angel Memorial, and here's the owner of the dog, Mr. Shanahan. And this actually, the press got word of this story, and they came to interview Little Poppy. And so you can see he's kind of having his glory day here at Angel Memorial. So to me, this was actually a really exciting moment in my career, and also the people in the lab who are part of this project, because we had always thought, that the impact that we would secure at some point in the distant future would be on humans, and we never realized that there was this amazing opportunity in our backyard, literally, to help animals as well. And so now this has become one of the missions of the lab, and I've actually gone over to Angel Memorial and talked with a number of vets over there. I gave a lecture in their lecture series, and I'm trying to figure out ways that we can move some of the other technologies in the lab to help animals in a shorter period of time. So I just want to share with you one other tool that we've used. I talked a little bit about bioinspiration, but we also use another tool in the lab that I like to refer to as radical simplicity. And again, it's really this notion, the paradigm I've tried to establish is to not just publish papers, but develop technologies that can help patients in the shortest period of time. And I can tell you so many stories that have happened where we just developed really complicated approaches that never made it out of the lab. And I think it's this notion that in academia, there's almost this pressure where the more complicated the technology you develop, the greater the chance you have of publishing in a good journal. But the challenge then is you have a lower chance of actually helping anybody, because it's the simple technologies that can get, that can advance the fastest to help patients. And so, so what we've been really doing in the lab is trying to find ways to keep the technologies as simple as possible at every single stage. So let me give you an example that is focused in the area of stem cells. So one of the big challenges in cell-based therapy, and in particular for stem cell-based therapy, is that we can grow stem cells, almost any type of stem cell now, in a petri dish, and we can control almost everything we want. We can pattern the media, we can pattern the substrate, we can get the cell to do anything we want. But the challenge is that when you go to transplant those cells in the body, you lose all of that control. They're entirely at the mercy of the biological milieu, and if the cells end up in different sites in the body, they're going to behave completely differently. And so we've been interested in developing technologies that can control cells following transplantation using very simple approaches. And I'm happy to say that Professor James Ankrum, whose picture here is right in the middle, is in the audience today, previous PhD student from my lab who's now faculty in Iowa. And so what we did is we looked at this and we said, there's all kinds of complicated things that we could do. And it was really the first thing that we thought of was, how could we solve this problem? And it was all these complex ideas came to us, but we really wanted to keep it simple. And it took us a while, but we eventually figured out a way to control cells following transplantation using perhaps one of the simplest technologies you possibly could imagine. So there's all kinds of degradable materials that already exist in the clinic right now, like degradable sutures and pins and plates, screws for orthopedics. And we actually end up using some of those materials. And the concept here was, how could we deliver devices that would be internalized into the cell and deliver molecules to the cell after it was transplanted. So that was really the concept that we wanted to advance. And so what we did is we discovered that particles that were around 1 micron in size were readily internalized by certain types of stem cells. We could put small molecules into those particles, and those particles would be internalized into the cell. And we found a certain size range where those particles would stay in cells for weeks and weeks. And what would happen was we could control the degradation of that material. These are FDA-regulated materials that we're using, already scaled to very, very large-scale manufacturing. And we could put in any molecule we want and deliver that. As the material was degrading, it would release the molecule to the cell, and we could activate pathways in that cell to control it after transplantation. And so here what you can see is in green, we have the particles, and the red is the cell membrane. So you can see these particles are inside the cell, and we were able to show we could keep them there for weeks and weeks. And not only were we interested in controlling the cells, we thought, well, if we could develop this type of technology, maybe we could control the environment around the cells as well, which is extremely important to consider. It's not just about the cells doing their function, it's the cells interacting with the environment around them, and we wanted to control that as well. And what we found is that by putting these molecules into the particles that were inside the cells, as these particles degrade, because they're made out of degradable suture material, the molecules were able to activate the cells, but also cross the cell membrane and interact with neighboring and distant cells. And so we were able to show that we could control all kinds of different properties of cells and neighboring cells, and we've published a number of papers on this technology. We've shown that we can boost the immune modulation of mesenchymal stem cells as one example. We can improve the imaging of the cells following transplantation. We can control the differentiation, and the list goes on and on. Again, this is a very simple technology. Microparticles, nanoparticles already exist in industry, in the clinic. The materials we're using are already FDA regulated, and the molecules we're using are also already FDA regulated. And so really it's just this combination of technologies that already exist has created a very powerful solution so that now we can control cells following transplantation, and we're exploring this for a wide range of therapeutic applications. In addition to this, we've also been interested in applying this concept of radical simplicity to drug delivery. And so, most people are familiar with taking a pill. And the challenge is that, this is a standard mode of therapy is taking a pill, but then you have to bypass the stomach. You have stomach acid there. You have to get it into the gut. It has to be absorbed. Sometimes it goes, through the liver and it gets degraded. And so there's all sorts of barriers that the drug has to go through. So you end up getting a very small amount that ends up in the circulation, and then it goes throughout your body. But often you only want that drug in a specific part of your body, localized. And so there's huge unmet need for all kinds of therapeutic applications where you'd want better control of drugs locally. And we wanted to know whether we could apply the concept of radical simplicity to drug delivery. And this would have implications for arthritis, ulcerative colitis, all kinds of different disease states. And so, again, we initially thought of these complex approaches. We stepped back, we stopped ourselves, and we went to the generally recognized as safe list by FDA. And this is a list of agents that exist in foods, they exist in cosmetics, and they've been deemed safe by the FDA if you apply them topically or if you use them in certain concentrations. And the nice thing about this list is these agents exist in ton quantities, GMP grade. And so what we did is we scanned this list and we identified small molecules that had hydrophobic and hydrophilic domain, so water-hating, water-loving, and an enzyme cleavable bond. And I'll explain why in a moment. We found these on that list. And what we were able to do is take those molecules and put it into water, and by coaxing the system just very simply with temperature and with some solvents, we were able to get that single molecule to stack over and over and over again and form a gel-like material that looks like this under electron microscopy. Now, what you're looking at here is a single molecule that's stacked over and over and over again. There's no excipients present here. This is not a polymer. These are non-covalent interactions. It's a single molecule that's just stacked and interacting with itself. And during the assembly process, we can incorporate all kinds of drugs. We figured out how to do hydrophobic, hydrophilic, and now even biologics like peptides and proteins. And then in the body, in response to inflammation, so patients who have arthritis, for example, there's many cases where you have a flare and then a period of remission. And so if you have a drug delivery system, you only want it to be releasing drug during the flare and not during remission, because then you're just depleting your stores. So we created this system so it would be enzyme responsive or inflammation responsive. So in the presence of inflammation, you have all these degradative enzymes, and so that look like this. And what happens is it breaks down these molecules, this enzyme cleavable bond, and releases the payload. And we've shown that this has potential to treat all kinds of different diseases, such as ulcerative colitis, arthritis. We've been applying this to limb transplantation. The list goes on and on, a number of types of cancer. So we've tested this in 6 different, now actually I think it's 7 or 8 animal models. A number are large animal models, including pigs. And really, the advantage of this technology is that it's very simple. We're not synthesizing materials. We're just buying them from the generally recognized as safe list. And we recently started this company called Alivio Therapeutics, which I co-founded with Bob Langer at MIT, as well as PureTech Health, which is a VC firm in Boston. And we've set up our lab, and we're moving full speed ahead to the clinic. So if all goes well, this could enter human testing within the next 2 or 3 years. And then finally, I just want to end with one last example of bioinspiration. I want to come back from radical simplicity to bioinspiration, because we were really interested in asking the question, could we apply bioinspiration to tissue regeneration? And when you think of tissue regeneration, you really think of, it's really a holy grail. But if you step back and kind of look at the animal kingdom and nature, you realize that there are many, many examples of tissue regeneration occurring in adult creatures across the planet. So we know that sharks are able to regenerate their teeth throughout life. We know that lizards, if you chop off the tail, will actually regrow the tail, and the list goes on and on. And so the question, though, is that why don't humans have these abilities? So if we see these abilities all over the animal kingdom, why don't we have those? And the answer is, and I think, as we think about this a little bit more, perhaps when you look at nature, we have the building blocks out there. We really just have to decode the blueprint, right? And when we look at our bodies and we study biology, we realize that we actually do have many instances where tissue regeneration is already occurring, in everybody in this room, in fact. So the intestine, the lining of the intestine, is one of the most regenerative tissues that has ever been discovered. It completely regenerates every 4 or 5 days. And so we were really intrigued by that, and we wanted to see if we could identify some of the cues that were responsible for this regeneration. And so what we did is we turned to the intestinal stem cell. So there's a stem cell that exists at the base of crypts in the intestine, which are labeled green here, and the red that's the intestine. You see the villi and the crypts. And these stem cells actually are the powerhouse behind creating the epithelium, the lining of your intestine, the powerhouse of creating this new epithelium every 4 or 5 days. And so we stepped back and we really looked at the signaling that goes on within these cells and the neighboring environment, because what we wanted to do was control these cells. If we could control these cells, we might be able to activate tissue regeneration in other places in the body. And so what we did is we looked at the pathways, we found small molecules that could activate these stem cells, and we were able to take a system like this and produce nearly pure populations of intestinal stem cells simply with small molecules. And so we published this work a few years ago, and we've been looking at other systems to apply this to. And recently, about maybe 3 or 4 years ago, we started looking at the inner ear. And what's amazing is that these intestinal stem cells are marked by a ligand called LGR5. And that marker was discovered within the inner ear. And we thought, if we could apply the knowledge that we've gained from the intestine to the inner ear, maybe there's an opportunity to restore hearing. So each person, everybody here was born with 15,000 hair cells per inner ear. And you essentially die with the same cells. They never regenerate after you're born. And due to noise damage and various types of drugs, those hair cells are being killed all the time. So as you go on in life, the number of hair cells you have decreases, and so does your hearing. And so what we did is we were interested to say, could we now take this understanding of the basic cues in the intestine, this highly regenerative organ, and apply it to the inner ear? And what we found is that we could take our molecules, apply it to the inner ear. We were able to proliferate the stem cells. There's a supporting cell, a progenitor type cell within the inner ear that's the progenitor of hair cells. We were able to find molecules that could amplify those progenitor cells and produce tons and tons of new hair cells. And that's what you're looking at here. You can see these pristine hair bundles, which are responsible for interacting with sound and then converting it through connections with neurons into signals your brain can interpret as sound. And so we've now found that we can promote the regeneration of hearing in animals, and we've formed a company based on this technology called Frequency Therapeutics. This is really a platform. The inner ear, so to restore hearing, is the first application. And we're in the process of moving this very rapidly towards the clinic. But this is a platform, and we can apply this to many other sites in the body. And this company, Frequency Therapeutics, is just in the process now of coming out of stealth mode. We formed it a couple of years ago and have been very busy moving this technology and building the team to advance this to the clinic. So in summary, today I talked about some of the tools that we've been harnessing in the laboratory, namely bioinspiration and radical simplicity. For bioinspiration, this is not biomimicry. We're not just trying to mimic creatures that we see as solutions, every single detail. We're actually just taking a basic idea from nature and then improving on it for our own purposes. And then radical simplicity is really stopping ourselves from trying to do the traditional academic process and develop these complicated technologies, and really step back and say, how can we keep this as simple as possible at every single step? And really think ahead the full translational spectrum in terms of scalability and manufacturing of the technology. So at this time, I want to acknowledge the wonderful people in my laboratory who have created the data that I've shown you today, and all really wonderful people, all my collaborators, of which there are many. And I'd like to thank you for your attention and my various funding sources, and I'm happy to answer any questions you may have. All right, thank you. Are there questions? I think it's on now. I can hear you, yep.
My question is, with regard to the hydrophobic, hydrophilic, or amphiphilic grasp substance that you mentioned, how does it interact with cells, or maybe how do cells respond to being in that matrix that you showed? And what is the substance as well?
Yeah, so the question is, how do cells interact with these gel-like materials that we've been developing from generally recognized as safe agents? And what exactly are those materials? So I'll answer the second question first, and then I'll come back to the first one. So we've found molecules like ascorbyl palmitate, which is long-acting vitamin C. It has a hydrophobic and a hydrophilic domain and an enzyme cleavable bond. And so, you can go to the drugstore right now and buy that. And all we've done is change the format. So instead of that molecule existing in like a free format once it gets into solution, we found a way to create a gel out of it. And there's many examples like that. Another example is we've taken an emulsifier that's in ice cream, and we've been able to create a gel from that. So there's many different molecules that we've shown this works for. We've also been looking at, so these are on the generally recognized as safe list, which typically means that they're safe in certain concentrations when you put them on the skin or if you eat them, but not necessarily if you inject them into the body. And so we've done a number of studies where we've looked at the biocompatibility of these materials, and we found for the most part that we have very good biocompatibility. So we've done head-to-head comparison with degradable sutures, so which is polylactide coglycolide. And in most cases, we have much less inflammation than the existing degradable materials that exist in the clinic. Yeah, so we haven't really advanced many projects yet where we've mixed cells with these gels. We've typically been looking at this more as a drug delivery platform, an inflammation responsive system. So for treatment of ulcerative colitis, as well as arthritis and limb transplantation. But this is something that we're actively looking at, is whether we can. So for example, we've taken tacrolimus, which is an immunomodulating agent for limb transplantation, and we've put it into the gel and shown that we can, with a single injection in a transplanted limb, we can keep it alive from 100 to 150 days. We think that this system could be very interesting for transplanting allogeneic cells. So for example, beta cells, which may get quickly rejected. So if we put them into a gel that has an immunomodulatory agent in it and can be released for long periods of time, that could be an interesting platform, and that's something that we're looking at now.
Yes, I was just wondering about the ulcerative colitis. You were talking about how the cells regenerate in the intestines, but then there's a chronic inflammation.
Yeah.
With it. So if you do actually add stem cells in there, what will they do in ulcerative colitis, and when will you have a treatment for that?
Yeah, so ulcerative colitis is really a tricky disease because, well, there's a lot known, there still is a lot unknown in terms of the systemic effect of a systemic inflammatory effect versus local effect. There has been studies where people have taken these LGR5 stem cells that exist within organoid structures, which is just like a ball of cells, and a small fraction are the stem cells, and they've done infusion, enema infusion in animal models of ulcerative colitis. They're able to attach and engraft and accelerate the healing process, but the animal models of ulcerative colitis are not very good predictors of what's going to work in humans. It really only mimics, one aspect of the disease and not the background inflammatory response. And so I think right now, at least my thinking is that for stem cell therapy to work in the context of inflammatory bowel disease, you probably will need to have an immunomodulatory agent as well as the stem cell, to complement the stem cell therapy. What we've focused on with our drug delivery system is almost every ulcerative colitis patient will require an enema at some point, enema treatment at some point in their therapy. And that's problematic for a number of reasons. One, they have to hold it in, which is really challenging to do. Second is that you have a lot of systemic absorption of drug, so there can be toxicity associated with that. And third is the dosing is every day, so compliance can be very low. So we showed if we formulate these gels with a steroid, just as a first example, that we could get and break the gel into particles in a suspension, an enema suspension. We showed in several animal models that this could attach rapidly within seconds to minutes to the ulcers, so the patient wouldn't have to retain the enema. We showed that we could reduce the systemic exposure by 5 to 10 times, and we showed that we could dose every other day instead of every day with our system, because the gel attaches to the ulcers and stays there and continuously releases the drug right at the site of the ulcer. So we are advancing that. So we're looking at that actually from two separate angles. One is drug delivery, and one is from a stem cell-based therapy. Other questions? No? All right, thank you so much.
Over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age in medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age in medicine, I really do, where we don't have to treat cancer with chemotherapy. I mean, we would all like to hope that people could remain healthy while they're being treated for cancer, that would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine. For over 10 years, the Genetics Policy Institute has presented the World Stem Cell Summit. The event brings together over 1,000 attendees from over 45 countries around the world. The summit provides the premier forum for doctors, scientists, industry leaders, policymakers, and patient advocates to interact and discuss the quickly evolving and incredibly important science of stem cell research and regenerative medicine.
So it's really important for STEM biologists to be here at the World Stem Cell Summit, primarily because everybody who's anybody in the industry is here.
You can get to see not just the scientists, but the patients, advocates, and it's a very unique place. It's like the place to be, to connect and collaborate with worldwide.
We're really honored to be able to exhibit at a place like this and to be able to interact with the customers and the key opinion leaders that a conference like this brings in.
I think everybody hopes that the future is that we learn from stem cell biology so that the body can repair itself.
I think they represent a new age of medicine, I really do, where we don't have to treat cancer with chemotherapy. We would all like to hope that people could remain healthy while they're being treated for cancer. That would be absolutely fabulous.
We're happy to partner with the World Stem Cell Summit. It gives us a great opportunity to interact with this community, educate them on the availability of the space station as a viable research platform, and also educate them on the hardware and the process available to get them to station to conduct that research.
This is the only meeting that I know of that combines stem cells and patient advocacy and legal issues and ethical issues and regulatory issues, and you get to talk to philanthropists. So this sort of gets us out, gets us out of the lab.
The summit features the Stem Cell Action Awards, a gala celebration honoring the community's leaders, innovators, and those championing the furtherance of this important science. The World Stem Cell Summit is the place to be for dedicated stakeholders committed to the advancement of stem cell science and regenerative medicine.
Is Dr. Correa in the audience tonight? Dr. Correa? I heard he fell off the podium today. I was concerned. I hope I don't get sued. That was my first instinct. They're having to put the wine away before people will leave. Can I have a show of hands? How many of you have attended a World Stem Cell Summit before? Wow, thank you. Thank you for returning. Thank you very much. Are we ready back there? Ready? Okay. All right. Well, good evening, everyone. My name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation and founder and co-chair of the 12th World Stem Cell Summit. I'm so delighted to welcome so many friends back to a World Stem Cell Summit. We just had a show of hands, and a lot of people in this audience at this initial plenary session, this kickoff session, have been here before, so thank you for returning. It's a special privilege for me as a founder of this meeting to see us grow every single year. We should have record attendance this year, as we now have 8 tracks in our very complex program. As you know, this is a networking meeting. It's unlike any other meeting in the world, because what we do here is that we represent and provide a gathering place for our ecosystem of all those that are interested in stem cells and regenerative medicine. And back in the day when I first became an advocate, the first meeting that I went to by invitation was the founding meeting of the International Society for Stem Cell Research. And it was very special for me to be there, and I, as a patient advocate, a cancer survivor, I really believed in this important technology back in 2003, but there wasn't a single thing I understood inside the lecture halls. You know, I could speak to the 5 exhibitors that were there and to the scientists that had invited me, but I realized that in order for this field to move forward, we needed to get individuals involved, patients, because after all of us will suffer one day from something. We're all mortal, aren't we? We all have loved ones, friends that are afflicted with chronic disease or medical conditions or pain or irritations where there's no really cure. So I became, I guess, a fanatic about these amazing cells, the curative potential for regenerative medicine, the advanced technologies. And what we now have is a convergence of technologies. So a scientific revolution that in human history might have taken centuries or decades, now in our own lifetime, in this next decade, and it will impact our lives, our children, our grandchildren. It's something very, very important. So all of you who are gathered here are going to enjoy the networking aspect of the event. That's the key. Ask questions, be open with people, and break out of the silos that we are all in our chosen fields. Scientists who work with cells in a dish can actually meet a patient that suffers from ALS. An industrialist who wants to commercialize can connect with an investor, can connect with the patient, the end user, speak to someone from an insurance company to give reimbursement. FDA is in the house. We're going to hear from Celia Witten tomorrow, and we'll understand throughout this meeting the impact of the Cures Act, which may have passed the Senate today. I didn't get a chance to look it up tonight or tomorrow. And for the first time, I would submit to you that in the United States, regenerative medicine is taking its place in a way to find an accelerated pathway, something our organization worked very hard in Washington with the Bipartisan Policy Center to move forward. This is a critical time, and this is the original translational meeting. So I welcome all of you here. Before I introduce some of the representatives of the distinguished organizations that are co-organizers for this meeting, and we're going to introduce them in two sets, some of them tonight and then tomorrow morning as we kick off the first full day of the summit. I wanted to talk a minute about the public day that we all experienced. I don't know how many of you were able to spend much time in the public day, but I take pride in my team. Alan Fernandez, our associate director, and the rest of my team take great pride that we were able to do this, because we had about 500 to 600 attendees, 300 from the wonderful organization of Hadassah, which really supported the meeting tremendously, but also school children that prepared the wonderful artwork that you see outside, because science, technology, engineering, and math, STEM, you add the A, arts, to that, STEAM, and that's what we had out there. And these were third and fourth graders that all of a sudden have something they've learned about cell biology. How amazing is that? And they were inspired by the incredible art of Eluse, that were some of the images of the cells here, and it was sponsored by some local organizations, nonprofit, Prime Time, the Center for Creative Education, and the South Florida Science Center, and we were so grateful for their support. And we think this is going to get worldwide attention to show that this is something that we're all in, to create something important that people will be enthusiastic about, because with public support, as Lincoln said, with public support you could do anything, without it nothing can get done. So as we kick off this meeting, I want to introduce you to representatives of four of the institutions that are co-organizing. Tomorrow we'll be hearing from the University of Miami the Stem Cell Institute, Wake Forest Institute for Regenerative Medicine, the Center for Commercialization Research, the Center for Commercialization for Regenerative Medicine, CCRM, from Canada, Kyoto University and their Institute for Integrated Cell Material Sciences, and our friend Dr. Norio Nakasushi, who is here again, and the Cure Alliance. So we have some people we'll hear from tomorrow morning. But at this moment, first I want to introduce Dr. Thomas A. Ganwa. Dr. Ganwa is the Jorge and Leslie Bacardi Associate Director, Center for Regenerative Medicine in Florida, the Mayo Clinic, Professor of Medicine and College of Medicine. We've had a wonderful relationship over the years with the Mayo Clinic, pathfinding institution. Their Center for Regenerative Medicine is a colossal enterprise. We have many researchers and MDs and nurses and everyone from the Mayo Clinic. We're grateful for here to have them, and some opening remarks and slides from Dr. Ganwa.
I was asked to give some introductory remarks, and if you could launch my slides, you have them back there. Mayo Clinic has been a proud sponsor of this meeting for the last several years, because Mayo Clinic has decided through its actions that regenerative medicine is the future. Now, we'll see if this works. Mayo Clinic has embraced regenerative medicine as a strategy, as a strategic investment in the future of healthcare. I'm always amazed, and maybe it's just because I'm part of Mayo, that most people don't know that Mayo actually has 3 campuses spread across the country, which gives us the breadth to do many things in 3 different geographic areas. I represent the one here in Jacksonville, just north of here, a quick 4-hour and 15-minute drive. So it's always a pleasure to come down here. And we've embraced it, and we've embraced this model, which I've showed at this meeting before, that you have to have a blueprint and you have to have all the pieces. Many people who work in regenerative medicine may work in one or another of these various places, but we try to work in all of them. We try and have a portal where patients can get in. Then we have to get all the basic scientists involved. in systems biology and everything else. But we also need to have the clinicians involved so that eventually we can have the regenerative medicine programs. And these are, in order to do this, we also have to have all the platforms in place to be able to deliver the product eventually to the patient. Because at Mayo, it's all about what's best for the patient. And we've adopted this method of accelerating the research continuum, the so-called DTA, which is discovery, translation, and application. And we advance this throughout the entire clinic using the discovery toolkit. Obviously, the basic science is necessary. Clinical trials, which is the translation, and finally the application to actually get it to the patient and make it part of our everyday practice. One of my jobs is to be in charge of what we call the platforms. The platforms are what we're going to need to be able to deliver product to manufacture it. Human cell therapy, everybody is familiar with. This is where all the cellular therapies are developed. This is important, and we have human cell therapy GMP facilities on all 3 campuses, Phoenix, Rochester, and Florida, so that we can deliver the product at all 3 places. But we also have built up a biotrust, which is for iPSC cells and cell lines, located in Rochester with a branch in Florida, mainly concentrating on the neurosciences. We have a huge library of cells and iPSC lines derived from patients with Alzheimer's, various types of dementias, and Parkinson's disease, both for basic research, drug discovery, and other things. A biomaterials facility located in Rochester so that we can actually put together scaffolds and products that we can use with cellular therapy to treat patients. And finally, what's now our newest one called the Advanced Product Incubator, which is tasked with finding non-cellular regenerative products. And they are working on their first 2 products now. And if you look through this, you can see the scope. We spend a lot of money and have a lot of different products along the line. This is at the foundation level. One thing Bernie was interested in is what are we doing here in Florida, so I thought I'd spend just a second talking a little bit about Florida. These are all the regenerative medicine clinical trials, either preclinical or in clinical, that are currently going on within the Mayo system. The ones that have the red stars on them are ones that patients in Florida can actually participate in. So you can see that we have a broad range and we cover a broad spectrum of medicine. In Florida alone, this is internal funding that we are using over the last 5 years since we started, and in this last year we're up to over $3.5 million in funding just in Florida to help fund this effort. The Human Cell Therapy Lab in Florida is a 1,200 square foot, 400 square feet of GMP space, has 3 full-time allied health, various platforms, and mainly we're concentrating on bone marrow and adipose-derived MSCs, exosomes, stromal vascular fraction, developing various other new therapies, and we have specialized services, and currently developing a master cell bank for manufacturing. There's 4 INDs in development, and we're currently planning to expand another 5,000 square feet of our GMP capability in Florida. These are current clinical projects that will probably be coming online in the next year, other than the ones we already have. Trials in osteoarthritis, bronchiolitis obliterans, hemorrhagic stroke, pressure ulcers, PVD, heavily involved in the development of exosomes as a therapeutic tool. The first trials will probably be in the transplant arena. GI fistula, some very interesting new neurosurgery trials using regenerative techniques. And then we just have treatment of osteoarthritis and alopecia. The other thing we've concentrated on in Jacksonville is, because we're not as big as Rochester is, it's nice to have one place where you can deliver all these therapies. So we've actually developed a regenerative medicine therapy suite. It's in the transplant center because I ran transplants, so I got to control the space. And as you can see here, we have 2 treatment rooms, which are in the front there. And in the back, there is a processing room so that we can actually take specimens from patients, take them in the back, process them, and give them right back to the patient. We have 2 rooms working at the same time. We also can process things that come from the Human Cell Therapy Lab there. And this is up and running. We opened it up about a month ago. It's actually built. These are the blueprints, and This is just kind of what it looks like. The picture on the right is the actual treatment room, and it's the back you can see looking into the two rooms to where the therapy can be delivered. We believe that by concentrating everything in one area, we can get better expertise and better use. Finally, just a plug, shameless, that Mayo Clinic will be holding a symposium out in Arizona. For those of you of the northern climes, you might want to come visit our Phoenix campus in February. I know Bernie's going to be coming. But, I'll end there. Thank you.
How many of you ever wanted to fly in space? Our next speaker, yes, one. I bet more of you did. If you're as old as me, you remember the Mercury program. Listen, we have, our next speaker has not only flown in space once, but twice on the Endeavour, and that's Gregory H. Johnson, the President and Executive Director of the Center for Advancement of Science in Space. They're based right here in Melbourne, Florida, but their mandate is to the stars, right? And among the things that CASIS is involved with is the commercialization of LEO, low Earth orbit. They are the managers of the U.S. National Lab on the International Space Station. So I know a few of you are wondering, well, why are they speaking at the World Stem Cell Summit, and what does this have to do with regenerative medicine? Well, it just so happens that they are providing grants for amazing cell research, mind-bending cell research. As if you listen to the Public Day presentation, you would learn the importance of microgravity and cells. And in fact, around your neck is a lanyard with the company's Zero Gravity Solutions, which is the first commercialization that's coming out of the research on the U.S. National Lab on the International Space Station that has a case study later in the program. So we're so honored that CASIS decided to be a co-organizer of this year's meeting. They have some wonderful panels scheduled, including an expert lunch tomorrow at our Conversations with Expert lunch. So Greg, thank you, thank CASIS, and say a few words.
Thank you. I'm humbled to be up here on the stage with celebrated researchers, medical doctors, PhDs, Juris Doctorate. And here I am at the World Stem Cell Summit talking to you, but representing a group, a very special group that gets to do research on the only laboratory not actually physically located on the planet. It's orbiting the Earth about 250 miles up. And so I'd like to thank you all for all the work you do. It's important to me, my family, our country, the world, and we're trying to help using the International Space Station. So the Center for the Advancement of Science in Space has a mission to manage and provide access to this very interesting and unusual vehicle in outer space. It took the country, those of you who were at some of the different discussions in this public day, we had a talk with some local students and other lifetime learners of all ages who joined in our education mission where we talked about this wonderful asset. But the space station is obviously much more than an educational opportunity. It's a one of a kind innovation platform with facilities in space that are space versions of facilities that you all have used for years on the ground. Our part of that mission is to provide access and to think about research and technology demonstration differently than NASA. Because NASA's mission, one of its many missions, is to extend our presence outside, past low Earth orbit, to the moon or Mars or a near-Earth asteroid. But meanwhile, we also have other work to do in low Earth orbit in this one-of-a-kind facility where we can take gravity out of the equation, where researchers can look at things differently and understand and discover where gravity has shielded other phenomena that we can't experience here on the Earth, or just phenomena that are accelerated. So here at the conference, we have our cases team. We're a small team, but congressionally mandated to build the commercial market in low Earth orbit, to do different, to work on projects that have a different focus than what NASA has traditionally done in the past. So I could babble on and on about the space station, but I wanted to, bring up some points here, and I know that I'm kind of going over my 5 minutes. I don't have 10 slides, but I have one slide that kind of goes around and around, and it's pretty cool. But here's some thoughts for you. One, CASE's funding of stem cell research to date, we've had 5 projects, and it's really not about the funding, it's about the access. It's about the subsidized access to a unique laboratory in space. And so we've funded, we've sponsored, we've awarded, we've supported, we've facilitated 7 stem cell projects. And today, one of those projects was heart cells. And I'm, the actual beating of cardiomyocytes in space. And I see a researcher nodding in the audience. Dr. Wu from Stanford and his team saw for the first time this year beating heart cells in space. We've got a cases talk this week. I think it's on Thursday, and I encourage you to learn more about these 7 projects that we've supported on the space station. CASES is working with NIH and NCATS on a sponsored program, a $12 million program to have tissue chips using stem cells in space. And so that solicitation is out there. And again, the value is getting the, the access, the allocation to get your experiment onto the space station. But I think the proposals are due about the 15th of December. CASES is working with commercial companies to find breakthrough therapies that utilize the unique environment of the International Space Station. And we all recognize, and our involvement in this conference is because we believe in in the work that you're doing, and we want to help. So if something in microgravity interests you or your research, if you want to know more about how you can get to the International Space Station, part of our secret sauce is we think we can get you there quicker. And we want to understand what you're doing and build on your discoveries. Then get to know members of the CASES team, ask questions, and let us know how we can help. So anyway, thank you very much for being here. We're excited about the opportunity. The agenda looks very interesting and fruitful, and enjoy the week.
A long, long time ago, back in 2003, I had an idea that there was something more interesting than practicing law. It was to create a nonprofit organization to advance the field of stem cell research. It started off as the Genetics Policy Institute, later merged with another nonprofit that was founded by Dr. Tony Atala, called the Regenerative Medicine Foundation. So we've adopted that name. It better describes what we do. But as I was assembling a science advisory board for that organization back in 2003, the initial scientists that were engaged were Ian Wilmot, Dolly the sheep's father, and Dr. Rudolf Janisch, Lasker Award winner and prominent research at MIT. But as I was researching the field it was recommended that I reach out to someone in Miami, where I was at the time, by the name of Dr. Camillo Ricordi at the Diabetes Research Institute. And I remember how nervous I was because I worked with this longtime assistant, Mabel, and actually had an appointment with Dr. Ricordi, and I didn't know that much about diabetes, and I explained to him my passion and what I was going to do, and he became the third member of the Science Advisory Board of the then Genetics Policy Institute. Over the years, Camillo and I have worked together very closely on numerous projects. Our first meeting actually was at the United Nations, and Camillo spoke at that meeting in 2004. And the work that he does extends beyond, of course, the Diabetes Research Institute, where as scientific director, he's the Stacy and Joy Goodman Professor of Surgery, Distinguished Professor of Medicine, Professor of Biomedical Engineering, Microbiology and Immunology, Director of Diabetes Research Institute and Cellular Transplantation. He also found time to create an advocacy organization, the Cure Alliance. Well, Camillo and the Diabetes Research Institute Foundation for the second time are co-organizers of the meeting, and Camillo will be honored later tomorrow night with the Stem Cell Action Advocacy Award. So I want to invite my friend up, Dr. Camillo Ricordi.
Thank you, Bernie. That was my talk. Thank you very much for having me. Well, actually, it's a great pleasure to be here again, and I'm so pleased to see the evolution of space, laboratory, and science, because I started, believe it or not, in '84 in St. Louis at Washington University with Paul Lacey's group, and we had this big partnership with McDonnell Douglas Corporation that had the original technology to isolate islets from the human pancreas, and was the time where there were the first space shuttle experiment on microgravity of islet cell culture. And then I ruined the whole partnership because I developed the automated method for islet isolation from the human pancreas, and they split the partnership with McDonnell Douglas Corporation, and then we switched to what is the procedure that is still used today in clinical trials of islet transplantation. But has been a tremendous path towards the cure of diabetes, and I'm so privileged to be directing the Diabetes Research Institute and the Cell Transplant Center at the University of Miami, where we were able to start a new model, an affiliation agreement with the university, but that allow us to plan a strategic plan around patients and the development of a cure and remain focused on the strategic objective of the Institute. So I will not tell you about our programs because you can visit. There is a booth both for the Diabetes Research Institute with all the outline of our programs, as well as one for the Cure Alliance, that is an organization that I started a few years later. But what I would like to bring you is the concept that was quite disruptive 20 years ago when we started, but now it's becoming mainstream. That is the fact that if you keep patients and the development of cures at the center of your strategic mission, every decision step becomes clear. So you have to collaborate. There is no silos, there is no ivory towers, there is no big center institute. You have to open the way and break the barriers, the geographical barriers to scientific collaboration. We started in the diabetes area with a network of over 30 centers worldwide, now comprising the Diabetes Research Institute Federation. And we carry out trials not only to favor international collaboration, but also to try to overcome and resolve some of the barriers to clinical translation that time to time you can encounter in the United States or in other countries. So that one of these big challenges is the cost of doing clinical research in the US. So an islet transplant here costs over $200,000, in Canada $20,000, in Italy €30,000, in China $10,000. So that by working as a federation of centers with the same standards, the same IND, the same regulatory control and oversight, but we can capitalize on, if you have only 10 million a year to carry on clinical trials, you can do more if you work as a team worldwide, not just within your own institution. And then we expanded this model to the Cure Alliance, that is We just became a 501c3 this year, and now a little over 50,000 supporters worldwide, but is extending the concept of diabetes. We can use the same system of telescience platform technology, try to collaborate worldwide, open access for other disease condition to try to speed up and try to reach a cure in the fastest, most efficient, and safest way possible. So these are some of the themes that we will discuss also during this Congress. And for me, it's been a tremendous privilege to be working with Bernie and seeing the evolution of this conference from a small team to this major event now worldwide, with so many followers also internationally and through webcast. So it's really a pleasure and an honor to be here, and thank you very much for being here.
Well, there's a new university that has joined us as a co-organizer of the World Stem Cell Summit, Nova Southeastern University and their Cell Therapy Institute. You might not be as familiar with Nova Southeastern University, but if you live in South Florida, in the state of Florida, you understand it is an enormous private university with very successful graduate schools. And now it's really exciting to see the work that they're doing in cell therapies. Leading the institute is Dr. Richard Jove. Richard, I first met when we did a World Stem Cell Summit in Pasadena, California, and he was the scientific director and the head of the Beckman Research Institute at City of Hope. And the City of Hope, 2 years, joined us as a co-organizer of the summit. Rich is an amazing builder of institutes. He was in charge of science at the Moffitt Center, built the Beckman Research Institute in the City of Hope, and his journey has continued to Florida, where now he's at Nova Southeastern University. I like to think that his affiliation with us might have been a small, tiny lure that might have attracted him to Florida when the City of Hope was a co-organizer at our meeting here in 2012. So Rich brings with him a strong relationship with the Karolinska Institute in Sweden, and we have many fine researchers from the Karolinska that have an affiliation with Nova Southeastern here at the meeting and speaking at the meeting and attending. So these are folks you definitely want to meet. So let me introduce Richard Jove.
Thank you, Bernie, for your kind words. I do remember very clearly back in Pasadena when we were involved in co-sponsoring the World Stem Cell Summit at that time. Those were the early days of the World Stem Cell Summit. And I have to say, I think I've attended just about every single one since then, and I've seen the tremendous growth of the World Stem Cell Summit. And I have to say, I can't think of any other single person in this field who has done more to advance the cause of stem cell therapy and regenerative medicine than Bernie. So I'm very pleased to say a few words. Now, I didn't have enough wine, so I'm going to have to read this, okay? First of all, I want to add my thanks to Bernie and colleagues, everyone who worked very hard for organizing yet another magnificent World Stem Cell Summit. As you heard and you all know, this is truly a unique conference in bringing together patients, physicians, scientists, investors, and other major stakeholders, including astronauts, who share a common mission to accelerate the development of next-generation cell-based therapies. That's what we're all about here. As you heard, Nova Southeastern recently made a major commitment to biomedical research, and that is why I'm here as director of the Cell Therapy Institute, which is about a year old. and particularly with the opening of the new biomedical research building at Nova called the Center for Collaborative Research, which is a mix of for-profit and non-profit research entities. So I think that's a harbinger of what's to come in the future, this close collaboration among academia and industry. So also, as Bernie mentioned, the Cell Therapy Institute is also a close collaboration with our colleagues at the Karolinska Institute in Stockholm, Sweden. We're very honored and privileged to be working with our colleagues, many of whom are here at this meeting giving talks. So in conclusion, Nova is a proud sponsor of the summit, and we will contribute increasingly to the cell therapies mission. Thank you all very much for being here, and have a wonderful time.
You can leave, yes. Yes, you can leave. You can now leave the stage. Let's give a round of applause. Everyone likes to have a round of applause when they leave the stage. Did you know that the Regenerative Medicine Foundation has an official partnership? Our official journal is the journal Stem Cells Translational Medicine of AlphaMed Press. And tomorrow, if you haven't picked up already in your conference bag, there will be a World Stem Cell Report 2016, some of the best of the Journal of Stem Cell Translational Medicine, whose editor-in-chief is Dr. Tony Atala. So we're very pleased that we have this publication again to give you. It gives a bird's eye view of some of the great happenings in our field. Well, we have one other speaker this evening who is going to be recognized tomorrow evening with a Stem Cell Action Inspiration Award. As many of you know, the Regenerative Medicine Foundation had major outreach related to something called the Regrow Act, a visionary view of how we might be able to have a conditional pathway for FDA approvals of cell therapies. It's my worldview, our worldview, that if we treat everything as drugs and have to spend $1 billion or $1.5 billion in 10 or 15 years, we may never reap the benefits of these curative technologies. So our work with the Regrow Act and the Bipartisan Policy Center, a wonderful 501c3 organization in Washington, led us to do a conference in April where we presented scientists, patient advocates, some industry leaders to be able to understand the scope of what an accelerated pathway might mean. Of course, we're thrilled that this week the Cures Act is passing both houses of Congress, the Senate perhaps today or tomorrow. I'm not sure, I didn't get a chance to check the news. But there is in the Cures Act an accelerated pathway in recognition of regenerative medicine as it takes its place with drugs and biologics as a way to have a pathway forward to move things under the guidance, of course, and of the Food and Drug Administration. Well, one of the speakers at that meeting in April was Sandy Barker. Sandy and her husband, Gary, suffered the loss of a child. And it's a very, very moving story. But Sandy has taken that loss, as many people do, and has decided to become an advocate for pediatric cancer awareness and cures, the Gold Rush Cure Foundation. And her story was so compelling, and her mom, Jenny, is in the audience here today. Hi, Jenny. That I wanted her to share as the kickoff keynote address a little bit about, to tell us a little bit about her experiences. Let's face it, we're all in here for patients. We're all in here for a reason. Even the equine track is here for a good reason, because large animal studies have human implications. It's all important work. So I want you to give a warm round of applause to a wonderful person and a wonderful speaker, Sandy Barker. Sandy, the platform is yours.
Well, I didn't have wine either, so I have to read some of mine. But I'll get through this because I have a lot of people praying for me. Hi, my name is Sandy Barker, and I will be speaking about why we advocate for stem cell therapy and regenerative medicine. When I saw the list of speakers, I was in awe. There's some incredible people here. And while I felt a bit like I was crashing the party maybe, I realized that it is important for people to hear a patient advocate's perspective. I've been a patient advocate in DC many, many times. I have my 20th trip coming up in February, since my son passed away almost 9 years ago. But I have only talked about what I'm going to talk about tonight one other time, and that was when I was a panelist for the Bipartisan Policy Center to introduce the Regrow Act last April. Speaking about this subject is very difficult. It's very personal. So forgive me if I have to stop here and there. But I feel like it's important because it really does highlight the urgent need for all of us to advocate in moving medicine forward, namely through regenerative cell therapy. I'd like to tell you about my family's difficult journey that started 10.5 years ago. My husband, Gary, and I live in California, and we have 3 children, Christian, Garrett, and Kendall. Christian is our oldest son. We were a normal family like many of your families. Then on May 7th, 2006, Christian was diagnosed with an extremely rare and aggressive form of childhood cancer known as hypodiploid acute lymphoblastic leukemia. We were told by his doctors that his only chance of survival would be to have a bone marrow transplant. So after having a meltdown and realizing that was counterproductive, I started researching. And I researched centers of excellence for bone marrow transplant throughout the United States. And there were a lot of great treatment centers. We elected to have Christian's bone marrow transplant done at Seattle Cancer Care Alliance or Fred Hutchinson Cancer Research Center in Seattle. The doctors chose to use my son Garrett to be the donor. Both of my kids were donor matches. And they chose to use Garrett to be the donor because he was a little bit older. So we went to Seattle to have the transplant done, and it was really hard. Those of you who know about bone marrow transplant, it's very, very difficult, especially since our family was separated. I was in Seattle with Christian, and my husband was at home with Garrett and Kendall, and they would fly back and forth, and my awesome mom stayed home with my kids. But we endured it with the hope that this would eradicate his cancer forever. And after his day 100 testing, we came home thankful for our healed son. Unfortunately, only 6 weeks after we returned home, I noticed petechiae on Christian's hands and feet, and my heart sunk. I knew that petechiae was a symptom of low platelets, and low platelets was a symptom of relapse. So I took him to the oncologist the next day and just pleaded for her to tell me this wasn't true. And after some testing, it was confirmed that he had already relapsed. So we started chemo in our home state, and it was very difficult for him to get back in remission. After some intensive chemo, he did finally reach remission, and we flew back to Seattle for a second bone marrow transplant. This time we used a non-related donor since he had relapsed so quickly with a related match. The second bone marrow transplant was even harder than the first one. But Christian endured it because he wanted to live so badly. We found out before his day 100 testing that his cancer was already back again. We were devastated. The bone marrow transplant doctors decided to reduce his immunosuppressants, his prednisone, hoping for some graft-versus leukemia, which he got, but this is also when his graft-versus-host disease started. Christian's doctors work really hard at trying to balance the prednisone and increase it little by little and keep the graft-versus-host disease from progressing, but it didn't work. Even though he ended up being on massive amounts of prednisone, the graft-versus-host disease wreaked havoc on his body, especially his gut, his liver, and his skin. Christian became prednisone refractory, and all other graft-versus-host disease therapies didn't work. So his doctors petitioned for a compassionate use exception for prochimal, which are mesenchymal stem cells. We were made aware of this therapy when a very good friend of ours contacted Dr. Randy Mills, who at the time was the CEO of Osiris, who was the maker of prochimal. Dr. Mills went above and beyond trying to get the approval for our son and trying to save Christian's life. I'll be grateful to him forever. Dr. Mills and Christian's doctors worked really hard trying to get the prochimal approved. It was delayed a bit in the internal review board, but then it was approved. Then, when the request was made to the FDA, they denied it, stating that Seattle had already had one compassionate use exception and they wouldn't grant him another one, even though they knew how dire the situation was. We were devastated, as was Dr. Mills and Christian's doctors. So the doctors ended up opening a clinical trial for Christian so that he could attain access to the prochimal. But this all took time that Christian didn't have. When Christian finally did get the prochimal, it was too late. We watched Christian's body being ravaged by the graft-versus-host disease. Every day was worse than the prior. He was in horrific pain. And he was vomiting and stooling up to 4 liters of blood a day. His room literally looked like a crime scene. There are no adjectives to describe the helpless feeling of watching your child suffer so badly. The fear on their face. From seeing all the blood and their body being destroyed by a disease that may have a cure, but not being given access to that cure. It was nothing short of hell. When Christian finally did get the prochimal, like I said, it was too late. He ended up coding on Christmas morning, 2007, and he was resuscitated. We were told by the doctors Christmas morning that he would die soon. Christmas Day, we signed his DNR papers. And as hard as he tried to hold on, he died on December 29th, 2007, after a brutal 20-month battle that included over 16 months inpatient, 2 bone marrow transplants, 3 relapses, tons of chemo, total body radiation, surgeries, procedures, and finally, liver failure caused by graft-versus-host disease. Christian was cancer-free when he died. The World Stem Cell Summit focuses on stem cell therapy and regenerative medicine. I'd like to talk about my perspective on stem cell therapy and the role that it may have played in my son's treatment. Regarding the mesenchymal stem cell therapy, I truly believe that Christian's graft versus host disease would have never progressed to the state it did had he been given the therapy when it was requested. I'm still in close contact with his main Seattle Cancer Care Alliance doctors, and I know that they have used Prochymal for, I believe, 7 other patients who were prednisone refractory, not responding to other graft versus host disease therapies, which there are very few, and headed down the same path as Christian. And every one of those children had their graft versus host disease reversed, and they're alive today. And that's only one doctor. I know there are other doctors who have had the same good outcomes with the Prochymal. Cell therapy works. It doesn't mask symptoms with potentially harmful treatments. It heals. I want to personally thank every individual and company who I'm sure works tirelessly trying to advance cell therapy. Being part of the World Stem Cell Summit gives me hope. Seeing so many individuals and companies working so diligently to advance this area of medicine. It's exciting to see the progression of therapies because they're extremely effective. They cross over to so many different diseases and illnesses, and they can help so many people. Now, we need to continue advancing stem cell research and regenerative medicine, as well as make these therapies readily available to the people who need them now. I'd like to share with you what led me to make the trek from California to Florida and why stem cell therapy is so important to me. First of all, this is too close to home. There's no way I couldn't come. I truly believe in stem cell therapy, and I work really hard trying to advocate for it because I know there are so many people who need it now. Many treatment plans being utilized today are either ineffective or super toxic. Christian did everything that was asked of him because he wanted to live so badly. He literally fought till he took his last breath. But he was denied the tools that may have been able to eradicate what caused him so much suffering and what ended up taking his life. My son's case was not an isolated situation. My husband Gary and I founded Gold Rush Cure Foundation, which is a childhood cancer nonprofit after Christian died. Our focus is advocacy and patient support. One of the ways that we support patients is by mentoring families who have a child diagnosed with cancer. And I have heard stories from distraught parents that are desperately looking for a more effective, less toxic treatment for their child, and they're unable to find it. I've also heard stories from parents who aren't able to attain approval for promising cell therapies that they did find for their child who's suffering from either the cancer or the toxic ramifications of the treatment. I know the FDA has a huge job. And I know there have been improvements in the compassionate use acceptance rate, which is wonderful. Believe me, no one's happier to hear that than me. But there are still so many people being denied access to therapies that they need. I don't want others to have to suffer through what we went through. It doesn't have to be this way. I find it ridiculous that people are going out of the country to gain access to therapies where the safety and efficacy has already been proven because they can't gain access to them in the United States. But I'm afraid things won't change unless we advocate for it. If we want to move medicine to the 21st century, we all need to join forces by letting our voices be heard. Fortunately, that doesn't mean you have to go to DC all the time like I do. There are several wonderful organizations and foundations and companies who lead efforts to improve the state of regenerative medicine. There is power in numbers, and it is attainable. Some advocacy efforts are welcomed by House Senate members with open arms. They're super, super supportive. Sometimes they're not. There are several House and Senate members that we love because they are so supportive. And there are other House and Senate members that we don't love so much, but we don't give up. I'd like to tell you about one of my advocacy stories. Like I said, I'm going on 20 trips. I probably met with one House member in particular about 15 times. And one of my early trips, it was, I believe, to introduce the Childhood Cancer Caucus. And what I always do when I'm meeting with a member is introduce myself. Now I know a lot of them, but I tell them why I'm there. I educate them about the bill or caucus or whatever we're introducing. So this particular member, I said, this is why I'm here. This is bipartisan, it's bicameral, there are no appropriations attached to it, thinking we're gonna get 2 thumbs up. And he said, I won't support this, nor will I ever support anything that's disease specific because I don't earmark. Sorry, I meant to move that, there we go. So I thought, I'll work on him. So several trips later, he will not support any disease-specific drug because it's earmarking. And he's told me, if I support this, I'm gonna have some other illness that comes to me, well, you supported that, not this. And he's not alone. There are other members that are the same way. So when I came to DC to introduce the Regrow Act last April, I was really, really excited because I thought, oh my gosh, this has his name all over it, and a lot of other members as well. It's the Regrow Act, like Bernie said, is encouraging the FDA to restructure the approval process, kind of a fast track of it, and make stem cell therapies more available to the people who really need them. So when I met with him, I said, okay, I've got the perfect bill for you to pass, or for you to support. And I told him about it, and I said, I probably wouldn't have done this if it was my first or second trip, but you get a little more gutsy each time you go. And so I said, I think this is a great one for you to support. And actually, not only do I want you to support it, I want you to co-sponsor this bill. And it shocked me a little bit, he co-sponsored it. So, you always want to ask. Yeah. So advocating sometimes does take thick skin, but it's necessary and important for progression to happen. You never know who you'll get through to. I mean, even people that have told you no a ton of times, sometimes you will. Regarding why I do advocacy work, it's very necessary. It's very necessary. And I've seen too much, and I've certainly lost too much to not do something. Also, I have met some of the most amazing, passionate, driven people in my advocacy trips to DC. I can tell you, there's no greater advocate than someone fighting for a cure for somebody they love. I have the deepest, deepest respect for my fellow advocates. Speaking of what passionate advocates are capable of doing, Bernie mentioned this. This was just released Wednesday in the media. After 3 years of wrangling the House on Wednesday, November 30th, the House passed a $6.3 billion bill that would dramatically overhaul the way drugs are approved in the United States. 21st Century Cure Act is a bipartisan effort that's been in the works since April 2014. The idea behind the bill is that the Food and Drug Administration, their current approval process is clunky and out of date, delaying potentially life-saving drug devices from reaching the patients who need them. This bill aims to modernize the process, accelerate drug and device innovation by boosting funding for research and get cures on the market faster. Patient advocacy groups, drug and device companies, and more than 1,400 lobbyists have pushed for this bill's passage over the last several years. This is the first step to this happening, but it's super exciting. Going to DC to advocate and raise awareness for children fighting cancer and any other disease is definitely a marathon, not a sprint. But that's not to say that powerful opportunities to speed things up don't exist. That's why I'm so passionate about regenerative cell therapy. This is one example of achieving research gains that previously have been perceived as impossible. There's so much indication that cures are definitely plausible through research. It's a powerful message to take to our members and our federal agencies, but it's got to be followed up with educated and motivated advocacy. This is where you guys could come in. There's a lot of examples. I'm going to give you one. Children with cancer and people with rare diseases are unique. They face a battle that many in the public don't understand, including our lawmakers, but they're often overlooked. Through regenerative cell therapy and stem cell research, we have the opportunity to change the message about childhood cancer and other rare diseases, and believe that there truly could be breakthroughs that remove traditional and often ineffective and super toxic treatments of the past. This message has to be delivered by people who make these decisions. If federal agencies and Congress know about the promise for innovative therapies, like that example of the House member that I spoke of earlier, once we educate these members about this, then I know we have a chance to pass some of these bills and get more research, that kind of thing. Knowing that there's an option to cure our children and our adults through these modern answers gives me hope that we can push through the difficulty and objections to those who either don't understand or are afraid of the risks. I have a few takeaways that I would like to share with you, and what I'm hopeful will happen with regarding advocating for regenerative cell therapy. So this is kind of true. Cancer definitely brought me to my knees more times than I could count while my son was battling cancer. But it's also brought me to my feet to advocate for those who deserve and desperately need better therapies, but aren't able to advocate for them themselves. I challenge each company and representative here to look at how you can help advocates like me be more empowered and educated, and offer a path forward to those who desperately need it. By looking at how you can be part of the solution, as well as how we can bring research innovation to the bedsides of those who need cures now, will improve the lives of everyone. I also want to speak to the companies and organizations who financially invest in these treatments. We need you. We have to find a way to keep going, and that should and could include partnerships with federal agencies and efforts in Congress. Continue to communicate with advocates and ask them for their help to secure a path forward. We are a driven group. Utilize us. And as a parent and a founder of a Patient Assistance Foundation, I know how much this matters. Advocacy is vital if we want to make a change. And it needs to start now, because patients can't afford to wait. Patients are suffering and dying now. They need treatments now. There needs to be more advancement in stem cell research and an improved approval process to help people and families who desperately need this. This can happen if we all make our voices heard. My hope is that the legislative leaders will see the vital need for improvement for stem cell research and regenerative medicine by hearing stories like mine. I am here representing so many people. This is not about my son and my family. I'm representing so many people. My question to all of you is this: if your child or mother or father or brother, sister, loved one, friend had an illness and there is a therapy that may improve their quality of life, may cure their illness, and may even save their life, wouldn't you want that for them now? And wouldn't you want research to continue to progress for treatment for that? We do have this available to us, but we need the FDA and our legislative leaders to help us advance technology and make them available to those whose quality of life or life depends on it. It's too late for my son Christian, but it isn't too late for the numerous people with various different medical conditions who could be helped by stem cell therapy. I don't want other kids and adults to have to suffer and die like my son did. There are also countless other people who aren't even diagnosed with their illness yet. It could be someone you love or I love. This is what motivates me and my foundation to advocate for stem cell research and regenerative cell medicine. I urge you to please join our efforts. Thank you.
Thank you so much. Thank you, Sandy. That was an incredible talk, very inspiring to all of us here from our heart. And we'll be many of us will be watching you receive an Inspiration Award tomorrow night at the Stem Cell Action Award dinner that you richly deserve. I want to thank all of you for attending this evening's session. We've not done this before, kicked off the meeting in the evening, but I think we've all learned something here. I look forward to seeing you over the next 3 days. If you were planning to leave early, cancel the flight and extend it, because this meeting is an incredible meeting. It's an opportunity for you to learn, to forge collaborations, expand your knowledge, and hopefully deliver cures. Thank you, and I'll see you tomorrow morning.